Hi guys, am I audible? I hope I'm perfectly audible. Thank God, thank God. Okay, so people, am I audible to all of you? Am I audible to all of you? Hi everyone, am I audible to all of you? Hello? Am I audible? Hi everybody, am I audible to you? So welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma and let's start the session today. I'm sorry for the delay. My internet was not working and I was struggling with that part only. Right, internet was not working, so I was not able to start the class on time, and that is why I shifted it from 7 to 8. Right, now it's working fine. As of now, so it's working fine. Right, and it should. It should. This is what you have to pray today. Right, this is what you have to pray today. Okay. Okay, so do it guys quickly. Let's start the session. Let's start the botany part. And if you are regularly following our sessions, you know it very well that even in the zoology portion, right, bache, the part first zoology, basically the part first of biology, that is your zoology part, we have completed few chapters, and from that few chapter the most important topics are pending, right? Some of them are completed in the top 50 concept series. Okay, some students, they are still requesting now that we want the sessions, we want that top 50 sessions, top 50 sessions, top 50 concepts, rather, that particular sessions. So in the top 50 sessions I have completed many important topics, top 50 topics rather. In that particular session I have completed many topics, so you people can pick that up, number one. Secondly, we will complete it here, and today we have to increase our speed. We have to move very fast so that we can finish the remaining 1 or 2 chapters of zoology as well. Okay, and I have already shared the notes in the Telegram group, right? So quickly everybody hit the like button first of all here. Hit the like button and share the session with your friends. Tell them ma'am has started the class and we are all set. So keep a pen and paper with you all. Whatever are the most important points, you will just write down in the form of very, like, you have to write it in a very precise manner, right? Right, so that you can revise it very quickly. This is what you have to do. Okay, this is what you have to do. So everybody just hit the like button, right? Hit the like button, and you people know it, right, that this time you have the biggest offer. The 60% off is there, plus extra 2 months extension. So in the description box there is a link for my batch, and you guys can be the part of my batch as well. Okay, chali. So let's start the session, and that session is basically, what is that session? That session is this. What are we going to start first? We are going to start the kingdom Plantae first. So of course, because we are picking up kingdom Plantae, it is very important chapter. That is why I'm picking it up first, because we can expect 3 to 4 questions from this part directly. Okay, so I have already shared the sequence of the chapters in my Telegram group, in our official Telegram group as well. So let's do it quickly. Okay, so tomorrow it's Sunday. Tomorrow is Sunday. So today I am in full mood that I am going to take the lecture, even for 11 hours or 12 hours, whatever is required. Okay, whatever is required, right? I will take it. I'll take it as per that. But yeah, if students will not be there in the session, and if they are like, okay, ma'am, we are leaving in between, we'll watch the recorded, I'll end it too. So it depends upon the students. So I want you people to be consistent, and definitely, you know that comparatively we have less chapters, so we can even cater that few topics from the zoology part which we were not able to complete in the last session. Okay, or otherwise you can watch our top 50, right? Otherwise you guys can watch our top 50 concepts video. Okay, so let's get started. Let's start with the kingdom Plantae. So because we are starting with the kingdom Plantae, right? So I'm giving you a basic introduction from biological classification part as well. So if you remember, who gave the 5 kingdom classification system? R.H. Whittaker. It's a direct MCQ. Who gave the 5 kingdom classification system? R.H. Whittaker. So of course we start with the Monera. Then we talk about Protista. Monera is there. Protista is there. Fungi is there. Plantae is there, and then we have Animalia. What do we have then? We have Animalia. That is what we have. Monera, Protista, right, fungi, Plantae, and Animalia. That is what we have. Okay, that is what we have, number 1. Keep it in your mind. Now we are talking about kingdom Plantae. Okay, we are talking about kingdom Plantae. We are talking about the plants. So basically, you know that they are multicellular. Right, now you will be like, in the algae, madam, we discussed chlorella, that is unicellular. Okay, fine, unicellular algae is there, right? But as per Whittaker, it is there in kingdom Protista, but it's just that its life cycle is similar to, okay, right? Its life cycle is what? Its life cycle is similar to the algae, so we study it there. Okay, 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 done. So multicellular, autotrophic organisms, am I right? Autotrophic organisms, and of course eukaryotes, they are without any doubt. Multicellular, eukaryotes, autotrophic organisms, that they are having cell wall, and that cell wall is having the cellulose. Okay, it is having the cellulose. Other than that, we know it very well. Hemicellulose is there, right? Pectin is there. There are certain proteins as well, but this is something we have to keep in our mind. Am I right here? This is something we have to keep in our mind. Am I right here? Am I right here? So this is what we have in the kingdom Plantae. This is what we have in the kingdom Plantae. Now when you talk about the kingdom Plantae, students, right, here in kingdom Plantae, we need to read about algae, bryophytes, pteridophytes. Then beta we will go for gymnosperms, and we will go for angiosperms, right? Angiosperms. So quickly tell me in the chart section what we have to read. We have to go for the algae, bryo, pterido, gymno, angio. Algae, bryo, pterido, gymno, angio. Algae, bryo, pterido, gymno, angio. Quick, quick. Done? Done? So what we need to read? What we need to read? We need to read algae, we need to read bryo, we need to read pterido, we need to read gymno, we need to read angiosperms here. So how are we going to cover this chapter? Beta, we will firstly start with the life cycles. If you think that life cycles are not the part of syllabus any more, but trust me, if you don't understand the life cycles, you will not be able to understand this part. Okay, so I'll move quickly here. I'll start with the life cycle, then I'll pick up that taxonomy part which is given in plant kingdom initially. Okay, so when you talk about the life cycles here, right, when you talk about the life cycle here, what we discuss? Life cycle. Bache, we talk about the haplontic life cycle. Okay, we talk about the diplontic life cycle. Okay, and we talk about the haplo-diplontic life cycle. That is what we have to discuss at this point of time. Okay, we have to discuss about the haplontic, diplontic and haplo-diplontic life cycle. Please keep it in your mind. Okay, so life cycle, or you can say alternation of generation, is there. What is there, students? Alternation of generation is there. Alternation of generation is there. So when I'm saying alternation of generation, like even in the animals, we see that the sexual stages or asexual stages, they alternate, right? Sexual stages and asexual stages, they alternate. Yes or no? Right? Yes or no? Do they alternate? Definitely they do. Definitely they do. And here also in the case of plants, we'll see in their life cycle haploid and diploid phase, right? It is the haploid and diploid phase that will alternate. Okay, so basically for the alternation of generation, we use the word, right? What is the word that we use for the alteration of generation? Basically, beta, we use the word, right? We use the word, yes, anybody here in the class? We use the word metagenesis as well, right? If you people remember, we also use the word metagenesis. Do you remember that? Do you remember that? Yes or no? Do you remember that? Yes, do you remember that? So this is the word that we use. Okay, this is the word that we use. So it is the alternation of generation. Haploid diploid phase will alternate. So we have three life cycles: haplontic, diplontic, haplo-diplontic. In the case of plants, there are two words that you need to keep in your mind, right? One word, like these are the two words that you need to keep in your mind. Like you have gameto. You have one word is gametophyte, and one word is sporophyte. Firstly, in a very simplified way, I'll explain you this. Whenever you talk about the gametophyte, it is haploid. It forms gamete. What will it do, bache? It will form gamete. This is the part that you need to understand, and the entire topic is going to be very simple for all of you. With the help of tricks, we are going to revise it. So I want you people to pay attention here. So gametophyte is there. It forms gamete. Then sporophyte, 2N. Sporophyte. 2N. It will form spores. It will form spores. So always remember, your gametophyte will form gamete. Gametes are haploid. Your sporophyte will form spore, and even spores are also haploid. So the very first thing that we have is haplontic life cycle, right? And algae are going to show that, right? In which organism you people will see this? Algae. In which organism you guys are going to see it? You will see it in algae. And students, right, today itself I have posted one video on Unacademy NEET English channel, and that is the best revision video that you people can get these days. Okay, in this particular revision video, I told you that how to divide your syllabus, what to prioritize, what not to prioritize, how many mock tests you need to give, right? How to analyze that, the importance of mock tests, everything we have discussed. So I was just reading the comments, and students are like, there is some issue with the voice. No, this is some YouTube setting. Okay, so you can click on that original audio thing. Okay, you can get that original audio thing. So you can go for that. Okay, you'll get my original voice. Right? I was reading the comments. Many students were like, ma'am, your own voice is good. So there is a voice over there. No. It's not like that, right? You can just go to the setting part here. There is some issue. There is some setting thing is there. Just say audio track 2. This is what you have to select. And on the basis of that, you'll get my original voice. That's it. Okay. So do watch this video. It is very, very, very important for your revision. Okay. Okay. So haplontic life cycle, who's going to show it? Algae. Who's going to show it? Algae. But when you talk about the humans, okay, when you talk about the humans, or you can take any other organism as well. So in the case of humans, but whenever you will see any of our body cell, we are diploid organism, right? We are developed from diploid zygote. We all are diploid organism. What are we? We all are diploid organisms. What are we? We all are diploid organism. So in our body, if you will check any of the cell, it is going to be diploid. In our body, if you talk about the cells, what are they going to be? They are going to be diploid only. But in the case of humans, like mainly, of course, we are the diploid organism. We have 46 chromosomes, right? How many chromosomes do we have? 46 chromosomes. Now here, right here, please keep it in your mind. Now when you talk about the gametes here, in our case, our gametes are haploid only. In our case, our gametes are haploid only. Are you getting my point? In our case, our gametes are haploid only. Our gametes are haploid only. Otherwise, all our body cells are diploid, right? So basically, we are a diploid body. We are a diploid body having haploid gametes. Now, in the case of plants, that is not true always, right? That is not true always. This is something we will see in the case of animals, not in all the animals, of course, right? This is something we will see in the case of animals, right? This is something that we will see in the case of animals, okay? In the case of animals. But which test link are you talking about, right? And why are you spamming here with the test link part only? Yes, please. Yes, please. Which test link are you talking about? Right, you can just have access it again now. That is in, right, I have shared it in my Telegram group. You people can check it. Okay, right? So now let's focus here. So now I'm talking about the algae. When I'm talking about the algae, remember this thing, you know, na, algae, they have thallus-like body. So when we say thallus-like body, what is the meaning of that? What is the meaning of that? They are not differentiated into, what is the meaning of that? That they are not differentiated into true root, stem and leaves. They are not differentiated into true root, stem and leaves. They are not differentiated into true root, stem and leaves. What do we call it? We call it as algae, thallus-like body, thalloid structure it is, not differentiated into true root, stem and leaves. So just imagine this is thallus, right? This is your algae ka thallus, okay? This is your algae, and when you talk about the algae, of course, as I said, it is not differentiated into true root, stem and leaves. So in the case of algae, if you talk about any of the cell, my dear students, that cells are going to be haploid only, right? Here the main plant body, right? Here the main plant body, or I would say the dominant phase, which will be there for the majority of the lifespan, right? So main plant body or dominant one or the photosynthetic one or the photosynthetic body. It is haploid. It is haploid. Main plant body is haploid. So I told you, when I'm saying haploid, can I use the word gametophyte here, my dear people? Can I use the word gametophyte here? So that is why even it is the haplontic life cycle, because haploid phase is there. Right, bache? Right, bache? Because the haploid phase is there. And that haploid phase is there for the longer period of time. Right, because the haplontic, and what is happening here, the haploid phase is there, and that haploid phase, right, that haploid phase is there for the longer lifetime, right, for the for the majority of lifespan. Mostly it is a haploid phase. So if haploid phase is the dominant one, what is the word that we will use? What is the word that we will use? We will use the word gametophyte. If haploid phase is the dominant one, then what is the word that we are going to use? We will use the word gametophyte. We will use the word gametophyte. So main plant body, the photosynthetic one, this green one is the gametophyte, means it is haploid. Now, it is a gametophyte. Please keep it in your mind. It is a gametophyte, student. It is already haploid. What is it? It is a gametophyte, people. What's that? It is a gametophyte. It is already haploid. It is a gametophyte. It is already haploid. So now, gametophyte means it forms gamete. Sporophyte means it forms spores. So if it is a gametophyte and if it forms gamete, if it is a gametophyte, it is forming gamete, it is already haploid. So it will mitotically, right? Mitotically, it is going to form the gametes. Mitotically, it is going to form the gametes. Mitotically, it is going to form the gametes. Now, sporophyte is diploid. Even sporophyte needs to form the haploid spores. So it is already diploid. If it has to form something which is right, it is diploid if it has to form something which is haploid. So the meiosis will be the division. So here in this portion, anybody having any doubt? Here in this particular part, anybody is having any doubt? Anybody is having any doubt? Yes? Yes? Fine now? Okay, chaliye. So, it is the gametophyte. What this gametophyte will do? It mitotically, right? Mitotically. That is the equational division. Mitotically, it will form haploid gametes. So, there will be, right, male gamete, female gamete. They'll fuse. The fertilization will be there. And you will get the diploid zygote. You will get what? You will get the diploid zygote. I'm talking about the haplontic life cycle first. If you will understand gametophyte, sporophyte initially, trust me, the rest of the topic is going to be very easy. One question will definitely come from this part. One question minimum. Two questions can also come. Okay, two questions can also come. So please pay attention. Okay, so what are we saying? We are talking about the algae. I am saying undifferentiated body. It is mitotically, it is going to form the gametes. So now these gametes, what these gametes will do? Now these gametes, they'll fuse, they'll form zygote. Now, but beta, this zygote will undergo meiosis. It will undergo reductional division. Yeah, immediately it will undergo reductional division. Because this zygote is undergoing meiosis, we call it as zygospore. What do we call it? We call it as zygospore. Why do we call it zygospore? Why do we call it zygospore? Right, we call it zygospore because here the zygote is undergoing meiosis. This zygote is undergoing meiosis here, right? This zygote is undergoing meiosis here. So that is why we are calling it as, what? We are calling it as zygospore. So it will immediately undergo meiosis. You know that when it is the meiosis, it is the reductional division. If you know it, very good. If you don't know it, let me tell you this thing, right? And let's have a very quick revision there. Just a minute. Yes. So let's have a very quick revision there. So what I just said, it is meiosis. So in the meiosis, there is a diploid cell, will undergo meiosis 1. 2 haploid cells will undergo meiosis 2, will get 4 haploid cells. This is what meiosis is. So now here in this particular case, what is happening? You have zygote. What do you have? You have zygote. What do you have? You have zygote. This zygote which is diploid, I'm calling it as zygospore. So I am saying it will undergo meiosis. So of course, if it is undergoing meiosis, it is forming these haploid spores. So these haploid spores, because they are the result of meiosis, we also call them as meiospores. We also call them as meiospores. Right, we also call them as meiospore. So it is the zygote only that will be the, it is the zygote only, right, that is diploid. Rest all are haploid here. Rest all are haploid here. Is that clear? Is that clear? So here you can see that zygote is the only cell of sporophytic generation. Then of course these haploid meiospores will form, as I said, right? These haploid meiospores will form. Now when these spores will germinate, means when they will grow again, right? When they germinate means when they grow again. So of course, because they are haploid, mitotically they will again form the haploid plant thallus. This is the haplontic life cycle. Which life cycle it is, bache? Which life cycle it is? It is the haplontic life cycle. Just have a look here. Just have a look here. So dominant phase is the haploid phase. Which phase is the dominant phase? Haploid phase is the dominant phase. Which phase is the dominant phase? Haploid phase is the dominant phase. Is that clear? Is that clear? So here you can see it is the zygote, right? You can write down here in this diagram only, right? I'll share the PDF in our official Telegram group as well. So this is the zygote, beta. It is the only diploid cell in the life cycle of this haplontic life cycle, right? In the case of the algae, only diploid cell, or I would say it is the only sporophytic cell, only cell of sporophytic generation. Okay, it is the only cell of it is the only cell of, yes, it is the, I'll write it here. So it is the only cell of sporophytic generation. Are we clear here? It is the only cell of sporophytic generation. So when I'm saying sporophytic generation, I am talking about the diploid phase here. I am talking about the diploid phase here, right? Right, so sporophytic generation is represented only by one celled zygote. That is an important question because we are also discussing the NCERT, we are also discussing the algae. So definitely this part is important for us. So we are saying that there is no free living sporophyte because this sporophyte, it is the zygote only. It depends upon gametophyte. So here sporophyte is not free living. So that's how you can also make your short notes. Understand it, make your short notes. So meiosis in the zygote will give us haploid spores. These spores will divide mitotically and you will get the gametophyte. What will you get? You will get the gametophyte. Is that clear? So the dominant photosynthetic phase in such plant is the free living gametophyte. What's that? It is the free living gametophyte. What's that? It is the free living gametophyte. Am I clear here? What's that? It is the free living gametophyte. So this kind of life cycle is haplontic life cycle. Algae will show this, and yeah, few algae they also show haplo-diplontic or the diplontic phase. So these are the examples that you need to keep in your mind. Yes, these life cycles are important. These life cycles are important. Now let's move to the next part, and that next part is your, right, that next part is your diplontic life cycle. Which phase do we have? Yeah, which life cycle do we have? It is the diplontic life cycle. It is the diplontic life cycle. So when I am talking about the diplontic life cycle, okay, diplontic means diploid. So obviously diploid phase is dominant. Diploid phase is dominant. What is dominant? Diploid phase is dominant. Diploid phase is photosynthetic. Diploid phase is dominant. It is photosynthetic, or you can, of course, if it is photosynthetic, it is independent and it is the one which is long-lived, right? It is there for the majority of the lifespan. Okay? Okay? Is that clear? Everybody, is that clear? Sure? Sure? Okay, so do remember it. It is also required for class 12 sexual reproduction in flowering plant chapter. So let's suppose when you are talking about the diplontic life cycle, we will see it in the case of gymnosperms and angiosperms. Where are you going to see this? You are going to see this in Gymnosperms and Angiosperms. Where are you going to see this? Gymnosperms and Angiosperms. So when we talk about the Gymnosperms, just say here we have the plant. So it is the sporophytic plant body. What's that? It is the sporophytic plant body, students. What's that? It is the sporophytic plant body. Sporophyte it is. It is diploid. Okay? It is sporophyte. It is diploid. Is that clear? Is that clear? So it is the sporophytic plant body. It is the sporophyte that you need to keep in your mind. Okay? That you need to keep in your mind. This part is clear. Now, this is the sporophytic plant body. So, of course, here, right, just say whenever we talk about the sexual reproduction, sorry for that. So, whenever we talk about the sexual reproduction, so you know that in the case of sexual reproduction, people, right, in the case of sexual reproduction, 1 minute. Yes, so it is the sporophytic plant body. What kind of plant body it is? It is the sporophytic plant body, right, bache? Sporophytic plant body. It is the sporophyte, diploid phase. So, of course, if you will relate it a bit with the sexual reproduction in flowering plants, you remember anther, filament, stamen, your stigma style and ovary part, right? So basically you have microsporangium, megasporangium. So I'm not entering the depth of that, right? I'm just telling you this thing here, right, bache? Yes, bache. I'm just telling you this is the sporophyte, which is the main plant body, which is the photosynthetic one. Okay, okay, which is the photosynthetic one. And here it is going to form, bache, spores. What are that spores? Spores are haploid, of course. So this sporophyte, with the help of course, with meiosis, it will form spores. So now, if you will ask me, right? If you guys will ask me that, madam, this entire this complete plant structure is going to form the spores. So again, as I said, right, as I said, yes, people, something is going on, my internet and everything. Is there any glitch? One second, my laptop is not working properly. Let me refresh once. Students, let me restart. Students, am I audible now? Hi everybody, am I audible? Now am I audible? Thank God. Thank God. Right, I was literally not interested in taking this session again, right? Let's finish it today only, and then let's move to the class 12th. Hannah, let's move to the class 12th. So I hope I'm perfectly audible and visible. Something happened to my laptop. I don't know what was that. Right. I don't know what was that. Something happened and like it was all black. Okay. Done. So all I'm saying is that this is the sporophyte. Okay, this is the sporophyte. So this is the main plant body, sporophytic. It means it is diploid, and then the spores will form. So students, your spores are haploid. Just keep it in your mind. These spores are haploid. Like if you remember sexual reproduction in flowering plants, we discuss microspores, we discuss megaspores. right? We discuss microspores, we discuss megaspores. So because we discuss microspores and megaspores as well, microspores and megaspores as well, right? If you can relate it. So here basically meiosis will occur. After that reductional division, the spore formation will be there, and this spore is going to form gametophyte. Remember, microspores, they form the pollen grain, which is the gametophyte. So because this spore, it develops from this gametophyte, it develops from the haploid spore only. It is itself haploid, and mitotically, with the help of mitosis, it will form gametes. Okay, it will form gametes. And here, when we talk about the gametes, they will fuse, they'll fertilize, they'll form zygote. That zygote will also grow mitotically, and after that you will get a multicellular sporophyte, and that will grow further. Okay, that will grow further. So how can we compare it? How can we revise it? Okay, how can we compare it and how can we revise it? It's simple. In the case of haplontic life cycle, the story was simple. The main plant body was gametophytic. It was haploid. So if it is a haploid, so of course if it has to form the gametes, it will go for mitosis. Mitotically, it will give us the gametes. That gametes will fuse, they'll fertilize, you'll get the zygote. But here the point is, zygote is the only diploid cell in the case of haplontic life cycle. Zygote is the only diploid cell, right? It will immediately undergo meiosis. That's why we call it as zygospore as well, and then these haploid spores will germinate again, they'll form the haploid gametophyte. That's it. Now let's come to the next part where we are talking about the sporophytic generation, diplontic life cycle. So it is diplontic, so of course it is the sporophyte which is the dominating one, and the photosynthetic one. And here the gametophyte, right? The gametophyte, if you ever make mistake here, just think of pollen grain, which is the male gametophyte. Just think of embryo sac, which is the female gametophyte. So of course, they are dependent on the sporophyte only. They are dependent on the sporophyte only. So let's have a quick revision of that. Let's have a quick revision of that. So diploid sporophyte is the dominant photosynthetic and independent phase of the plant. It is the diploid. Yes, bache. It is the diploid sporophyte is the dominant photosynthetic and independent phase of the plant. So gametophytic phase is represented by single to few celled haploid gametophyte. Right, it is represented by single to few celled haploid gametophyte. So this type of life cycle is known as diplontic life cycle. So in the case of diplontic life cycle, so you can see the gametophyte is represented by single to few celled haploid gametophyte. That is the MCQ that you need to remember. That is the PYQ that you need to remember. Okay. So where are we going to see this? In the case of gymnosperms, in the case of angiosperms, and of course the exceptions are there. So exceptions are very important. Please write it down. Whatever points I'm writing extra, you can directly write it in your NCERT, and you can directly write it on a white paper, and you can use them for revision. So exception may write fucus. Fucus is what? It is an algae. It is an algae, and this algae, instead of haplontic life cycle, it shows diplontic life cycle. It shows diplontic life cycle. Is that clear? Is that clear? Are you sure? I hope this is clear. So in addition, all seed-bearing plants, gymnosperms and angiosperms, they follow this pattern with some variations, wherein gametophyte phase is few to multicell. Now the next is your haplo-diplontic life cycle, as the name is suggesting. So here both the phases are present. So your bryophytes and pteridophytes are going to show that. Who's going to show that, students? Your bryophytes and pteridophytes. Who? Your bryophytes and pteridophytes are going to show haplontic diplontic life cycle. Now in the case of bryophytes, please remember it. Now, in the case of bryophytes, my dear students, the haploid phase, that is your gametophytic phase or the gametophyte is dominant. It is dominant. It is photosynthetic. It is long-lived. Okay, so this one is dominant, this one is photosynthetic, long-lived. Sporophyte, when you talk about the diploid sporophyte here is multicellular, not like haplontic life cycle single-celled. It is multicellular, but it is wholly or partially, matlab completely or partially, right? It is completely or partially dependent. It is completely or partially dependent on gametophyte. So it is taking its nourishment from gametophyte. It is partially or completely dependent on that. Now let's talk about the pteridophyte. Let's talk about the pteridophyte. Here the dominant phase, the one which is for long duration, right? And even here also short duration is of sporophyte. Long duration, dominant phase, long duration, photosynthetic, independent is your sporophyte, diploid phase. Which phase? Diploid phase. Which phase? Diploid phase. It is the dominating one. Okay, it is the dominating one. Okay, but here one thing that we all need to remember is, but here gametophyte, though it is short-lived, is green and photosynthetic. So just like gymnosperms and angiosperms, in pteridophytes also, it is the sporophyte which is the dominating phase. It is the sporophyte which is the dominating phase. It is the sporophyte which is the dominating phase. But here the gametophyte is also green. It is also photosynthetic, and so basically it is not dependent on that sporophyte. So that is what you need to remember when you talk about the haplo-diplontic life cycle. So let's have a quick revision from the NCERT as well. Right. So a dominant, independent, photosynthetic thalloid phase is basically the gametophyte in the case of bryophyte. So it will alternate with short-lived multicellular sporophyte. So this is your question. So in haplontic life cycle, people please remember this thing. In haplontic life cycle, this sporophyte is single-celled, unicelled. It is single-celled, unicelled. But in haplontic life cycle or even in diplontic life cycle, it is one or few cell stage. But here it is directly multicellular. Okay. Okay. Then the diploid sporophyte. It is, yes, Vache, the diploid sporophyte, it is represented by a dominant, independent, photosynthetic, vascular plant body. Right? Vascular plant body. So please pick up these points. Vascular plant body. So actually pteridophytes are the first plants where you will see the vascular bundles. Okay. So it will alternate with multicellular sporophyte. Right? It will alternate with what? It will alternate with multicellular sporophyte. Right? So sporophyte is, so it alternates with multicellular sporophytic, yeah, autotrophic, independent. It alternates with multicellular sporophytic, autotrophic, independent but short-lived haploid gametophyte. So main is it's saprophytic. I'm by mistake I was saying sporophytic. No, it's saprophytic. So diploid sporophyte is the dominant and independent phase, and it alternates with, again, here gametophyte is multicellular. It is saprophytic. It will absorb its nutrition. Right? It is going to absorb the organic matter. Right? That's why the word is saprophytic here. It is independent, but it is short-lived. So here the gametophyte is also independent. That can be asked as a question. So in bryophyte this, in pteridophyte this, but yeah, exceptions are there. Right? Just say ectocarpus, polysiphonia, calps, they are haplodiplontic. These are the algae which are haplodiplontic, and fucus is an algae which is diplontic. Question can come from the exceptions as well. Please write down. Please write down. Quick, please write down. Okay, done. Okay, so now what we have to do, we have to pick up the another important points. So students, I always mention this thing, where whenever things are repeated, they will definitely come in paper. Just here again, they're talking about the Whittaker. We have already discussed that. So when we are talking about the Whittaker, do not forget the year as well. R.H. Whittaker, an American taxonomist, in 1969, he was the one who gave the 5 kingdom classification system. So is it important? Of course, it's a previous year question. It's a previous year question. Okay. Okay. So now you're talking about the plants and see here, these are the general things. Okay. Just say you know that in 2 kingdom classification system, your algae, blue-green algae, right, on the basis of cell wall, they were all placed together in Plantae. But here in R.H. Whittaker classification system, unicellular eukaryotes, they were in Protista, rest were there in this Plantae part. Okay. But what we need to mark here, are we going to mark this part here? Are we going to mark this part here? No. We have to talk about the systems of classification. So please with me, write down these keywords. So first is your artificial system, right? So basically these, right, what we have to discuss the classification system, and here we are talking about the artificial system first of all. What are we talking? We are talking about the artificial system first of all. So in artificial system, what will be considered? Yes, students, what will be considered? Gross, right? Superficial, that is superficial morphological characters, habit, color, number and shape of leaves. Means here the scientists, they just saw the morphology, the outer appearance, the number, the habit, the shape of the leaves, and they are like, right, they are like, okay, fine, classify the organism. So what are we saying? That it was mainly based on vegetative character or on the Andrecium system, and this was given by Linnaeus. So all the points that I have underlined here, they are important. One question is expected from this part. Okay, one question is expected from this part. Is that clear? Is that clear? Yes or no? I am going to pick up the most important topics only, and that is why I request you people to please write it. Okay, kindly write it. Okay, so first thing that you can see here, artificial system of classification. So when we are talking about the artificial system of classification, people, the one thing that you have to remember here is that it was based on the morphological character. Kindly mention it in the chat section, please. Kindly mention it in the chat section. Quick. Yes, please. Done? Do write it down, it is important. I hope it's done. I hope it's done. Okay, this is important. Number one. Okay, so because it was based on vegetative characters, right, that can change. Okay, so this system of classification, fine bache, so mainly on the basis of vegetative characters, mainly on the basis of vegetative character, this system of classification was. Okay, done? So next point is, see, the artificial system, what is the drawback? This is the main drawback here, equal weightage to vegetative and sexual characters. Okay, equal weightage to vegetative and sexual character. Okay, and we know that vegetative characters are very easily affected with environment, so it was not accepted. So this is the artificial system. So next is the bit of natural system. So when you are talking about the natural system, it is also known as 2D system of classification. Which system? 2D system. So here what they considered, it was not just on the basis of, right, which were based on. So you can read it here. This natural system, it was developed, it was based on natural affinities, means similarity among the organisms, and consider not only the external feature. It means along with external features, they also considered the internal features. Is that clear? Right, along with external features, they also considered the internal features. Okay, they also considered the internal features. So when I'm saying internal features, ultrastructure, anatomy, embryology, phytochemistry, everything will be considered. Okay, so who gave it? Bentham and Hooker. Bentham and Hooker. Scientist name is important. Of course it is important. Done? Of course it is important. Done? Yes, of course it is important. Chali, next system is phylogenetic system, which is also known as vertical system. So when we are talking about the phylogeny, we are considering the evolutionary relationships. What are we considering? We are considering the evolutionary relationships. What are we considering, my dear students? We are considering the evolutionary relationship. That's something you have to keep in your mind. So at present, the phylogenetic classification system, it was based on evolutionary relationship, right? Evolutionary relationship. So between the various organisms, and it was acceptable. Okay. Okay. So as per that, all the organisms which are belonging to same taxa, if they belong to same taxa, you understand now? We are talking about the taxonomic categories here, right, of any rank. So they are saying it is belonging to the same taxa, having a common ancestor. Is it clear? Having a common ancestor. Is that clear? Is that clear? So this is something you need to keep in your mind. Okay. So phylogenetic system, it is also known as vertical system, and yeah, you can also call it as 3D system. So same way artificial system, a natural system rather, it is also known as horizontal system. Okay, it is also known as horizontal system. Then numerical taxonomy, as the word is saying, right, numerical numbers you are considering. So basically they are saying it is carried out by computers and it is based on all observable characteristics. Right? So numbers and codes were assigned to all the characters and the data were then processed. Understood? Numerical taxonomy. So for numerical taxonomy, phonetics is also the word that we can use. So it is also known as taximetrics. It is also known as taximetrics. So that's something you have to keep in your mind, okay? That's something you have to keep in your mind. Is that clear? Is that clear? So numerical taxonomy, it is easily carried out using the computers, right, bache? So it is based on all the observable characters. Is that clear? Is that clear? Now, the next, so in this way, each character was given equal importance, and at the same time, hundreds of characters can be studied can be considered, okay? 100 of characters can be considered. So up to this part, all clear? Up to this part, all clear? Are you sure? Done? Done. So then comes what? Then comes the cytotaxonomy. Cyto word is associated with cell, bache. So because it is associated with cell, so it is based on the cytological information. So cytological information, chromosome number, structure, behavior is there. Chemotaxonomy mein all the chemical constituents of the plant. So when it comes to chemotaxonomy, cytotaxonomy, numerical taxonomy important, also known as phonetics. And as I said, we can carry out by using what? Computers, right? So so basically we will consider all the observable characters and numbers and codes are assigned to that. Numbers and codes are assigned to that. That's what you have to keep in your mind, right? Chaliye, so life cycles done, this taxonomy part done. 2 questions are expected from it. Now when we talk about the algae, as I said, we have to go very fast, right? We have to go very fast. So we have to pick up the most important things here, right, bache? So in the case of algae, one thing that I told you, and yes, I would like to make a flow chart here, right? I would like to make a flow chart here. And of course, you know that these are the flow charts which are very, very, very essential for the quick revision. Am I right? Am I right? Am I right? Am I right? Sure? Sure? Done? Chaliye. So let's make that flow chart, and what we have to start, we'll start with the embryo stage first. So basically, which kingdom do we have? We have kingdom Plantae. Which kingdom do we have? We have kingdom Plantae. Now in the case of kingdom Plantae, first of all, pick up the embryo part. Embryo absent, embryo present, right? It is the embryo absent, embryo present. So now embryo absent, it is there in algae. Now if you think how, let's have a quick revision. Let's have a quick revision. In the case of algae, right? In the case of algae, what you have studied? Green algae, green algae, brown algae, red algae. Okay, okay. So we said, right? This is what we have discussed, that your diploid sporophyte will undergo meiosis. Matlab zygote is the only cell of sporophytic generation and that will undergo meiosis. So if zygote is undergoing meiosis and giving the haploid spore, so it means it is not developing further to form the embryos, right? It is not developing further to form the embryos. So that is the point here. Now here comes what? Here comes embryo present. So of course, embryo is there in all other, right? Embryo is there in all other, right? It is there in all other plants. Is that clear? So next thing that we have to pick up is the vascular tissue, right? What is the thing that we have to pick up? Vascular tissue absent. So vascular tissue, I'm talking about the xylem phloem here. So it is the bryophyte. So how can you revise it? How can you revise it? Yes, please. How can you revise it? You can simply revise it. In the algae, embryo is absent and the vascular tissue is absent. But in bryophyte, embryo is there, because here sporophyte is multicellular, but vascular tissue is absent. So when I'm saying vascular tissue present, so the plants, right? So the plants having vascular tissue, they are known as tracheophytes. So the plants having vascular tissue, they are known as tracheophytes. Tracheophytes. What are they? They are known as tracheophytes. Right? They are known as tracheophytes. So the very first thing that we have considered is this plants, vascular tissue. Then beta, we'll talk about the seeds. If seeds are absent, okay, or if seeds are present, if seeds are absent, or if seeds are present, right? Or if seeds are present. So seeds are absent, right? Seeds are absent. So if seeds are absent, so you are talking about the pteridophyte here. So what will I say? That it is a kind of plant which is not having the seeds, but yeah, it is having the vascular tissue, it is having the embryo. So seeds absent, what is the word for such type of plants? The word is cryptogams. That's what you have to remember. So if seeds are present, the word that we have to use is phanerogams, and along with phanerogams you can also call them as spermatophyta. Spermatophyta. Okay, it is spermatophyta. So here seeds covered and seeds naked. So that's the complete flow chart here. So seeds covered, seeds naked. Okay, so seeds covered and seeds naked. That's what we have. So when it is naked, you are talking about the gymnosperms, because they are without flower. So seeds, that's why they are not covered. Ovule is exposed. When seeds are covered, you are talking about the flowering plants, you are talking about the angiosperms, and in the case of angiosperms, you know that seeds are covered. So this is something you have to keep in your mind. This is regarding your kingdom Plantae. Okay, so now come to this part. Do they have different shapes? Of course they do have. Now we have to speed up, because if gametophyte, sporophyte is clear, if this flow chart is clear, then it is quickly, right? Then quickly you have to proceed further. Okay, what do you have to do, people? Then we have to proceed further. We have to revise it further. Okay, we have to revise it further. So algae may, yes, chlorophyll bearing, it's a plant. Of course, it will go for the photosynthesis. Let's not pick up such kind of basic points here. So simple, chlorophyll bearing, simple, thalloid, autotrophic, and largely aquatic. So largely it is aquatic. You will not say all are aquatic only. And why aquatic? Because they don't have xylem and phloem. They don't have xylem and phloem, right? Right? So moist stones, soils, wood, they all are having them. Okay, so you know that they also show the symbiotic association. So do not forget the example of lichen first, right? So in lichen, you know, it is having algae plus fungi. They both are in symbiotic association. Mutualism is there. And other than that, in the yes, cycus root, coralloid roots. Do you remember? In the Chalo, that is basically a blue-green algae. That is not the part here that we will discuss in Monera only. So in animals on sloth bear. Okay, in animals on sloth bear. Is that clear? Yes, everybody. Is that clear? Okay. So the fur of the sloth bear, it is having trichophilus. That trichophilus is nothing but it is the green algae. So it lives in association with what? It lives in association with green algae. Understood? So as we said, the form and the size is highly variable, right? So it can be colonial, round volvox. So in volvox, if you will talk about that single cells that are present there, they are actually beta chlamydomonas. Okay, and then eulothrix and spirogyra, they are filamentous, and then marine forms are there like your kelp. So here you are talking about your brown algae. Is that clear? You are talking about your brown algae. So when it comes to the reproduction method, vegetative, asexual and sexual. So when we talk about the vegetative sexual method, bache, vegetative sexual method, so you know that we will discuss about this fragmentation thing here, right? Fragmentation thing here. Okay, so here the thallus can regrow. That is something you have to keep in your mind. Okay, okay, done. Done. So fragmentation is the thing, and this is something we also read where in the living word chapter. So fragmentation is there in the algae. It is also there in the fungi. It is also there in the fungi. So each fragment will develop into the thallus, and in the case of asexual reproduction, you will talk about the spore formation. So when the word is vegetative propagation, fragmentation, and when the word is asexual reproduction, then you will go for the spore formation. What do you have to discuss here? You have to go for the spore formation. And here, the most common one is the zoospore. Why do we call it as zoo? From zoo word, remember animal. So animal are motile. This spore is also motile. It is having flagella. It is having flagella. So if I hope you guys have seen my detailed lectures as well, right? Where we also discuss few things which are out of NCERT, but now we are running out of time. Right now we are running out of time, and because we are running out of time, so it is our duty, it is our kind duty to complete the NCERT first. Okay, so I will be focused on NCERT mainly. Okay, done. Only in the case of plant physiology will touch the another part, but mainly this is something we have to discuss. So you know that zoospore. So zoospore is the motile spore. So do we have zoospore there only? No, we have other spores also. Do we have zoospore only? No, we have the other spores also. But yeah, just keep it in your mind, the zoospore. So they are flagellated, so that's why motile, and it will give rise to the new plant. Okay, okay, it will give rise to the new plant. So if you really want to revise it quickly, so you know that we have aplanospores. Other than that, aplanospores are also there, hypnospores are also there, right? Palmella stage is also there. All of them are showing asexual reproduction, but let's not discuss that. Right, let's not talk about it. Let's stick to this part only, because at this point of time it is a NCERT revision that we have to do. Okay, okay. Now they are flagellated. They are flagellated, and on germination they give rise to new plants. They are flagellated, and on germination they will give rise to new plants. Sexual reproduction may, of course, if it is sexual reproduction, gamete formation is there. Gamete formation and fusion is there. So here we have three words. Now these three words are important because their examples can come, right? These three things are important. Why they are important, bache? Why they are important? Because their examples can come in paper. Okay, their examples can come in paper. Okay, so when you are talking about the reproduction, one is isogamous. Iso means same. They are having the same gametes, right? These gametes are similar, right? So you can see these gametes can be flagellated and similar in size. They can be non-flagellated, but again, similar in size. Number one, they are saying it is isogamy. So basically, gametes, they need to be similar in size, right? If one is flagellated, other needs to be flagellated, but they needs to be similar in size. So isogamous me, you have the example of Chlamydomonas, Ulothrix, Actocarpus, Spirogyra, है ना? Chlamydomonas and different species of Chlamydomonas actually, right? Different species. So even your Ulothrix is also there. Your Spirogyra is also there, right? Some people call it Spirogyra. Take so Spirogyra, Chlamydomonas, right? Ulothrix. Other than that, even Actocarpus. So these are the examples which are given in your book, right? So that's why I'm writing it here. Okay, that is why I'm writing it here. Is that clear? So Ulothrix, Spirogyra, Actocarpus, Chlamydomonas, and other than that, Cladophora, which is again a green algae, right? They are basically showing isogamy. Then comes anisogamy, anisogamous type of sexual reproduction. So one other point that you have to remember, just see, in Ulothrix, gametes are flagellated, but in Spirogyra, they are non-flagellated. So in this table, that is what you can mention, non-flagellated gametes, flagellated one. Right, they are the flagellated one. Okay, so anisogamy mein Chlamydomonas and Eudorina. Again, Chlamydomonas species and Eudorina, right? So this example is given in NCERT, right? They are not explaining it anywhere else, but this is something you have to mention. Then oogamous, which is considered as the most advanced type of sexual reproduction, definitely in the case of volvox, in the case of fucus, we have to see this. So question can come from this particular example, right? So fucus is there, volvox is there, and yes, sargassum, Cara is also a green algae. And yes, few species of Chlamydomonas, right? Few species of Chlamydomonas, they also show this. So that's something you have to keep in your mind. Okay. Okay. Done. So oogamy, volvox and fucus example is there. But again, as I said, the examples that I have mentioned in that table, they are the part of your syllabus. So for sargassum, Cara, they both are given. Take? They both are given. Now here, when it comes to the sexual reproduction, so sex organs are there. Sex organs are there, but they are non-jacketed. They are non-jacketed means they are not covered, right? Just gametangia. We are also using the word gametangia, male sex organ, right? So they are non-jacketed, means they don't have any covering. And of course, they are unicellular, right? Just say, do you know any word here? Do you know any word here? Yes. So we are saying that sexual reproduction, sex organs are there, they are non-jacketed, right? They are non-jacketed and they are unicellular. Okay, so we have the word like for male sex organ, the word is antheridium. For female sex organ, the word is oogonium. The word is oogonium. What is the word? It is oogonium. But here exception is there, right? I discuss about this exception. This is Cara, which is the green algae, and here the sex organs are jacketed and multicellular. Okay, in this case sex organs are jacketed and multicellular. They are jacketed and multicellular. That's what you have to keep in your mind. So what are they? They are jacketed and multicellular. Is that clear? Is that clear? Is that clear? So Cara is a green algae, multicellular and jacketed sex organs are there. So this information is important. Baki here the economic importance is mentioned, right? So here just mark it. You know that half of the total carbon dioxide fixation on earth is carried out by algae. Now I'll pick up one more question here. That is again from the NCERT, bache. As you know, that is again from the NCERT, but yeah, we have to know each and everything from NCERT. We cannot say that that particular chapter is from this and this, that this point we will discuss in cell or something. No, we have to continue it here only. So when I'm talking about the algae, when you talk about its cell wall, you know that cellulose is there. Other than that, galactans, they are also polysaccharides, mannans. Remember this word, algae cell wall. So in the cell chapter, that is what they mention. So cellulose is there, galactans is there, minerals are there, and mineral like calcium carbonate. And they have mineral like calcium carbonate. That's what you have to keep in your mind. Okay, that is important for the quick revision. Now, next PYQ here. So they are the paramount importance as primary producer. TK, that is done. Now, many species like Porphyra, Laminaria and sargassum. Right now you write down these words. Okay, right now you write down these words. Later I'll explain everything. Right? Later I'll explain everything. Is that clear? Later I'll explain everything. Right now you just mention these words. I'll give you the tricks so that you will be able to remember all these examples. Okay, so we have Porphyra. What is the word that we have here? We have Porphyra. We have Porphyra. Right, bache? Right, bache? Done, bache? So Porphyra, Laminaria, sargassum, they are the edible one. If they are used as food, matlab they are the edible one. Then marine brown and red algae, they'll give what? Hydrocolloid. Which hydrocolloid? One is having water holding substance, algin. So algin is obtained from brown algae. That is again a PYQ. The question will come from the economic importance. Please write it down. The question will come from the economic importance. Please write it down. Then carrageen is there, that is from red algae. So it's not like carrageen, so green you will say. No, it is the red algae. It is the red algae. So they both are having commercial importance. Baki, we know about agar. We know about agar, isn't it? We know about agar. So agar, in the case of agar, it is obtained from Gelidium and Gracilaria. Again your PYQ. Again your PYQ. Okay, so it is used to grow microbes and in the preparation of ice creams and jelly. So chlorella is a unicellular algae rich in proteins and as a food supplement by space travelers. Okay, so now here there is a very famous question, right? That the like astronauts. Hanji. Yes. Astronauts. What they carry to space? They carry this. One is spirulina and another is chlorella. Why? Because they are food supplements. They are rich in protein. That's a famous question. They are food supplements. They are rich in protein. They are rich in protein. So spirulina is your BGA, blue-green algae, Monera, but it is your unicellular eukaryote. It is your unicellular eukaryote. It is your unicellular algae. That's the point. Is that clear? That's the point. Is that clear? Is that clear? So it is important. It is very important, right? So that is why I mentioned it. It's a direct previous year question. Okay? It is direct previous year question. Theek? Done? Sure? Sure? Okay. So let's write down the tricks. Let's write down the tricks and ensure your 4 marks. So firstly, let's talk about the green algae. So when you are talking about green algae, also known as chlorophyce. Also known as chlorophyce. So here the trick is US has very clean college. US has very clean college. There is one more trick. SUV Chalkara. SUV Chalkara. So U stands for Ulothrix here. S stands for Spirogyra here. V stands for Volvox here. C stands for, as I said, Cara here. And C stands for Chlamydomonas. So yes, you should know their mode of sexual reproduction as well. That is important. So it is chlorophyce, right? Most advanced one. This is the green algae actually, and it is the most advanced one, and it is considered as the ancestors of our higher plants, right? It is considered as most advanced one and the ancestor of our higher plants. So it is green algae. US has clean college. Then comes brown algae. Then it is brown algae. Brown algae. That is your Pheophyce. That is what? That is your Pheophyce. What's that? That is your Pheophyce. Pheophyce. So in the case of brown algae, my dear people, it's A and C. Which chlorophyll? Like here, you will have chlorophyll A. You will have chlorophyll B. Right? Chlorophyll A is there. Chlorophyll B. Here it is A and C. And the brown color is due to fucoxanthin. Important PYQ. Here the trick is self-DP. What is the trick? It is self-DP. What is the trick? Trick here is self-DP. I'll write it here only. I'll use a space here. S stands for just now sargasm. Sargasm. S stands for sargasm. Right? Self. E stands for actocarpus. E stands for actocarpus. Right? Then comes L. L stands for laminaria. L stands for laminaria. F stands for fucus. F stands for fucus. Then it is D and P. D stands for dictyota. D stands for dictyota. P stands for porphyra. P stands for porphyra. So sargasm, actocarpus, laminaria, fucus, dictyota, porphyra nahi, Pheophyce. Porphyra is the red algae, my bad. So it is Pheophyce. Right? Pheophyce. Then comes what? Then comes red algae, that is known as Rhodophyce. Rhodophyce. Here the trick is simple. P2G2. Here the trick is simple. P2G2. P porphyra. P polysiphonia. P porphyra. P polysiphonia. G gelidium and G is gracilaria. G gelidium. G is gracilaria. That's it. That's it. Done and dusted. That's it. Done and dusted. Okay. So already, right? See, in the case of brown algae, So this is about the green algae. So students, you know that at this point of time, we have to speed up. We have to speed up. It is very important for us to speed up, right? So let's focus on the most important things here. So please be a support and revise accordingly. So different types of chloroplast. Question can come from this part. So I'm mentioning it here also. So chlamydomonas is there. What is there? Chlamydomonas is there when it is cup shaped. So when it is spiral shaped, when it is what? When it is spiral shaped. When it is what? Spiral shaped. So as for spiral, ribbon like Spirogyra. Spirogyra, Spirogyra, whatever it is. So now it is the reticulate. Next is what? Reticulate. So reticulate is your Edogonium. Reticulate is your Edogonium, network like. Reticulum word is associated with that. Now beta, it is girdle like. It is what? It is girdle like. So when it is girdle like, it is Eulothrix. When it is girdle like, it is Eulothrix. It is what? It is Eulothrix. Okay? 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 Done? Any doubt? So ribbon like, spiral like, same, reticulate, done. So discoid is pending. Yeah, disc like is pending. Discoid or disc like. So that is zygema, right? But that is not given here, so I'm not touching this part. Done, bache? So here the pyrenoids, they contain proteins in addition to starch, right? So main storage body, it is, again, it is important, right? When it comes to the sexual reproduction, I have already discussed that with you all. And here we have the self-DP or self-defense, whatever trick you want to use, you can. So I keep it self-DPP, reminds me of Phaeophyce, not of Porifera, but of Phaeophyce, okay? But of Phaeophyce. So we have laminaria, we have this fucus, we have this dictyota. So now here in this case, you will see a stipe. You will see a stalk-like structure. You will see there will be the hold fast. So hold fast will help to get attached to substratum. To get attached to substratum. To get attached to substratum. Okay? Right? Hold fast. This is the stipe then. This is the stipe then. So leaf-like part is frond. That frond is for photosynthesis. That frond is for photosynthesis. That frond is for photosynthesis. Is that clear? So now look at this fucus. So this question can come. So here you have air bladders. So that is why diagrams are important. That is why diagrams are important. So this fucus is having what? It is having the air bladder. Okay? Then comes dictyota, dichotomously branched. Look at the branching pattern here. Look at the branching pattern here. Just look at the branching pattern here. So it is the dictyota. It is the dictyota. What is that? Dictyota. Now in brown algae, the diagrams, sorry, the examples, they say fucus. It is having diplontic life cycle. Your Actocarpus is having Haplo-diplontic. Sorry, laminaria na? Actocarpus is having Haplo-diplontic. So here what will you see? That zygote will not undergo meiosis immediately. Am I right? Here the zygote will not undergo meiosis immediately. Is that clear? Is that clear? What are we talking? Brown algae. In the case of brown algae, no zygotic meiosis is there. So this is something you can write down. No zygotic meiosis in brown algae. No zygotic meiosis in brown algae. Am I clear? No zygotic meiosis in brown algae. Am I clear? Am I clear? Okay. Then Rhodophyceae, Rhodo, A-D, red color, like erythrocytes, red now, so phycoerythrin it will be. That is giving it the red color. Okay, so stored food is your Rhodophyceae, Floridian starch. What's that? That is your Floridian starch. And in brown algae, when you talk about the brown algae, so just keep it in your mind, people. In the brown algae, the stored food is your laminaria or mannitol. Okay, laminaria or mannitol. So in the case of red algae, this is the point that you have to remember. Okay, that majority of red algae are marine with greater concentration found in warmer areas. They occur in both well-lighted region, close to the surface of water, and to the great oceans where the light cannot penetrate. Okay, right? Very little light can penetrate. What can penetrate? Very little light can penetrate. Is that clear? Take. And that is, why is it so? Why is it so? Why is it so? Okay, so remember your phycoerythrin, they have this pigment now that is giving it the red color. So this phycoerythrin can absorb blue-green. It can absorb blue-green light. And this light is having short wavelength. This light is having short wavelength. So because it is having short wavelength, right? It is having what? It is having short wavelength and it can penetrate. It is having short wavelength and penetrate deep into water, because it can penetrate deep into water. So because it can penetrate deep into water, so of course this pigment can absorb it. That is why they are able to grow there. Okay. And here the another important point in the case of red algae. In the case of red algae is beta, in the case of red algae, right, they reproduce by fragmentation, okay, fine. But when it comes to the spore, right, there will be, you won't see any motile structure in their lifespan. So non-motile spores are there, and even sexually also by non-motile gametes. So you are not going to see any motile structure here. Okay, you are not going to see any motile structure here. Is that clear? You are not going to see any motile structure here. Okay. Done. Okay. So examples important, tables important. So this table, yes, very important it is. Chlorophycetic green algae, AB, Pheophyceae, Rhodo, D is D. So remaining one is brown having C. Why brown color? Fucoxanthin. Why red color? Phycoerythrin. Can absorb blue-green light having shorter wavelength that can penetrate deep into the seawater. That's why they can grow there. Stored food, starch. And even we discussed the pyrenoid as storage bodies there. They have what? Protein besides starch. Then mannitol, all sugar. It is actually the sugar alcohol. Okay, and laminarian can be the stored food. So here cellulose and algin is there in the cell wall. Here just cellulose is there. Very important it is. Then Floridian starch, right? Then you have what? You have the Floridian starch. So Floridian starch is the stored food here. Floridian starch is the stored food here. So cellulose, pectin and polysulfate esters are present in their cell wall. They do not have any motile structure in their life cycle. This is important point regarding Rhodophyceae. No motile structure in their life cycle. And here bache 2 to 8 flagella, right? Just let's suppose if this is the tip, right? Tips are having this. They have equal size. They are at the tips. 2 to 8 equal size flagella. 2 unequal lateral flagella. So their size is also unequal. So one will be smooth, one will be tinsel. Tinsel as in one will be smooth, one will have hair-like, right? Hair-like structures over there. And here it is absent. So fresh water, brackish water, salt water, fresh water, you understand, salt water, you understand, brackish water is having salt concentration in between this fresh and salt. That's it. That's it. So if somebody is thinking that madam is going very fast, we have to, because we don't have any option, right? We have to complete it ASAP. After that, let's jump to anatomy. Then let's jump to plant physiology. Okay. No, beta, I started the class late. Whatever the reason is, I know I started the class late. So we will finish plant kingdom first. Then there will be a dinner break from my side, right? Then there will be a dinner break from my side. So if you need a dinner break, I'm very sorry. You can just, end the lecture, not end the lecture. You can just have your dinner and watch the lecture later. Okay. So bryophyte is the word. So dominant bodies, yeah, main plant bodies. So this is the example here, right? The Markensia that we discuss. This Markensia. Okay. Look at the cells here. How beautiful it looks, isn't it? Chalo. So dominant photosynthetic. Dominant photosynthetic phase is what? Yes, people. When it comes to the dominant photosynthetic phase, what do we have there? Yes. when it comes to the dominant photosynthetic phase. Dominant photosynthetic phase. Just now I told you it will be gametophyte. It will be gametophyte. But here, sporophyte is also multicellular. Sporophyte is also multicellular. Okay. What's that? It is multicellular. Understood? Understood? Chaliye, let's go for that. So here you discuss about the liverworts. They are the lower bryophytes, and here you have the example. The example is Marcasia, and Rixia is also one of the examples, but in NCERT only Marcasia is given. So you can just write it down. Okay, you can just write it down. So what's that? It is your bryophyte, that also known as amphibians of plant kingdom. Also known as amphibians of plant kingdom. Okay? So Rixia is also there. Marcasia is also there. These are the names. Okay. Okay. Done. Okay. So lower bryophytes they are. Do they have vascular tissue? Already told you no. Do they have embryo? Yes. Their sporophyte is multicellular. Mosses, they are the higher bryophytes. So, bache, whenever you talk about the succession, let's suppose there is a barren land, right? So, you will see that lichens and mosses, that is also PYQ. That's why I'm telling you here, they are the pioneer communities. What are they? They are the pioneer communities. What are they? They are the pioneer communities. They are the one that started, right? They are the one that they will start the further, like they will grow on the rocks, they'll convert it into soil. So they are the pioneer community, the one that are going to start the life there. That is the meaning here. Now, bryophytes may mosses and liverworts are there. Hornworts are also there, but here we are not discussing that. We'll stick to liverwort and mosses. So commonly in moist shaded areas in the hills. Okay, so here these are the diagrams. But when you talk about their thallus, they are also thalloid. But yeah, right, they can be erect or right. So basically they are also thalloid, but yeah, better differentiated, right? They are better differentiated than your algae. So they are erect or prostrate, right? When you talk about their thallus. So this is the example of Marcasia that they have given you here. This is the example of Marcasia. Remember, Marcasia is dioecious. Dioecious, when we are saying, it means it is unisexual. It means it is unisexual. It means it is unisexual. So this is BMH. So this is the trick that you have to remember. Just from BMW cars, just make it BMH. Okay, from BMW car, just make it BMH. So B stands for beta bisexual, M stands for monoecious, and H stands for hermaphrodite. H stands for hermaphrodite. So bisexual, monoecious, hermaphrodite. Bisexual, monoecious, hermaphrodite. Bisexual, monoecious, hermaphrodite. So what is the word that we have? We have the word Marcasia here, which is unisexual. So archegoniophore, G word is there. G for girl. So this is the female part. Archegoniophore. And Pteridophytes, male part it is. So gemma cup, they are basically for the vegetative propagation. Okay, so here you don't have true root, stem and leaves. Okay, we don't have true root, stem and leaves. And here instead of that, basically their structure is better than the algae, more differentiated than algae, but still it is not having true root, stem and leaves. Rhizoids are there, so they can be unicellular. So we will see them as unicellular in your liverworts, and they are multicellular in your mosses. So they are not roots, they are root-like. So you can see dorso-ventrally flattened thallus as well. So that is how you have to revise the diagrams these days. Right here, this is the gemma cup, which is meant for what? Right, these are the gemma cup, which are meant for what? Vegetative propagation. So here you people can see in between, right? This is that complete cup, and these are the gemmae. So they'll grow, right? They'll grow into a complete plant. They'll grow into a complete plant. Is that clear? They'll grow into a complete plant. Is that clear? So this diagram is also there. This is of funaria. And this is of what? This is of sphagnum. So sphagnum, right? The trick is also there here. It is a peat moss. So look at the structure. Do not forget that. So you can see gametophyte is the dominant phase here, right? Sporophyte is dependent. Sporophyte is dependent. One is completely, one is dependent partially, right? So sporophyte mein you will see foot, setae and capsule. So let's have a quick revision of diagrams as well from this part. So sphagnum is also monoecious. What is it? Monoecious. You can see that it is having archegonial branches and entheridial branches. So diagram part is clear. Diagram part is clear. So in this sporophyte, spore formation will be there. In this sporophyte, spore formation will be there. So beta, I am repeating this again. The bryophytes are also known as amphibians of plant kingdom. We also call them as amphibians of plant kingdom. What are they? They are amphibians of plant kingdom. Why do we call them as amphibians? Amphi means dual. Amphi means dual. So they're right. So basically, they were the first plant who started taking the step towards the land habitat. Right, algae. Right, they started taking the step to. Just amphibians are the one who started taking the step towards the land habitat. Yeah, for the right land itself. Here also this is the thing. But yeah, they need water for their sexual reproduction. Right, they are also dependent on water. They need water. Right, they don't have xylem and phloem. They don't have xylem and phloem. They need the water that they can take. Is that clear? Is that clear? Sure? Sure? Are you sure? Are you sure? Yes, please. Chalo. So next part. So let's have a quick revision. Again from the diagram, I have already mentioned the important points. Okay. Okay. I have already covered. Life cycle I have already covered. So let's complete the NCERT like a super fast train. Take. Done. So guys, are you okay with the speed part? Again, the issue is there. Just a minute. There is some issue with the charger. Whenever I'm putting the charger, my laptop is not working. One minute, beta. So I hope I'm perfectly audible. The speed is fine, right? The speed is fine, right? Okay, it's fine, right? Speed is okay, but my laptop is not. Ma'am, faster. है ना? Even I'm thinking the same. Even I'm thinking the same. Chalo. Then let's pace it up. Take, chalo. So bryophytes, amphibians of plant kingdom, because, right, they started, right, because they can live in soil. These plants can live in soil but are dependent on water for sexual reproduction. Important point, right? Important point, and a very basic point as well. And a very basic point as well. So where do you see them? You see them in the damp, humid and shaded localities. Okay, they don't have the vascular tissue. They prefer such kind of locations only. Okay, they prefer such kind of locations only. Now the next point is there. Next point is there. They play an important role in plant succession, right? So when I'm talking about the succession here, I am discussing the mosses, and I already told you lichens and mosses, they are considered as the pioneer communities. So why do we discuss that? Why do we discuss that? Because of course it is important. That is why we are discussing it. So plant body, right? The sporophyte, the plant body of bryophyte is more differentiated than that of algae. It is more differentiated than that of algae, but it is still thalloid. It is still thalloid. Is that clear? Is that clear? So that can be erect thallus, that can be prostrate. That can be erect thallus, that can be prostrate. That can be erect thallus, that can be prostrate. This part is clear. This part is clear. Now they have unicellular or multicellular rhizoid. How will they help? They'll help them to get attached to the substratum, to attach to the surface. They will help them to get attached to the substratum, to get attached to the surface. That is what you will need to keep in your mind. So no true root, stem and leaves, no basically the vascular bundles, already discussed that. So the main plant body, it is the gametophyte, it produces the gamete. So sex organ here are multicellular, right bache? Male sex organ is antheridium, female one is archegonium. Female one is archegonium. Am I clear here? The female one is archegonium. Is that clear? Is that clear? So they are multicellular, but yes, here you can also write it down that they are jacketed. When I'm saying jacketed, matlab they are having that outer covering na. So they are jacketed. They are jacketed. So when you are talking about the antheridia, from anther, anther is also the mate part na, antheridium. From antheridium, just remember antherozoid. So they are the male gametes and they are biflagellated. They are also known as biflagellated sperms. They are also known as biflagellated sperms. They are also known as biflagellated sperms. So female sex organ is archegonium. Female sex organ is archegonium. G for girl, right? Female part, right? So it is flask shaped and produces a single egg. It is something like that. Of course, it is also jacketed and it is something like this. Okay, something like this. Okay, now they'll be released in water. Antherozoid, they'll be released in water. They'll go for the egg, right? They need to contact with the archegonium. They need the water. And of course, the chemo attraction will be there, right? The zygote will form. So here the question can come like this. Already in Haplo-diplontic life cycle, I told you. Already in Gayatri flowchart, I told you, right? Already in that flowchart, I told you. Here, the zygote will not immediately undergo meiosis. Zygote will undergo mitosis first, right? It is unicellular. It will become multicellular. So when, it is unicellular. Of course, zygote is not unicellular. My bad. So I am saying that zygote is diploid here, right? It is diploid here, right? It is single cell. It is single cell. Mitotically, it will grow. So when this zygote grow na, it become multicellular. It become what? It become multicellular. It will become what? It will become multicellular. Are you getting my point? So of course, this zygote is actually developing in the sporophyte. So of course, it will become multicellular and then this will acquire something like that. Just a foot will be there, right? Setae will be there and the capsule will be there. And within this capsule, my dear students, within this capsule, there will be the spore mother cells. So if it is the mother cell, meiosis will occur. Haploid spores are obtained. Haploid spores. So this sporophyte is dependent on the gametophyte. Am I clear here? This sporophyte is dependent on the gametophyte. Okay. Right. So foot, seta and capsule. Foot, seta and capsule. Foot, seta and capsule. It is not foot, seta and capsule. So in Markensia, that is how you are going to see. Only in Rixia. That is an extra example. Rixia, it is. In Rixia, sporophyte is just capsule. Okay, here it is this. Understood? Understood? Sure? Sure? So Rixia is your this monoecious. Okay? Any doubt? Any doubt? Pakka? Chaliye, next part. So here sporophyte is multicellular. So the next part is saying that, see, zygote do not undergo reductional division immediately. We are not saying it will not at all undergo the reductional division. No, that is not something that we are saying. We are not saying that it will not undergo the reductional division at all. All we are saying that it will undergo reduction division, but not immediately, right? Not immediately. First of all, it will grow, right? It will grow. It will become multicellular. It will form sporophyte. And because it has to grow, it has to form diploid sporophyte. So of course, firstly, the mitosis will be there. So zygote will divide mitotically. Zygote will form the sporophyte. And this is not free living, but attached to the photosynthetic gametophyte here will take its nourishment from that. So some cells in the sporophyte will undergo meiosis, will form spores, and I told you already about that. I told you already about that. Okay, so bryophytes in general are of little economic importance, but mosses, they provide food for herbaceous mammals, birds, and so sphagnum, this is an important example. What's that? It is an important example. So when you people talk about the sphagnum, it's a moss, and it is also known as peat moss. Bit of this peat, okay, it is very, very, very important, right? This peat part is very important. So it is used as a fuel, right? It is like sphagnum will not, when, let's suppose this thallus, right, when it will get accumulated for a longer period of time, it will result in the formation of this peat. Okay, right? So this peat was used as a fuel, as a packing material, and for the transshipment of the living material because of their capacity to hold the water. So they have that retention, water retention. What do they have? They have that water retention. Is that clear? Is that clear? What do they have? They have that water retention. Am I clear here? Am I clear? Sure, done. So mosses along with lichens, already discussed. The decomposed rocks, discussed that, from dense mat, done. So liverwort specifically, so I have discussed two examples with you all. One is your Markensia, another is your Riccia. It is dioecious, that is also PYQ, but this one will be monoecious. Told you about it now. So they grow usually in moist shady habitat, done and dusted. The plant body is thalloid, already told you, and this thallus is dorsal ventral and closely attached to the substrate, right? Attached to the substrate, right? So they have tiny leaf-like appendages and stem-like structure. So what will happen here? Fragmentation will occur here, and the gemma cup, you remember gemma will help in the growth of the new plant. So gemma, they are nothing, they are but green. They are multicellular asexual buds. They are green, they are multicellular asexual buds. They are green, they are multicellular asexual buds which develop in small receptacles called gemma cups. Told you already. So sexual reproduction is also there. You know that, right? In Markensia, because it is unisexual, we have different thallus. Okay, so they'll form gametes. Sporophyte will be like this. This is already we have discussed. Now when it comes to the mosses, in the case of mosses, the one thing that we have to remember is protonema. What is the thing that we have to remember, students? We have to remember protonema. What is the thing that we have to remember? We have to remember protonema. So when you talk about the life cycle, here they have two stages. This is suppose that you have the haploid spore, right? The sporophyte will form haploid spore. That spore will not immediately form gametophyte. That spore will form protonema first. And it is having evolutionary significance as well. So this protonema, it develops from the spore, right? This protonema, it develops from the spore, yes or no? It develops from the spore, yes or no? Important it is, right? It is creeping, green, branched and filamentous stage, right? And this stage, it shows, right, it is having evolutionary significance because, see, this stage is showing that it is creeping, it is green, it is filamentous, it is creeping, it is green, it is filamentous, right? So it shows that the mosses, they grow from the, right, algae-like ancestors, okay? So after that, you will have the leafy stage. So here the gametophyte is leafy. Okay, so it will develop from the secondary protonema as the lateral bud. Okay, it is, so protonema is the first stage, which is filamentous, creeping, green, branched and juvenile, right? It is having branches, beta. Okay, it is having the branches. Okay, okay. Take, so when it is the protonema, it is having, branches. One branch, let's suppose this is another branch. It will be your chloronemal branch. So it is extra. All I'm showing is that this will show photosynthesis. One is rhizoidal branch as well, that will just absorb the nutrient. That's it. That's it. So this is the first stage, and then there comes the leafy stage, which is the second stage here. Which is the second stage here. So protonema is going to form that, where, right, beta, they consist upright cylinder axis having spirally arranged leaves. Okay, so here the rhizoids are multicellular. So when I was explaining you the diagram, I told you this is important PYQ. Okay, again, this is also an important PYQ. Done? Done. So what is going to happen here? Fragmentation and budding. The vegetative propagation in mosses is by fragmentation and budding in the secondary protonema. So secondary protonema will form that bud. From that bud, basically here it is written also, beta, that from secondary protonema as a lateral bud, your leafy gametophyte will form. Understood? Your leafy gametophyte will form. So sex organ and 3D archegonia, right, bache? So they are jacketed as well, multicellular. So zygote will develop into sporophyte having foot seta capsule. The sporophyte here is more elaborated. Okay. Okay, so spores are formed after meiosis. They have an elaborate mechanism of spore dispersal, जैसे elaters and all we discuss. But now stick to the examples also. So Funaria, Polytrichum, sphagnum. So sphagnum is the peat moss. Have a look of this diagram. If there is any doubt, do tell me. Do let me know if there is any doubt here. Any diagram, doubt in diagram here. Diagrams are important. So have a look quickly. Have a look quickly. Done? Sure? Okay. Let me finish this part, then we will have break. So these are Markensia, these are that Gemma cups. ठीक, so just have a look. This is the archegonium egg. Sperm will approach here, zygote will form. So zygote will not undergo meiosis immediately. It will undergo mitosis. It will become multicellular. It will grow into foot seta and capsule. Now within this capsule, there are certain cells which will undergo meiosis, and you will get the spore. That spore will germinate. That spore will germinate and the gametophyte is obtained. Take, and the gametophyte is obtained. So this is the gametophyte that we have. Female gametophyte, right? Archegonia, right? Bache, female gamete will form here. You can see this. This is the male gamete. So male gamete is biflagellated entherozoid. What's that? It is biflagellated entherozoid. Okay, okay. Any doubt? These are the rhizoids that are for the attachment. So mosses mein, ye dekho mosses. They'll grow on the barren land. They are the pioneer communities, right? So foot seta and capsule is there. So here you people can see that when underwater, male sperm cells will to join the female cells. Again, the same thing is there, but here you will see, right, that the protonema will form first. What will form first? Protonema will form first, right? Next is pteridophytes. So in the case of pteridophytes, again, haplo-diplontic life cycle is there, as I said. Haplo-diplontic life cycle is there, as I said. What is there, right? What is there? Haplo-diplontic life cycle is there. Number one, dominant photosynthetic phase for a longer duration is what? It is your sporophyte here. So sporophyte here. But what we have to remember, that the gametophyte is also photosynthetic and independent it is. Take, right? After this, next point is xylem and phloem are present. So they are the first plants where we will see xylem and phloem. So when we consider tracheophytes, when we consider what? Tracheophytes. So in the case of tracheophytes, you have pteridophytes, gymnosperms and angiosperms. Understood? Pteridophytes, gymnosperms and angiosperms. Understood? Pakka? Pakka? So here you can see, and here you will see in pteridophytes, then I'll read it from the NCERT directly. You please try to understand this part. Pteridophytes. Here you will see true root and stem and leaves. Right, but remember their gametophyte also need water, right? So their distribution is restricted. So now here, as I said, you will see true root, stem and leaves. That's what I just said. Now when you talk about these leaves, right, they are right, some leaves, right, out of that, some leaves on that leaves, right, you will see some structures. Which structures? Right, we call that as sporangium. Sporangium. What's the word? Sporangium. Sporangium is singular. So this sporangium in which spores form. Structure in which spores form. It is the structure in which spores form. It is the structure in which, yes please, it is the structure in which spores they form. Are you getting my point? Right? So now when you're talking about, like, leaves are there. Let's suppose you're talking about certain leaves. You're talking about certain leaves and that leaves are having a kind of structure on that. Right? It is having a structure. We call it as sporangium. What do we call it? We call it as sporangium. What do we call it? We call it as sporangium. Right? Sporangium. So sporangium singular, sporangia plural. Within them spores will form. Sometimes even sporophyte is also known as sporangium because in that also the spores will form. So you have the sporangium here. What do you have here? You have the sporangium here. So these leaves that are bearing sporangium, that are having sporangium, we call them as, right, what do we call them? We call them as sporophylls. Right, leaf-like structure it is. We call it as sporophyll. What do we call it? We call it as sporophyll. Yes, people, what do we call it? What do we call it? We call it as sporophyll. Right, why? Because it is having sporangium. What is going to happen to the sporangium? Sporangium, yes, sporangia, it is plural. It is basically plural, nothing else. So here within it, spores will form. What will form? Spores. So initially when you talk about these spores, right, this is, let's suppose this is your sporangium. This is your sporangium. In this spores will form. These spores are of course haploid. They are haploid. So if they are haploid, how will they form? They will form with the help of meiosis. So they are the spores that we have. All used to be same. So whenever the spore used to germinate, it is haploid. So when it will germinate, it will grow mitotically, it will form gametophyte. It will form gametophyte. So when you talk about the spore, right, this general spore, gametophyte, haploid, this gametophyte used to be monoecious. Monoecious, I told you, na, BMW. From BMW, let's make it BMH. So it is bisexual monoecious hermaphrodite. It is bisexual monoecious hermaphrodite. It is bisexual monoecious hermaphrodite. So from these spores, the gametophyte used to form, and that is monoecious, bisexual. So this gametophyte will give male gamete, right? Will give female gamete, right? Again they'll fuse, again the zygote will be there, again zygote will grow and will form sporophyte. Okay? That is the story that we know. That is the story that we know. So what happened, right, in the case of pteridophytes, what happened, right, in the case of pteridophytes, there are certain pteridophytes which started forming like these are the same type of spores. They are sporous. These type of pteridophytes are homosporous. Homo means same. They are forming same type of spore. But some Pteridophytes, they were heterosporous. They were what? Heterosporous. They were what? Heterosporous. Heterosporous. So hetero means different. Hetero means different. Hetero means different. So different types of spores are there. Different types of spores are there. What are there? Different type of spores are there. Different types of spores are there. Heterosporous. So there is a trick that we have. It is he, she, sa. He stands for heterosporous. Selaginella. Salvinia. Right? Salvinia. So right, heterosporous, Selaginella, salvinia. Heterosporous, Selaginella, salvinia. Heterosporous, Selaginella, salvinia. Heterosporous, Selaginella, salvinia. That is something that we have. Okay, that is something that we have. Heterosporous, Selaginella, salvinia. Heterosporous, Selaginella, salvinia. Hetero means different spores. Here you will have microspore. You know, in the nature, male gametes are small. Here you have megaspore and microspore. So microspore, small spore it will form it will form male gametophyte, right? Because they are also haploid, they'll grow, they'll form gametophyte only. So this one will form male gametophyte and megaspore will form female gametophyte, people. What will it do? It will form female gametophyte. That's what you have to keep in your mind. Right, so this trick, it is important. Right, this part, it is important. Right, now there is a point which will show you the precursor of seed habitat, which will be as a precursor of seed habitat. Now see, so many sporophylls together, they also get condensed. They form this cone-like structure, strobili, or cone-like structure, strobili, or cone-like structure. Right? And see, look at this. Look at the diagrams here. These are that sporophylls. Right? These leaves which are bearing the sporangia. So these structures here, you all have seen the pteridophytes. You all have seen the ferns. Just Google it, you'll get to know. Done? Done? Sure? So see this rhizome, right? Sporangium, leaf. We are using the word frond here. Understood? Sure? Yeah, देखो, these are the diagrams. So it looks like horseshoe tail, na? Horse tail. So that is the name. So strobilus, yeah, strobili is plural. This is the cone that we are talking about. Roots, stem, leaves. So here you will have true root, stem and leaves. Understood? Here you will have true root, stem and leaves. Okay? Okay. So diagrams are important, right? So, this is your Selaginella. This is your Selaginella. Even I forgot to mention it. Right, this is your equisetum, horseshoe tail. Equisetum, horseshoe tail. So let's have a look. Baki ye dekho, these are the cones, right? Strobili. They'll have sporangia. We'll discuss it, don't worry. So homosporous, which will form same type of spore, different type of spores, done. Array. Look at the diagram, sporangia. Look at their position. Right? Look at their position. So these are the different diagram examples. Jaise lycopodium. Right? I'll discuss the I have a trick here, right? So if you will search now, if you will search plant kingdom tricks, Ambika ma'am, so you will get to know about these tricks individually. Don't worry about that. I'll discuss it with you all. Okay, we will discuss it. So firstly, let's revise it from the NCERT. Then I'll show you the pictures here. So of course, so pteridophytes will have horsetails and ferns. Discussed? Yes. Ferns. Remember, they are ornamental as well. They are ornamental as well. We plant them just for the decoration. Right? So pteridophytes, they are having medicinal purpose. They are the soil binders. Matlab, they'll prevent the soil erosion as well. Right? But let me finish this chapter, then there will be a break. Okay, then there will be a break. So they are frequently grown as ornamental, told you already. So they are the evolutionary, they are the first terrestrial plant to have vascular tissue, already discussed. Right? So cool, shady, damp places they need to flourish. Right, they can even flourish in sand conditions because they have xylem and phloem. Okay, so bryophytes may, you know that dominant phase in the life cycle was gametophyte, but in pteridophyte it is the sporophyte. We have discussed it. So in pteridophytes, the dominant phase is sporophyte, and that sporophyte is having true root, stem, and leaves. Right? That sporophyte is having what? That sporophyte is having true root, stem, and leaves. What do they have? True root, stem, and leaves. Is that clear? So these organs, they have well differentiated vascular tissue. Right? Well differentiated vascular tissue. So the leaves in pteridophytes, they are small. Okay, so when they are small, the word used will be microphyll. Word used will be microphyll. So microphylls are there in Selaginella. Selaginella. And remember now, Selaginella and Salvinia, they have different types of spore. So they are having microphylls as well, right? Microspores that they are formed, and they have large, right? Ferns are having large leaves, right? Ferns, in their case, the leaves are what, beta? They are large. So sporophyll will have sporangia. So what is the sporangia? The structure in which spore will form. Okay, so the sporangia is present on sporophylls. Understood? Understood? And sometimes these staurophylls, they are all condensed, they form strobili or cone, just like Selaginella and equisetum. And just now I told you equisetum is also known as horseshoe tail. It is also known as horseshoe tail. It is also known as horseshoe tail. Is that clear? So sporangia produce spores by meiosis. The spores germinate to give rise to inconspicuous, not so prominent it is, inconspicuous, small, but multicellular. Inconspicuous, small, but multicellular, free living, mostly photosynthetic gametophyte. So gametophyte is multicellular here. Yes. Is it dependent? No. It is free living, and it is inconspicuous. It is inconspicuous. Is that clear? So these gametophytes, they require cool, damp, shady places to grow, and because of that, the specific restricted requirement and the need for water for fertilization, the spread of living, right? The spread of living pteridophyte is limited. Is that clear? Is that clear? So again, here also male sex organ is antheridia, which will form antherozoid. Archegonia is also there. Simple as that. The zygote will undergo mitosis first, multicellular sporophyte first, and then you know that it is the dominant phase. Any doubt here? Any doubt here? So ये देखो, majority of pteridophytes are homosporous, but Selaginella and Salvinia, they are having two types of spore, heterosporous. So that's why trick is he seesa. Done bache? Done bache? So megaspore will give female gametophyte, microspore will give male gametophyte. Okay? Okay. So what is the beauty here? What is the important point here? What is the beauty here? What is the important point here? The female gametophytes in these plants are retained on the parent sporophyte. The female gametophytes in these plants, are retained on parent sporophyte for variable periods. Right? The female gametophytes in these plants are retained on the parent sporophyte for variable periods. Is that clear? Done? right? So this is important. Female gametophyte in these plants, they are retained on this gametophyte that will form gamete. It will be retained on the parent sporophyte beta. So development of zygote into young embryo will take place within this female gametophyte, and female gametophyte is retained on the sporophyte. So this is the precursor. This is something that has started the seed habit. This is something that has started the seed habit. Okay. It is an important step in evolution. It is an important step in evolution. Is that clear? So examples are there. So basically in pteridophytes, how many classes are there? 4. Xylopsida. So it is Pleistero. Lycopsida, Sphenopsida, Pteropsida. Take. So it is basically Xylopsida, which will have Xylopsida, so xylotum. Lycopsida, Selaginella and Lycopodium. Then Sphenopsida, Equisetum. Then it is Equisetum. Then it is the Pteropsida, Dryoteris, Teres and Edientum. Take. So examples are important. So Xylopsida will have xylotum, right? This Xylopsida will have xylotum. What is the word here? What is the word here? It is xylotum. What is the word here? It is xylotum. Do write it down. It is important. Right. So it is also known as whisk fern. This xylotum is also known as whisk fern. Take. Next is Lycopsida, which will include your Lycopodium and Selaginella. Lycopodium and Selaginella. So Lycopodium is known as club moss. So bache, they are not actually the mosses, because mosses are bryophytes. They are not actually the mosses, because mosses are bryophyte. But this Lycopodium is the club moss, right? So it's not a true moss. So but this is the common name that they have. It is the club moss, Lycopodium. What's that? Lycopodium. What's that? It is the Lycopodium, which is the club moss. And Selaginella is spike moss, sometimes referred to as little club moss, ya fir spike moss. Understood? Next is Sphenopsida having equisetum, horsetail, but I don't know why do I mention horseshoe tail always. It is horsetail. It is horsetail, right? So there is a correction there where I have mentioned horseshoe tail. This is a horsetail. Horsetail. Is that clear? Then comes the Pteropsida. So Pteropsida, as the name is suggesting, Dryoteris, Teres, Edientum. Edientum is also known as walking fern. Walking fern, aquatic it is, walking fern, right? It is also known as what? Edientum. Edientum. So it is also known as walking fern. Okay. Yeah, midden hair fern. Midden hair fern. So Dryoteris is there, Teres is there. Okay. So do write it down, and it is the Edientum. What is the word that we have? Edientum. Okay. Also known as walking fern. Also known as, yes, please. Yes. Right. So Edientum, walking fern, maiden hair fern. So actually it is regarding what? This Edientum, maiden hair fern, or walking fern, it is like basically it is related to the method of propagation. Just fragmentation is there. Sometimes rhizome will help in that, and sometimes in this case the leaf tips, they'll touch the ground, they'll form adventitious buds, and then the new plant will grow, something like that. That's why it is this. So Gymnosperms mein what type of life cycle do we have? What type of life cycle do we have? We have the diplontic life cycle. Here in this case, remember the endosperm. Endosperm is haploid. Jैसे you know na, angiosperms are having triploid endosperm. Here endosperm is haploid because it is developed from, right? Because it is, I'm just writing the peculiar features here, yeah, most important points here. Because it is developed from female gametophyte. Because it is developed from female gametophyte. So it is haploid, and it is developed from what? It is developed from female gametophyte, number one. Secondly, in the case of gymnosperms, right, you know that seeds are naked. So basically here what is happening? The ovule is directly there, right? The ovule is directly there on that sporophyll. So no flower is there, so that is why it is naked. Okay, that is why it is naked. Just like angiosperms, right? Yeah, dicots, it's also show the secondary growth, right? Gymnosperms. Okay, so there are other important points as well. So firstly, tell me, up to this part all clear? Up to this part all clear? Yes, you need to revise the things a lot, right? And trust me, you will be able to do that. You people are amazing. You have to believe in yourself. Fine, you have to believe in yourself. So here, seed is naked. So matlab, these are the plants where first time seeds will form, right? Seed formation is there for the first time here. Okay, so ye dekho. We have the example. We have cycas. Cycas is beta also known as living fossil, right? Which plants or which animals are known as living fossils? The one that show the characters similar to the primitive, similar to the fossilized organism, right? They are the one that show the characters similar to the, right, bache? Similar to what? Similar to the, right, the organisms which are fossilized. Okay, or you can say that they are not evolving, they are showing the primitive characters. In any way you can define it. So we have the example of cycas, we have the example of pinus. So I'll pick up the examples here first. So C for cycas, C for coralloid roots, right? It is having coralloid roots. Basically in that roots, in the cells there, you will see that blue-green algae, right, which is also known as cyanobacteria. That blue-green algae, which is also known as cyanobacteria, right, which is also known as what? Which is also known as cyanobacteria. Which is also known as what? Which is also known as cyanobacteria. Cyanobacteria. Cyanobacteria, right? So here it is in the association there. Then comes the pinus. Pinus will show mycorrhiza. That is again a very important question. Mycorrhiza. Again, it is also the symbiotic association. Here, the roots of pinus tree and the fungal hyphae, they are showing the symbiotic association. So you can see the cycas here, right? These are the cycad cones here. You will see megasporophyll, microsporophyll, and remember the cycas is dioecious. Male and female is on different parts, but the pinus is monoecious. Again, these are the PYQs that I have discussed initially, right? These are the PYQs that I have discussed initially. Okay? So firstly, let's revise it. Then I'll show you this thing from the, I'll explain it with the help of diagrams as well. Okay, so there is no double fertilization in gymnosperms. There will be no triploid endosperms in gymnosperms. That will be haploid, that is developed from the female gametophyte. Just keep it in your head. So any doubt? Any doubt? So gymnosperms, what's the word? Gymnosperms, what's the word? Gymno. Gymnos is naked, sperma is a seed. Gymnos is naked, sperma is seed. Gymnosperms. So here the ovules are not covered by the ovary wall, they are exposed. That's why, because ovules after fertilization they form the seed, right? If you remember it, very good. If you don't, you can write it. Ovule after fertilization. After fertilization, it is the ovule that is going to form the seed. It is the ovule that is going to form the seed. Is that clear? So both before and after fertilization, basically it is exposed, right? So seed is the result of post-fertilization change. It is done. So gymnosperms mein beta tall trees, medium sized, everything is there. So this example is important. Sequoia, red giant wood, है ना? Giant redwood. So it is the tallest tree. So tallest tree is a gymnosperms, not an angiosperm. Again, a PYQ. So what's the word? Sequoia, and it is the giant redwood. It is the giant redwood. It is the giant redwood. Is that clear? So roots are generally taproot. So when we use the word taproot, beta, so basically from in embryo there is a part, radicle. I hope you know that. So from the radicle, these roots will grow. There will be primary root, then secondary and tertiary will be there, and tertiary will be there. So that is how it is. Right, that is how it is. So here you will also see mycorrhiza, like I told you. So mycorrhiza will be there in the pinus. So mycorrhiza is what? It is the symbiotic association. What type of association it is? It is the symbiotic association. Okay, symbiotic association. So symbiotic association mein beta, what is going to happen? What is going to happen here? Right. So basically the pinus roots, they cannot absorb the nutrition directly from the soil. They are not capable enough to take out all the nutrients. Matlab, they need these fungal hyphae. They'll absorb the nutrients, they'll give it to the pinus tree, and pinus tree will grow. So we cannot grow a pinus tree in a soil where, right, we cannot grow the pinus tree in a soil where these fungal hyphae, this association is not there. So if you remember, we also discussed glomus in our class 12. So it is the genus It is the genus of fungus, right? That will show this mycorrhiza arrangement. Understood? So this question is important, already discussed. Cycas is having some cells, right? We are calling these roots are choroid roots. They are having nitrogen fixing cyanobacteria. So because they have nitrogen fixing cyanobacteria, so of course this plant is helping in the nitrogen fixing thing. So stems are unbranched here. So in the case of cycas, just see, the stem is unbranched, and at the top of that, you will see all the leaves. You will see all the leaves. What will you see? You will see all the leaves here on the top of that. On the top of that, right? So we call it as caudex. We call it as caudex. We call it as such kind of stem is known as caudex. ठीक? But in pinus, branches are there. Okay? Okay? So pinus mein it is branch, pinus and cedrus. Again, it's a previous year question. Please do not skip it. Do not skip it. Then leaves may be simple or compound. Cycas mein pinnate leaves are there that will persist for few years. Okay, and gymnosperms may, because they can adapt in the xerophytic xeric condition, so they are well adapted to withstand the extreme temperature, extreme humidity and extreme wind. So conifers, what are conifers? They are the cone bearing gymnosperms, right? You'll see them in the hilly areas. We see them, right? They have the cones like your pinus. So they have needle-like leaves. When we have needle-like leaves, imagine we want, just remember, when we have needle-like leaves, basically it is to reduce the surface area, right? So that the reduced surface area, so the gaseous exchange will be affected. This is what we need. We want the less gaseous exchange here. So we don't want to lose the water in the form of vapors as well. So they have thick cuticle, they have sunken stomata, and that will help to reduce the water loss. This part is important. Please revise it quickly. We are about to finish it. Then I'll definitely give you the break. So please tell me. Done? Done? Okay, chalo. So let's discuss the next part, but students, do not forget to watch this revision video, right? Having the proper strategy, having everything you need to know for these last 20 days. So if you are struggling, if your syllabus is done and dusted, again, this video will help. But if you are struggling, right? Because see, in this video, I have just divided the chapter, which chapter you need to pick first, which chapter you need to do later, and which MCQs you need to consider. Okay, but if you are struggling with the syllabus completion, so you can see how to complete syllabus, number of mock tests, best routine, everything is discussed here. So you can watch it right in the break time. So now let's come to this part. Okay. Chaliye. So next part is, so this, all the points that I have mentioned, they are basically what? They are basically the PYQs. So do not miss that. Baki gymnosperms are heterospores for sure. Right? They all are heterospores. So they'll produce haploid microspores and megaspores. Theek? Okay. Okay. So they are saying, where will these spores form? Within the sporangia. Where is that sporangia? It is on the sporophyll. And they are arranged beta spirally coiled, right? They'll form cone-like structure. We see that cone in the case of pinus. I'll show you the picture as well. So if the cone is having microsporophyll, micro means small. So microsporophyll will have microsporangia, will form male, basically it is going to form microspores. So it is the male cone when microsporophylls together, because these microsporophylls are the one having microsporangia. So these microsporophylls together, they'll form a condensed structure which is known as male strobili, or you can also call it as male cone. Okay, so microspores will form within this microsporangia and they will develop into the male gametophyte. Okay, and this male gametophyte is highly reduced, confined to only a limited number of cells. You are talking about the pollen grain. So in the starting itself, I told you pollen grain, male gametophyte, embryo sac, female gametophyte. Pollen grain, male gametophyte, embryo sac, female gametophyte. Pollen grain, male gametophyte, and the embryo sac is what? It is the female gametophyte. Keep it in your mind. So the development of pollen grain take place within the microsporangia. So cones which are having megasporophyll, they'll have ovules, right? So these ovules are megasporangia. Take. So ovule is basically your megasporangium. ठीक, it is your megasporangium. Okay, and embryo sac is female gametophyte. Keep it in your mind. And again, I'm writing it here. Pollen grain, male gametophyte. And the stamen that we discuss in Angiosperms, it is not there in Gymnosperms. It is equivalent to microsporophyll. I'm talking about the Angiosperms here. And this stigma, style and ovary, your carpel or your pistil, this structure, we consider it as equivalent to megasporophyll. We consider it equivalent to megasporophyll. Just keep it in your mind. Okay, just keep it in your mind. This is important. You can even note down, you can even take a screenshot. You can even note down, you can even take a screenshot. So, right? So the cones bearing megasporophylls with ovules or megasporangia are called megasporangiate or female cones. Megasporangiate or female cones. So male or female cones, they may be born on the same tree, Pinus, which is monoecious. Cycas is basically beta dioecious. Cycas is basically beta dioecious. Just keep it in your mind. Right? Just keep it in your mind. So megaspore mother cell is differentiated from one cell of the nucellus. So basically what are they trying to say? Now the discussion is around the ovule. If you remember, this ovule structure we also discuss. Where do we discuss it? Beta, we also discuss it in the case of Angiosperms. In that regard, I'm telling you this. And even in Gymnosperms, you will see your ovules are unitegmic also. They are having the single integument, right? Single integument is there. So you know that outer integument, inner integument, this one, their integument is not there, this open end, micropylar end. Here, all that parenchymatous cells present here are nucellus. So nucellus is basically diploid, and from this nucellus, one cell will start behaving like megaspore mother cell. So if it is the megaspore mother cell, if it is a mother cell, it will be diploid and it will undergo meiosis. So it will form, megaspores, right? And then they will further grow. So this is what they have discussed. So megaspore mother cell is differentiated from one of the cells of the nucellus, right? So nucellus is having the it is protected by envelope. So we call it as integuments. So in Gymnosperms, that integuments can be one as well, right? So this is the ovule. So ovule will be there on megasporophyll, right? So that can form female cone. That's it. So megasporophyll mother cell will undergo meiosis, megaspores will be there. So it is the ovule that is the megasporangium, and it will develop into a multicellular female gametophyte, right? So that megaspore will form what? That will form multicellular female gametophyte. Keep it in your mind. And that female gametophyte will have one or two or more archegonia or female sex organs. So this is for the Gymnosperms. And for Angiosperms, I mentioned that points as well. You can keep it in your mind. ठीक? So this is the multicellular female gametophyte. It can have two or more archegonia or female sex organs. Just keep it in your mind. Okay? Just keep it in your mind. Done? Just keep it in your mind. ठीक? So here we have different examples. So I told you Cycas is the living fossil. So same way this diagram is of ginkgo, right? This diagram is of ginkgo biloba. That is also a living fossil. That is also a living fossil. ठीक? That is also a living fossil. So here you can see, unlike Bryo and Pterido, Gymnosperms में male and female gametophyte, they do not have independent free living existence. They'll be within the sporangia. They'll be retained on sporophyte. That is important. So pollen grain will release from microsporangiae, air currents, right? Wind will take it, okay, and then they'll come in the contact with the opening of ovules. Okay, so pollen tube carrying the male gamete grow towards the archegonia in the ovules. So here in Gymnosperms, we are saying archegonia is there in the ovule. So do not confuse it, right? Because in about Angiosperms, we discuss now sexual reproduction in flowering plant. Baki morphology or anatomy, maybe we discuss about it. Okay, so archegonia are there in the ovule, and the pollen grains will discharge their content there near the mouth. Take. So following fertilization, zygote will develop into embryo and ovule will grow into seed. So seeds are not covered here. That's it. That's all. And let me show you some diagrams here. Ye dekho. Baki, this is for the quick revision. I'll share the PDF in our Telegram group, right? I'll share it in our Unacademy NEET English Telegram group. And there is my Telegram group also, Love Biology by Ambika. You'll get it there. So ye dekho, cone, branch tip, specialized for reproduction. So it is having that microsporangia, or if it is a female cone, megasporangia, then ovule is there. Take. So this is the cycles. Hope you guys have seen that. So just look at this. This is your sporophyte, 2N, mature tree. These are the cones. So this is the male cone, which is there on the lower branches. Female cone grow in the upper branches. They will be fertilized by pollen blown on the wind from the male cones. Right? Right? So archegonia is there. So pollen grains, they'll reach there. Pollen tube will definitely form, beta. So here there is no, in Gymnosperms there is no double fertilization. Remember, there is no double fertilization. Okay? Okay? So we also discuss about the winged pollen grains, because that will help in the winged pollen grains. So you have to tell me, that is your homework. We have the exception here. In Gymnosperms we see this, but we have the exception here. We have the exception here, winged pollen grains. That's something you have to tell me. So I believe this part is clear to you. Yes? Done? Any doubt? So endosperm is haploid here. Okay. So it is the pollen tube that will carry the male gamete towards archegonia. Done? So beta, here we have 3 generations na seed mein. Right? This is something that you can write, because sometimes they ask the questions like this. So basically in the seed in Gymnosperms represents 3 generations. Seed in represent 3 generations. Which 3 generations do we have? So when you talk about the parent sporophytic generation, which part will show parent sporophytic generation? So sporophytic generation, it has to be diploid. So you are talking about the seed coat here. You are talking about the seed covering there, right, bache? And not just the seed covering, integuments are also there. And in some seeds, beta, nucellus will stay. It persists, right? We call them as perispermic seeds. In Angiosperms also we discuss. So that is what you can write. So this is the first generation, parent sporophytic generation, right? So nucellus, if present, it is not present always, I told you, right? If present. So this is the parent sporophytic generation. Next is gametophytic. I'm talking about the Gymnosperm seed here. It is endosperm, because endosperm here is haploid, right? Then comes the future sporophytic generation, that is your embryo. Future sporophytic generation, and that is embryo. That's it. That is embryo. That's it. Okay? I hope it's done. Any doubt? This monoecious, dioecious thing is important. Kindly do not forget that. So pollen grains are yellow colored here. 2 air sacs they have, but yeah, exceptions are there. Cheek. So just look at this. Okay. So whenever you will see the pollen grains na, in Gymnosperms, they are showing like this. So these 2 structures are acellular, but they are known as wings as well. Wings of the pollen grains. Matlab, it will help in the pollination. So they are acellular. It's not like that they are living. So pollen grains are produced in large number. So this is what you have to remember. So no chromosome is there in the winged pollen, but yeah, there are some exceptions where winged pollen grain is not there. That is something you have to read. So this is all about the plant kingdom now. And beta, when we talk about the angiosperms in sexual reproduction in flowering plants, we discuss about it. Two classes, monocots, dicots, ठीक, and flowering plants they are. So this is the part that we discussed there. So for your quick revision, you can see the advanced and not so advanced one. So seeds, cone, pollen is there, fruit and flower absent, present. So that's it. That's a quick revision. That's a kind of, quick revision. And now this part is not there in new NCRT, but we will discuss it now. Sexual reproduction in flowering plants, anatomy and morphology of flowering plants. They have this, right? They have this part. Chale. So now let's have a break. This is all about the chapter. So NCRT part is done and dusted, and now we can just go for that only. So let's have a break till 11:20 p.m. Right? So consider it as 10:50. So 10:50 to 11:20. So for half an hour, you guys are having break. Okay? Done? Welcome everybody, welcome. So people, due to technical glitch, so due to technical glitch, I started the class late, but I think you guys are having no energy left. Nahi? Right? You're done with marathons or you're done with biology marathons. You think it is very easy, we will manage it. Isn't it? Isn't it? Please say yes. Huh? Please say yes. No, ma'am. Our energy is too low. We are so done with the biology part. We don't want to read biology. We are done with it. We are focused on physics and chemistry because we know that we are going to get 360 out of 360 in biology, isn't it? Isn't it? है ना? नहीं? Nah, your energy level is very low. Very low. Number of students are very less. Right? In the first marathon, everybody was like, we are having the high Josh, we'll do this, we'll do that. Right? We are full of energy. We have this plan, we have that plan. And now you all are like, nahi ma'am. No chance. हमसे ना हो पाएगा. Class and time. So let's end it now only. Let's end it now only. है ना? Already you have everything on this channel. You have already everything on this channel. And yes, the extra added families. The families, that extra families, na? That families part. Okay, I'll put the recorded video like last year. Don't worry about that. Now you tell me, I'm really interested in ending the class now. Trust me, I'm too tired today. Right? I'm too tired today. Chalo. So let's start. So first of all, that word is tissue. So you know that tissue is the group of cell. How do we define the tissue? Tissue is the group of cell having similar structure, of function, similar structure and similar function, and having common origin. And having what? And having common origin. What do they have? The origin is common here. What do they have? They have the common origin. What do they have? They have the common origin. So what is the word that we have here? That it is the group of cell having similar structure and function and having the common origin. The most important thing is that the origin should be common. The origin should be common. Function can be they can be structurally similar or dissimilar. The function can also vary, but the origin should always be similar. That is what that is something we have to know when we talk about the tissue. Okay, and here, right, we'll make the flow chart as well. So when you talk about the tissue, here you will be talking about two types of tissue. The first one is the meristematic one, and the another one will be the permanent one, right? The another one will be the permanent one. So one is the meristematic one, another is the permanent one. So when it is the meristematic one, right, when it is the meristematic one, when it is what, beta? When it is the meristematic one. So of course meristematic one is what? It is undifferentiated. So when I'm saying undifferentiated, it is not having a permanent function, it is just dividing. So it is a group of small cells. It's a group of small cells. What's that? It's a group of small cells having the ability to divide. So it's a group of small, or I would say active, or I would say immature cells. They are not mature, they are small in size, they are actively dividing. So group of small cells having ability to divide. So they are just dividing. They are adding more and more and more and more cells. Okay, right? They are adding more and more and more cells, but they are not having a permanent function. That is what a meristematic tissue is, right? So that is why we have mentioned the word undifferentiated as well. What is the word that we have mentioned? It is undifferentiated. So here you have the immature cells. What kind of cells do you have? You have the immature cells. So here you have to remember that meristematic cells, because they are small in size, right? Meristematic cells, because they are small in size, so they are thin walled. What are they, beta? They are thin walled, right? They have dense cytoplasm. Their cytoplasm is also very dense, like I'm talking about the properties here. So meristematic cell mein, they are thin walled, right? They have dense cytoplasm and they have conspicuous, right? They have conspicuous nucleus, or you can say that prominent nucleus they have. So they have thin wall, they have dense cytoplasm, they have conspicuous nucleus, plus they have abundant plasmodesmatal connection, right? Abundant plasmodesmatal connection. So do you understand the meaning of plasmodesmata? So abundant plasmodesmatal connection, right? Plasmodesmata. So plasmodesmatal connection. So they have abundant plasmodesmatal connection as well. Just keep it in your mind. So what do they have? What do they have? They are thin walled, right? They have dense cytoplasm. They have conspicuous nucleus. They have abundant plasmodesmatal connection. So these points are important. The question can come from this part. And. If I talk about the meristematic cell, in their case, vacuoles are absent, right? So I'll say no vacuolation. No vacuolation is there. And even if vacuoles are there, they are going to be very, very, very small. Okay. Even if vacuoles are there, they are going to be very, very, very small. This part is clear? Done? Sure. Sure. So densely packed, right? This is what they have. So these are the properties here. I hope this part is clear. I hope this part is clear. So they just have thin wall. So thin wall, and that is the primary wall that they have. They don't even have the secondary wall. So that is why when we are saying thin wall, it is understood. Okay, it is understood. So these are the properties of the meristematic cell where the metabolism is very, very, very high. Is that clear? The metabolism is very, very, very high. Now the permanent tissue, they are the differentiated one. Okay, they are the differentiated one and they are having a permanent function. So here all itself, I'll mention it like you have simple permanent tissue, right? What do you have? You have simple permanent tissue and then you have complex permanent tissue, right? We have simple permanent tissue and then we have complex permanent tissue. So when we talk about the simple permanent tissue, people, here we will have parenchyma, collenchyma, sclerenchyma. Parenchyma, collenchyma, collenchyma, sclerenchyma, because they have one type of cells only, right? Sclerenchyma, one type of cells only. And when it is the complex permanent tissue, it is having xylem and phloem, right? It is having xylem and phloem. So remember it with the help of flowcharts only, okay? Remember it with the help of flowcharts only. So this part is clear. In the permanent tissue, they are differentiated. They have a particular function. So when it is having the simple permanent tissue, it is having one type of cells, right? Having one type of cells, but here they will have more than one type. So parenchyma, collenchyma, sclerenchyma is there. Parenchyma, collenchyma, sclerenchyma is there. And then there is the complex permanent tissue where you have the xylem and the phloem. Okay, now we have to go for what? We have to discuss what is the meaning of, right, meristematic tissue, the types there. So first of all, we start the discussion with promeristem. Do you know what is promeristem? Yes. Do you know what is promeristem? Beta, promeristem is primordial, right? Promeristem is primordial meristem. It is primordial meristem. So basically it is the embryonic one, right? It is the embryonic one. It will form during the embryonic stages, right? So it develops in the beginning. Develops in the beginning during embryonic stages. It develops in the beginning during embryonic stages. Okay, so it will give rise to your primary meristem, right? So it is actually this primordial one, something which is going to start something. So it is basically, it forms primary meristem. What will it do, bache? It forms primary meristem. What will it do? It forms primary meristem. That is what you have to keep in your mind. Okay. Done, bache. So after that, what is the another line that we have? We have the primary meristem. So basically, we have primary meristem, we have secondary meristem. Primary meristem is also having the type. So when it is the primary, of course, it is developed from prostem, developed from promeristem. It is developed from promeristem. So basically, it is developed from the embryonic meristem and helps in the elongation, right, of the plant. Okay, so it appears early in the life and it will form primary plant body, right? It appears early in the life and forms primary plant body. What will it do? It forms primary plant body. What will it do? Yes, please be quick. It will form primary plant body. That is the meaning of this primary meristem. Is that clear? Is that clear? Right? So when you talk about this primary meristem, okay, so when you talk about this primary meristem, here itself I'll tell you, it is a meristematic tissue, so obviously it is actively dividing. Here you have two types. You have apical and intercalary meristem. What do we have here? We have the apical primary meristem and we have the intercalary primary meristem. So it is on the basis of what, beta? It is on the basis of location, right? It is on the basis of what? It is on the basis of location. Overall, I'll explain you the differentiation, redifferentiation, everything that is also there in the plant growth and development. There we will not discuss, we will discuss it here only. So as for the location, it is, so when the word is apical means at the tips, yes or no? Apical means at the tips, yes or no? Yes or no? Yes or no? So it occurs at the tips, right? So in simple words, it occurs at the tips of root as well as shoot. So it is developed from primary meristem and that is how it is. So we can write it like this. We can call it like this. So it is the shoot apical meristem. This one is root apical meristem. So when it is the shoot apical meristem, SAM, root apical meristem, RAM, right, RAM. So shoot apical meristem, it is at the tip. Root apical meristem, it's subterminal as well, right? It is subterminal as well. It is not at the tip because at the tip, root cap is also there. So these are the kind of question you need to remember. Okay, beta. So overall, when you are talking about the apical meristem, right, it increases the length of the plant, right? It is responsible for increase in the length of plant or plant parts, of course, yes or no? Yes or no? That is how it helps. So basically it is responsible for primary growth, right? It is responsible for primary growth of plant. It is responsible for primary growth of plant. So it helps in the elongation of plant. How does it help? It helps in the elongation of plant. How does it help? It helps in the elongation of plant. Is that clear? Everybody here in the class, is that clear? Are you sure? Yes. So this intercalary meristem, right, it is present in between the mature tissue. So intercalary, in between. So it is present in between the mature tissue. So if I have to give you the example, this is suppose it is at the nodes of monocots. Monocots may you will have grasses, just like grasses. Are you getting my point? So it is at the nodes of monocots like grasses. So whenever, you know, the grazers, they eat the grasses are able to regrow because of this intercalary meristem, right? Because of this intercalary meristem. So it is present at the base of, it is present at the nodes of the monocots, right? At the base of the internode, or you can say that at the nodes of the monocot stems. Is that clear? So even it is present at the base of leaf, So that is how it helps. So it is intercalary in between. So in between the mature tissue it is present. Is that clear? In between the mature tissue it is present. Is that clear? So shoot apical meristem is there, root apical meristem is there, and then there is the intercalary meristem. Then there is what? Then there is the intercalary meristem. So this part is clear. This part is clear. So whenever you are talking about the intercalary meristem, remember that will help in regrow the parts. Which parts are we talking about here? Which parts are we talking about here? So they will help in regenerating the parts removed by the in grasses. So in grasses, regenerate the parts yes. So what will it do? It will regenerate the parts which are removed by herbivores. Herbivores are the one that will eat on plant material. So this part is clear. This part is clear. The role of intercalary meristem clear. The role of intercalary meristem here. Now, the next part here that we need to remember is that whenever you talk about the primary meristem, then I'll touch the secondary meristem topic as well. So primary meristem, it will form primary permanent tissue. It is the one that will form primary permanent tissue. Is that clear? It is the one that will do what? It is the one that will form primary permanent tissue. Any doubt? Yes. Any doubt here? Any doubt here? Are you sure? Chaliye. So now we have to understand three words. The first one is differentiation. The same is there in plant growth and development. That is why I always discuss plant growth and development right after the anatomy, because then we are able to finish this part. So we have to discuss differentiation, de-differentiation, and redifferentiation. So students, keep this thing in your mind that we have the primary meristem, and once it will get differentiated, right? We have the primary meristem, and once it will get differentiated, once it will do what? Once it will get differentiated, after the differentiation, what is going to happen? After the differentiation, what is going to happen? Yes, people, after the differentiation, you will get the permanent tissue. So basically what is happening? Initially, these cells are just dividing. They are small in size, they are just increasing the number. Now, when they acquire a permanent function, what they do, bete? When they acquire a permanent function, right? What are we saying? A permanent function when they acquire. So then, of course, we call them as permanent tissue. And this process is actually known as differentiation. This process is actually known as differentiation. So when we are saying differentiation, bache, right, do you understand the meaning? Right. So when we are saying differentiation, so in differentiation means your primary, right? This is your meristematic. And especially, of course, I'm talking about the primary meristematic tissue. This is your meristematic tissue. So even in, as I said, in plant growth and development, this part is there. So it is DM. Firstly, differentiation. After differentiation, there is the maturation. After differentiation, there is the maturation. So these cells, they are immature, right? They don't have a permanent function. These cells, they are immature. They don't have a permanent function. So when these cells, they will become differentiated, means they will acquire a permanent function. Just say in our body, we all developed from the single cell zygote. Initially, the zygote will undergo cleavage, multicellular condition will be there, so many cells will form, right? This is something that we already know. So many cells will form, but after some time, some cells are forming the muscle, some cells are there that are going to be the part of nervous tissue, some cells will be there and they will be the part of excretory tissue as well. So this is something that we have, right? So when you are talking about the meristematic tissue people, so these cells, they are the group of actively dividing cells. They will become mature, they will become differentiated. So once they become differentiated, they will get a permanent function, right? Now they will not just divide, right? They'll do a permanent job. Just for an example, your parenchyma, collenchyma, sclerenchyma, they have a proper function. They are not just dividing. This is what de-differentiation is, right? This is what de-differentiation is. Now let's suppose, what's going to happen? This is your permanent tissue, or I would say primary permanent tissue. Now this permanent tissue, right? Now this permanent tissue, Beta, there is one more word, that is de-differentiation. There is one more word, that is de-differentiation. What is that word? That is de-differentiation. So when you people talk about the permanent tissue, when you people talk about the permanent tissue, once again, Rohini MBBS, 50 concept series, please continue. Rohini, if you watch our lectures regularly, maximum topics are covered there. And after that, whatever is left, we are discussing that in the marathons. Why you people keep on, asking for the things which we have already done? Huh? Huh? No. There is no need to read the secondary growth for the NEET. So permanent tissue, when it will become de-differentiated, means it will again regain the ability to divide. It will regain the ability to divide. Are you getting my point? It will regain the ability to divide. So this is the permanent tissue. Once it will undergo de-differentiation, right, it will regain the ability to divide. So after de-differentiation, right, it will become secondary meristem. What will it become? Secondary meristem. Right? So now you will see that this is the permanent tissue having a permanent function, right? This is a permanent tissue having a permanent function. So now it will undergo de-differentiation. Now it will undergo de-differentiation. It will undergo what? It will undergo de-differentiation. What will it give you? It will give you the secondary meristem then. Why do we call it secondary, not primary? Because obviously it is not formed during the embryonic stages. It will form later on after the de-differentiation. So this is a PYQ, that how the secondary meristem form. It will form due to the de-differentiation of the permanent tissue. Is that clear? Is that clear? Now once again, when this secondary meristem, when again it becomes, just it's like, I always give this example. Let's suppose you are reading, you're studying, you have become a doctor, right? You got a job, you are a reputed doctor, but you don't want that. Right? You are like, no, I want to be an IS officer. So you are like, you will start studying again. So initially you got a job, right? You were not studying, you were working, and then you realize, no, I want to read again, I want to study again, I want to do something else. Right? So again, you will enter in that reading phase. So same way, there is a permanent tissue. That permanent tissue is like, I again want to go for the division phase, right? I again want to go for what? I again want to go for the division phase. So that is basically, it will become de-differentiated. After de-differentiation, what is going to happen? After de-differentiation, and now you are studying, you'll give the IS exam and you will become an IS officer. Take, so means you will be re-differentiated. You again, right? You again will become, right? You will again acquire a permanent function. So re-differentiation, due to which you will get secondary permanent tissue. What will you get then? You will get secondary permanent tissue. Is that clear? Is that clear? So that is something you need to keep in your mind. Okay? That is something you need to keep in your mind. Is that clear? Is that clear? Sure? Okay. So let's discuss it. So this secondary meristem is also known as lateral meristem. Why? Because it is present on the lateral side. So lateral meristem, cylindrical meristem, or secondary meristem, whatever you want to call it, you can. So why do we call it as secondary meristem? It is present on the lateral sides. Number one. Number two, it increases the girth. It increases the girth, that is diameter, circumference. So that's something you have to keep in your mind. Understood? Understood? Sure? Everybody here in the class, sure? Yes, please. I hope this portion is clear, crystal clear to everybody. Take. So it is the lateral, it is the cylindrical, it is the secondary meristem. It is present on the lateral side. It is responsible for increasing the girth. Okay. It is responsible for increasing the girth. And how will it form? It forms due to, again, this word is important, already mentioned it, but again I'm telling you, due to the de-differentiation. how will it form, bache? It will form due to the de-differentiation of permanent tissue, primary permanent tissue. De-differentiation of primary permanent tissue. That's something you have to keep in your mind. Understood? Understood? Yes, bache, I'll go faster. I'll speed it up. So up to this part, all clear? Up to this part, all clear? So we have the example. We have vascular cambium. Cocambium. Cocambium. It is also known as phallogen. It is also known as phallogen. So you can write it. You can write it, right, bache? So vascular cambium of root. So I will specify it slowly, of root, and it is developed from. Now we don't understand the. Now we don't read the secondary growth here, na? So vascular cambium of root developed from conductive tissue, right? Cocambium, phallogen. Then you remember interfascicular cambium. Interfascicular cambium. Yes. Do you remember that? Interfascicular cambium. We'll tell you once we'll discuss the structure of dicot stem. So these are the examples that we have. Okay. These are the examples that we have. Is that clear? Is that clear? Anybody having any doubt? Yes. Anybody having any doubt? So cocambium, interfascicular cambium, right? And these two are of course, these two are of your dicot stem, isn't it? Dicot stem. So they are what? They are secondary in origin. They are secondary meristem. Is that clear? Yes. So here, one thing I will about the intrafascicular cambium, na, I'll discuss it in the diagram part. So you can see the position as well. I hope it is clear to everybody. I hope it is clear to everybody. Done? Sure? Any doubt? Any doubt? So after that, let's come to the diagram part. So diagrams are very important. We are not allowed to miss. So when I told you about the root apical meristem, I told you it is subterminal. Subterminal means it is not exactly at the ends. It is not exactly at the tip, a little away from that. So because here at the tip you have the root cap. Okay, then here you have the root apical meristem. Here you have the root apical meristem. So what else you need to remember? See, central cylinder is there. Surrounding my cortex is there. The protoderm is there. There are some initials. So yes, now the word protoderm is written. Protoderm is written. Do you know the meaning of protoderm? Do you know the meaning of protoderm? Beta, as per Hemberland's theory, there are different theories here which we do not discuss, but the protoderm word is written here. So it will form the epidermis or epiblemma. This is what you can write down. This is what you can remember. It forms epidermis, you know, na, outer covering, or epiblemma. Epiblemma is the epidermis of roots. Epiblemma is the epidermis of roots. Is that clear? Epiblemma is the epidermis of roots. Is that clear? Is that clear? Right? So in dicots, it forms root cap, but in monocot, root cap is from a separate layer. Take. So in dicot, it forms root cap. Take. So here you can see shoot apical meristem, right? Axillary bud, which is also formed due to the shoot apical meristem only. It is present at the base of the leaf. Right, it is present at the base of the leaf, right? The axillary bud it is. So it is differentiating vascular tissue. That is it, right? The diagram part is important. Kindly do not skip that. Now, so this is for the quick revision, right? The flow charts are there. Now, let's come to the meristem. This meristematic tissue is done. Now, let's come to this part. So here, again, I'll go very quick, right? You know that in the detailed sessions, we have completed it, right? In the, we have discussed each and every aspect of that. But now we have to stick to the NCERT. So let's speed up accordingly. Okay, let's speed up accordingly. Done? Done? So first thing is parenchyma, right? First thing is parenchyma, which is the major component within the organ. And cells here are generally isodimmetric. So the word is generally isodimmetric. It is not. Iso means same diameter. So it is not always, but generally it is like that. Cells are having different shapes, and here walls are thin and made up of cellulose only. They are not lignified. They are not dead. It is living. It is living. So either it is very closely packed, means compact, or it can have small intercellular spaces, right, bache? So it is having different functions. Just I'll just quote one function here, right? Parenchyma is one type, parenchyma. Parenchyma having air spaces. So if they are having air spaces or cavities, they are going to provide buoyancy to the hydrophytes. Plus, in the case of parenchyma, one thing is idioblast. I don't know whether you remember it or not. This is also the modification here. Idioblasts. Idioblasts. So, bache, they do what? They store the, right, in their case, what will you see? That these are the cells which will store protoplasmic substance, or I would say ergastic substance, right? It will store ergastic substance. So when I'm using the word ergastic, it's kind of non-living, and here you can say metabolically inert, means it is not participating in the metabolism. It is not participating in the metabolism. So what are these kind of, so these are the ergastic, you can call it as non-living or metabolically inert. These are your tannins, resins, etc. Done. Idioblast. Then this parenchyma, if you remember, it can also form chlorenchyma, means when parenchyma cell is having chloroplast, it will do photosynthesis. So it is chlorenchyma. It is chlorenchyma. So these are the major points that I have picked up, right? These are the major points that I have picked up. So isodimmetric, may or may not have intercellular spaces, right? And their function. Now come to this part, collenchyma. So here you know that edges, right? The thickenings are there around the corner. The thickenings are there around the corners. Diagrams are also important. Directly question can come from the diagram part as well. So here just have a look. So it is, see, it occurs the layer below the epidermis in dicot plant. So this is something that you can note down, that in the dicots, when you will see the section of dicot, the hypodermis is collenchymatous. The hypodermis is collenchymatous. So it can be homogeneous, like a continuous layer, the proper cells, or it can be in some patches, like some cells are at this point, some cells are at this point. It depends. So thickening is there. Why? Because at the corner, cellulose, hemicellulose, and pectin is deposited. So revise it in this way. In parenchyma, just cellulose, but here these three are there. Now the speed is fine. Now is the speed is fine. Let's speed up, guys. So collenchymata, again, they are having different shape. They assimilate food, and intercellular spaces are absent there. So in parenchyma, so focus on the differences here. Focus on the differences here. Please write down the differences with the lecture itself. Please write down the differences with the lecture itself. So intercellular spaces are absent there, but in parenchyma they may or may not be, right? They can be, they cannot be. So it will provide the mechanical support to the growing plant, such as the young stem and petiole. So who's going to provide the support to the young stem and petiole? It is collenchyma. Do we need to remember it? Of course. Of course, we cannot miss that. Okay. So leaf lamina, its petiole, they are having this. Okay. Okay. Done. So collenchyma, beta, always remember, it is absent, and it is not even the part of your vascular bundle. So it is absent in, right? This is something that you can write. So it is absent in, please write down the particular points, and the mature or the woody plants. It is absent in mature or the woody parts, right? Or the woody parts, right? In stem, root, and monocots. In dicot stem, after secondary growth, even in root and in gymnosperms. Done. Now sclerenchyma. These are the two structures that you have to keep in your mind. So this one is the fiber. This one is the fiber. Look at this. It is elongated. It is pointed. It is elongated. It is pointed. Now look at the sclerids here. So sclerids, maybe you will see the pit and the pit channels are there. Right? Look at this. These are the sclerids. These are the fibers. So they are considered as dead. No protoplasm you will see in the mature state here. right. So this is, yes, this is important. Okay, this is important. So write down. Yes, clear. So overall you can write one more thing, that this collenchyma, regarding collenchyma I have mentioned one point here now, I just forgot the another. So collenchyma is absent in roots or stem. It is absent in roots and monocot plants. ठीक, it is absent in roots and monocot plants. मतलब roots में, पूरे roots में, and in the monocot plants it is absent, and in the monocot plants it is absent. Is that clear? So it is absent in roots and in monocot plants. It is absent in roots and in monocot plants. Is that clear? Is that clear? Is that clear? Sure. So these are the kind of lines from where the MCQ can frame. So do mention it. Now sclerenchyma having long narrow cell with thick and lignified cell wall. So what is the word that we have here? What is the word that we have here? Thick and lignified cell wall. And in their case, few or numerous pits are there. So dead and without protoplast, told you already. Dead and without protoplast. Right, they are dead and without protoplast. So yes, they have the different structure, right? So they can be fibers or sclerids. So fibers are thick walled, elongated and pointed. Just show you the you have seen the diagram, that's it. Sclerids, spherical, oval, cylindrical, highly thickened dead cell with very narrow cavity, right? So where do you see it? In the wall of nuts, pulp of fruits like guava, pear, sapota, and seed coats of legumes and the leaves of the tea. They have that and they support the mechanical. So examples are important. Are you done with that? Right? And where do you see it? When you will see the sections, you will see that it is in the, where do we find it? We find it in the hypodermis of monocot stem. Where do you get it? You will see it in the hypodermis of monocot stem. Hypodermis of monocot stem. Where do you see it? In the hypodermis of monocot stem. Done? Done. Chaliye. So that's it. Xylem, phloem. Let's have a quick revision of that. You know that they are forming the vascular bundle. What do they form, people? They form the vascular bundle, right? So lignin and all, sclerine wala part done, sclerid done, right? And yes, sclerids and the fibers. Hi. Sclerids and the fibers. Ye dekho. These are the diagrams. So in the case of fibers, right, bache? So in the case of fibers, so you have to remember that we have different types of fibers, right? Surface, xylary, phloem fibers are there. Phloem fiber example is very important. Xylary. Phloem fibers, which are also known as bast fibers. Remember? So surface fiber mein you have the example of coir of cotton. You have the example of coir of cotton, even coir of coconut and the coconut and the cotton fibers. These are the examples. Okay, so cotton fibers and the coir of coconut. These are the examples. Then xylary fibers are there. Xylem is basically the woody part, right? So they are very hard fibers. So xylem means your secondary, that is your secondary xylem, na wood. So it is also the woody fiber. And then this phloem. The example here is very important. It is a bast fiber and they are basically commercially used. Okay, they are basically commercially used, right? So they are having the commercial value. So they are extra xylary as well, commercially used. जैसे कि your jute. Jute is the example there, right? Jute is the example there, right? Hemp is the example there. Is that clear? Jute, hemp, flax. It is NCRT. They are the examples. Okay, so this fiber thing is important, especially this bast fiber. Okay, this fiber thing is important, especially this vast fiber. Is that clear? Sure. Okay, so now we have xylem and phloem, tracheid, vessels. These are the examples here. Dekho, tracheids, vessels, right? So you see, look at the bundles here, right? You have the xylem, you have the phloem, the way they are associated, xylem, phloem, cambium in between. So that is something we need to discuss now, right? We'll stick to the NCERT, of course, right? Without any doubt, we'll stick to the NCERT. So coir, hemp, cotton, jute wala part, do revise it. Then let's stick to the NCERT and let's revise it. So firstly, when you are talking about the xylem, you know that here the movement will be unidirectional. What kind of movement is there? Unidirectional. What will it carry? It will carry water and minerals. What will it carry? It will carry water and minerals. So what do we study here? We study here the tracheids, the vessels, xylem fibers, xylem parenchyma. Xylem fibers or xylem parenchyma. So basically these three, they are dead. This one is living. This one is living. Now these tracheid and vessels, they are actually known as beta tertiary elements. Why? Because they play a role in the water conduction. Okay, they are the one that play a role in the water conduction. Is that clear? Is that clear? Yes or no? Yes or no? Now, vessels. I'll pick up the important questions here first, then NCERT, of course. Vessels are the feature of Angiosperms, means it is an advanced feature. And where do you see it? You will see it in the Angiosperms. So vessels are what, beta? So vessels are the feature of the Angiosperms, right? Basically, it is an advanced feature. It is an advanced feature. That is what I'm trying to tell you. Now the point here is, okay, now the point here is, in few Gymnosperms, basically that's an exception. So in Gymnosperms of order Neetail, there is an order Neetail, the Gymnosperms of that order Neetails, right? जैसे कि your Neetum, Welwitschia, Ephedra, they contain xylem. Sorry, they contain vessels. They contain vessels. So yes, this is important. Okay, so this is the exception actually, that Ephedra, Neetum, Welwitschia, they are having the vessels, and even there are, right, even there are certain Pteridophytes also, right? In some Pteridophytes as well, like your Selaginella, Adiantum. Today itself we have discussed these examples. They have vessels. Okay, one more example is there which I'm not able to recall right now. So that's it. Okay, so they have, right, so Selaginella is also there, Adiantum is there, one more example is there that I'm unable to recall at this point of time. So here also you will see the vessels. Okay, here also you will see the vessels. So here they go. So xylem, let's have a quick revision of that. Question can come from the diagram indeed. So conducting tissue for water and mineral, and it is unidirectional from root to the stem and leaves. From root to the stem and leaves. So it provides mechanical strength to the plant parts, right? It will provide what? It will provide mechanical strength to the plant parts. So as I said, it is having tracheid, vessels, fibers and parenchyma. So Gymnosperms, they lack vessels. So in the NCERT, you can write down exceptions. Just now I told you, so you can also write the exceptions there. Then tracheids are elongated. They are tube-like with thick and lignified walls, and their ends are tapering. Right? What kind of ends do they have? They have tapering ends. What kind of ends do they have? They have tapering ends. It's not pointed. It's like that. Take, and then they are dead and without protoplasm. So why are they dead? Because they have to carry the water. They need more space there. So protoplast will be absent there, and more water will go. Is that clear? More and more water will go. Is that clear? Right? So inner layer of the cell wall, they have the thickenings. So flowering plants may, they are the main xylem, tracheids and vessels are the tertiary elements. So vessels, bache, they are long cylindrical tube-like, made up of many cells called vessel members. So many vessel members will together make vessels. They are lignified, having large cavity, and the cells of vessels are also dead. I told you already, without protoplasm. So vessel members, right, together they are connected through the perforations. Like perforations means pore. Let's suppose this is the one. Here having pore, here having pore. Next one is also having pore. So with the help of these perforations, they are connected. They have to make a continuous channel. Now the chapter transport in plants is deleted from your partner. So it used to be there. It is a wonderful, it used to be the part of your syllabus. And that's a wonderful chapter. Right, that's a wonderful chapter. Then, so xylem fibers have high thick walls, obliterated central lumen, right? Either septate or aseptate. So xylem parenchyma, it is living, it is made up of cellulose. They will store the food. That's it. So radial conduction of water take place by ray parenchymatous cells. I'll tell you this thing in the sections as well. So any doubt? Any doubt? Any doubt? Sure, bache? Right? So the development, protoxylem, metaxylem, and dark exarch, I'll discuss later. Then come to the phloem. So in the case of phloem, what do you have? So phloem will have sieve. Yes, the phloem will have what? The phloem is going to have the sieve tube. It is going to have the companion cells, phloem parenchyma, and phloem fibers. I told you, they are also known as bast fiber. I told you which one are economically important. So in the case of this phloem, these three are living. This one is dead. Okay. And here, this sieve tube at maturity, it is enucleated. It is without nucleus. At maturity, it is enucleated. It is without nucleus. Are you getting my point? Are you getting my point? So this is something you have to remember. So basically you have sieve tube, and each tube consists contain sieve elements. Each tube contain sieve elements. Is that clear? Is that clear? Sure. And now if you think that if they are without nucleus, who's going to take care of their function? The companion cell, right? So sieve tube and companion cells, they are all together. So basically the loading and the unloading of food is there, right? The loading and the unloading of food is there. So here I'll discuss few points. See, whenever we talk about the xylem and phloem, right, they are involved for long distance transport. They are involved for long distance transport, right? They are involved in long distance transport. So when we say long distance transport, so basically the word that we have is translocation. What is the word that we have? It is translocation, number one. So phloem is food, of course. It will help in the transport of organic substance, right? And basically in phloem, it is the sucrose, right? It is the sucrose that will transport biodust, right? It is the sucrose that will pass through that. Why? Because you know, the sucrose, it is the non-reducing sugar. So actually the sucrose will be there in the phloem. And in the case of phloem, the transport is bidirectional. It is bidirectional. What is it? It is bidirectional. Are we clear here? Done? Done. So these are few important points that you need to know. And whenever, so companion cell is there, it is having the nucleus. Sieve tube is not having the nucleus. And here, bache, when there is a loading and unloading of food, so this is an active process. So if I'm saying it is an active process, so it simply means ATP will be used, whether it is the loading or unloading. So it is the pressure gradient, the bulk mass flow hypothesis we used to read for this food transport. Okay. Okay, so they all are PYQs that I have covered here. Now, when it comes to the phloem transport, right, food material usually from leaves to to other part of the plant, right? So it is having seed tube elements, companion cells, phloem parenchyma, phloem fibers, right? Phloem parenchyma and phloem fibers, right. So when you talk about this phloem parenchyma, it is absent in most of the monocotyledons. So it is absent in most of the monocotyledons. Is that clear? Are you sure? It is absent in most of the monocotyledons. Is that clear? Is that clear? Are you sure? Are you sure? Yes? So gymnosperms, even they do not have the companion cells, they have albuminous cells and sieve cells. So sieve element, yeah, sieve tube having element cells, they are in angiosperms, but sieve cells are there in your gymnosperms and albuminous cells are there. Okay, so sieve tube elements are again long tube-like structure arranged longitudinally associated with companion cells. Done and dusted. Then their end walls are perforated in a sieve-like manner. Their end walls are perforated in a sieve-like manner and they form the sieve plates. ठीक? So a mature sieve element, it is having peripheral cytoplasm and large vacuole because it is what? It is not dead. It is not dead. Are you getting my point? Right? So they lack the nucleus. So who's going to take care of that? Companion cells. Okay. Okay. So other than that, you can see sieve tube element companion cells. They are connected by pit fields, right, bache, and they have common longitudinal walls. Okay. So the topics that we have, the questions that we have discussed earlier, now they are the most important one. Okay. They are the most important one. Right? Then see phloem parenchyma, and already I told you how the food loading and unloading will be there. That is an active process that will require ATP, and basically the gradient is required, pressure gradient, the bulk flow hypothesis we used to read, the mass flow hypothesis. It's not just one thing that will pass there, it is basically the mass, okay, the mass. So baki phloem parenchyma is made up of elongated tapering cylindrical cells which have dense cytoplasm. The cell wall is composed of what? Cellulose, and has pits through which the plasmodesmatal connection exists. That's it. That's it. So this line I have initially mentioned you, right? These are the points that we need to pick, bache. So in the most of the monocots, phloem parenchyma are absent. In most of the monocots, phloem parenchyma is absent, number one. Then phloem fibers, also known as bast fibers, they are made up of sclerenchymatous cells because they are also dead. It is the fiber. So these vast fibers, they are absent in primary phloem but are formed in secondary phloem. Okay, okay. So these fibers, bache, they are elongated and unbranched and have pointed needle-like apices. So cell wall of phloem fiber is quite thick. That's it. They lose protoplasm. They are dead. So this line also in the starting itself we have discussed now that they are commercially used. We have completed it initially. Okay. Now after this, we have to start the tissue system, right? So before that tissue system, again I'll discuss few points with you all here. But there are two words. One is protoxylem and another is metaxylem. Then there is the word protophlloem. Then there is the word metaphlloem. metaploem, right? Protoploem and metaploem. Now the word is protoploem, right? It will form first. When we are protoxylem. So what is the meaning of protoxylem? It is the first formed primary xylem. So it is first formed, basically, that's the point. So it is first formed primary xylem, right? It will form first. It will form first, where lignification will start before the completion of elongation, right? So it is having smaller tertiary elements. They are having smaller tertiary elements. Is that clear? Right? So then comes the metaxylem. So they are late formed primary xylem, right? It will form late, but it is primary xylem only. It's not like that it is secondary. This one will form first. So they form during the primary growth of the plant only, right? They form during the primary growth of the plant only. So they are late formed primary xylem having larger tertiary elements, having larger tertiary elements. So proto and meta, right? Proto and meta. Then same is for the protoploem and metaploem. So protoploem will form first, metaploem will form later. Protoploem will form first, metaploem will form later. Is that clear? So it is the first formed primary phloem. So it is having narrow sieve cells and it is having wider. That's it. That's it. Right? So this is basically the sieve cells or sieve tube. Obviously, so this is what we have. So protoxylem, metaxylem, clear? So remember, after double staining, it is with safranin and fast green. Right? It is with safranin and fast green. So xylem gives red color or phloem gives green color. Remember that. Okay, after double staining. How? After double staining. Is that clear? Is that clear? Sure. Any doubt? Chali, let's start. So now, bache, we have to go for the tissue systems. So when it comes to the tissue system, this, right, how are we going to revise it? We will revise it with the help of this flowchart. But yes, again, remember, we have two words, right, when it comes to the xylem, on the basis of xylem arrangement. On the basis of xylem arrangement, देखो, we have two things, exarch and endarch. Exarch and endarch. This is important. Okay, exarch and endarch is there, and this is important. This is important. So exarch outside. So here what is happening? Let's suppose if this is a section. In the case of exarch, your protoxylem, it is towards, it is facing periphery and metaxylem towards pith or center. Center. Okay, like let's suppose here, these small having small tertiary elements, right? So let's say this one is the larger. So it is from periphery. It is like this now, from periphery towards center, from periphery towards center. So what kind of arrangement it is? Centripetal. What kind of arrangement it is? It is centripetal. What kind of arrangement it is, people? It is centripetal arrangement, right? Centripetal arrangement. Okay, so such type of arrangement you will see in the case of root. And dark, your protoxylem is facing the center. And metaxylem, periphery. So it is from center towards periphery, towards periphery. So centrifugal. And where will you see this? Leaves or stem. Is that clear? Is that clear? Is that clear? Done? Okay, so this is what we have. Okay, so bache, if I have to increase the speed, what I need to do? Should I touch all the topics? Should I just not explain? Should I touch all the topics, Bhanu? Chalo, touch everything. Let's not discuss it. Right, right. See, if NCRT got updated, but here and that, things are on the basis of what? Basically, what is the point here? Right, you are right that NCRT got updated, but basically they just rectified their few mistakes. Not all the mistakes. They have rectified their few mistakes, and teachers are teaching you as per actual concept only. So there is nothing to worry about, Arijit. So this part is clear. So let's increase the speed and then let's revise with the help of flowchart. So tissue system, epidermal tissue system, ground or fundamental, vascular tissue system. These are the three things that we have. These are the three things that we have. Right? So epidermal tissue system is there, ground tissue system is there, vascular tissue system is there. Right? Epidermal tissue system is there, ground tissue system is there, and then we have the epidermal tissue system. Right? Then we have the epidermal tissue system. This is what we have, yes or no? Everybody please help me out. Everybody please help me out. Done? Done. So here we have epidermal tissue system will have epidermis and epidermal outgrowths, right? So when it is the epidermis, it is usually single layered. So if it is usually single layered, what is the word that we will use? Uniserrated. And right, it can be multi-layered as well. For multi-layered, we can also use the word multiserrated. So when it is unilayered, right, and multi-layered, it is in the case of, medium, ficus, right? Basically, in the case of your orchids, you will see the multi-serrated epidermis is there, and it is single layered. Take so in the epidermis, epidermal outgrowths. Epidermal outgrowths means when from the cells of the epidermis, some things are arising, some structures are forming, right? Then you have trichomes or here, and you have some emergence or prickles. So these trichomes, they are the part of shoot system. They are the part of shoot system. Please write down here only. Right, please write down here only, please complete the tissue system here only. So it is the part of shoot system and are usually multicellular. What are these trichomes? They are usually multicellular. What are they, students? They are usually multicellular. Please write down. Please write down. They can be branched or they can be unbranched. They can be branched or unbranched. So the question will come from their function, and please note down that function. Please note down that function. They help in preventing water loss due to transpiration. So it prevents water loss due to transpiration. Prevents water loss due to transpiration. Is that clear? Is that clear? Yes. Yes. So that is what we have to understand. Emergence, prickles, right? They are again multicellular epidermal outgrowths, right? They are also multicellular epidermal outgrowths. They are multicellular epidermal outgrowths from cortex. So that is not very important, but yeah, this trichome is very important. Now, come to this part. Now, when you are talking about the ground tissue or fundamental tissue, it is basically differentiated in roots and dicot stems. We'll discuss it, right? See, sometimes this ground tissue is differentiated. It is having hypodermis, cortex, endothermis, pith, and medullary. This should be the sequence. Outermost epidermis, and then below epidermis, hypodermis, then cortex, then endodermis is the innermost layer of cortex, right? Then pith and medullary ray. In the vascular tissue system, you have pericycle and you have vascular cylinder. You have pericycle and you have vascular cylinder. So vascular cylinder means vascular bundle means you have the xylem and phloem, and we need to talk about their arrangement. Right, we need to talk about their arrangement. What we need to discuss? Their arrangement. So before going forward, let's discuss in the epidermal tissue system, we are also having this stomatal apparatus. So stoma. So what's the opening there? Right, what's the stoma is the word singular, and the stoma or stomata. So basically what do you discuss? It is the opening. It is the opening. Just have a look. Have a look. So it is there in your epidermal tissue system. It is there in your epidermal system. So you know that epidermis cells, right? They are in the case of dicots. In the case of dicots, this stomata is opening. The stomatal pore is opening. What is happening through this? There is the gaseous exchange. There is the gaseous exchange and loss of water in the form of vapors. Loss of water in the form of vapors, right? What do we have? Gaseous exchange is there and loss of water in the form of vapors, right? That is transpiration. So this is the function of the stomata. Okay, important here it is transpiration, you know, not necessary evil. So now when you see in the case of dicots, this is that opening. It is surrounded by guard cell. So in the case of dicot, that guard cells are basically bean shaped, or we can also call it as kidney shaped. Bean shaped or kidney shaped they are. Bean shaped or kidney shaped they are. What are they? They are bean shaped or kidney shaped, right? What are they? They are bean shaped or kidney shaped. Is that clear? Is that clear? Are you sure? bean shaped or kidney shaped. ठीक? Right? And this in the surrounding, they are also the epidermal cells. What do we call them? Subsidiary cells. They are also known as accessory cells. So here remember that in monocots, these guard cells, they are dumbbell shaped. And when you look at this part, another important point is that inner lining, right? That inner lining here, it is thick. That's important again. This inner lining here is thick. This inner lining here is thick. And here we have see, radially oriented cellulose fibers. So these are the important points. Okay. So diagram, with the help of diagram, you can revise everything. Right? Yes, with the help of diagram, you can revise everything. So stomata structures done, guard cells done. So remember, outer wall here is, right, thin, but inner wall is highly thick. Right? They have chloroplasts that will regulate the opening and closing of stomata. So this opening and closing of stomata is associated with turgor pressure. Right? It is associated with turgor pressure. Okay. Okay, subsidiary cells are also there, and stomatal apparatus done. So each stomata bean shaped, enclosed stomatal pore done. So all the important points are discussed here. And remember, cuticle is absent in roots. So in roots, that epidermis is known as epiblemma or piliferous layer. That's it. Take, this is important. Okay, this is important. Any doubt? Any doubt? So yes, in the ground tissue, right, except all tissue except vascular bundle and epidermis, right, they are forming the ground tissue. Again, PYQ. So parenchyma, collenchyma, sclerenchyma, all are the part here. Parenchyma are usually there in the cortex, right, pericycle, pith and medullary rays, and in the primary stem and roots, right. And in leaves, that ground tissue is known as mesophyll. What do we call it? We call it as mesophyll. Okay. Okay. So this ground tissue is actually differentiated. Okay, this ground tissue is actually differentiated. It is having hypodermis, it is having cortex, it is having endodermis, it is having pith, it is having medullary rays. Take, it is having pith, it is having medullary rays. Okay, it is having pith and it is having medullary rays. Is that clear? Is that clear? Okay, done. Done. So endodermis is there. Endodermis is the inner most layer of cortex. Again, a previous year question. Right? Again, a previous year question. And it is absent in monocot stem. Again, a previous year question. Please write down. Please write down. Right? We'll discuss it individually in all the sections. Okay? So it is, sometimes this ground tissue is differentiated, sometimes it is not. So this diagram part is clear. Sorry, this flow chart is clear. Then I'll discuss the different types of vascular bundle. This flow chart is clear. If vascular bundles are clear, the section comparison will hardly take 10 minutes. Okay? The section comparison will hardly take 10 minutes. Even in the top 50 concepts wala topic, we have revised it. Okay? We have revised it there. In the top 50 concept wala topic, we have revised it. Okay? Okay. Done? बेटा ये देखो ना, cortex is the part of ground tissue, endodermis, pith and medullary ray. But pericycle, even if you will read the NCERT, pericycle is just like the boundary of the vascular tissue. This pericycle. Okay, this pericycle. So focus on the vascular bundle, and then let's discuss it further. So what we have to discuss? We have to discuss the vascular bundles now, and the topic of vascular bundle here is important. So we have radial vascular bundle. So when we are saying radial vascular bundle, R for radial, R for roots. And in the roots, already I told you the xylem arrangement is exact. Keep it in your mind like this. Okay. So it is exact. So it is basically centripetal arrangement. Okay. Okay. Now, in the radial vascular bundle, xylem and phloem, they are present at different radii, or you would say different radius. Yeah, radii is plural here. So it's like that. Imagine if this is your vascular bundle, right? Imagine if this is your vascular bundle. So it is exact. Protoxylem is facing the periphery. Metaxylem is facing the center. Metaxylem is facing the center. So that is how it is. That is how it is. And here you will have your phloem, right? Here you will have your phloem. So this type of vascular bundle where xylem and phloem is present at different radius. Okay, xylem and phloem is present at different. So this is xylem, this is phloem, this is xylem, this is phloem. Radial vascular bundle. Then comes conjoint collateral vascular bundle. So when it is conjoint collateral vascular bundle, it is conjoint collateral vascular bundle. What is the answer here? So it is the conjoint collateral vascular bundle. So conjoint collateral. So basically here xylem and phloem are present at same radius. They are present at same radius. Right? The radius is same here. The radius is same here. So conjoint is the word when they are present at same radius. So stem and leaves will show you the same. Stem and leaves will show you the same. So you have conjoint collateral, you have conjoint bicollateral, etc., etc. Different types are there. So in our syllabus we have this conjoint collateral. In our syllabus we have this conjoint collateral. So we have conjoint collateral, bicollateral and concentric, but conjoint collateral is the part of our syllabus. So when you are saying collateral, when you are saying collateral, so here first of all conjoint means xylem and phloem are present at same radius. They are present at same radius. What? Xylem and phloem. And when you talk about the collateral, so in the collateral one, xylem is present towards inside and phloem outside. That's how the things are. So see this, xylem and phloem. Xylem is phloem. So xylem is towards inner side, and you know that this type here you have end dark arrangement. End dark arrangement. So protoxylem is towards the pith. Okay, so this is xylem, this is phloem. This is xylem, this is phloem. Take, this is xylem, this is phloem. So see this, right? Right, conjoint collateral, right? So phloem, okay. Yes, bache. So phloem and xylem is there. Phloem and xylem is there. And as I said, the xylem is towards the phloem inner side, and phloem is towards the outer side. Xylem is towards the inner side, and phloem is towards the outer side. Now here, one more thing that you have to remember is that when it is conjoined, question can come from this part, please pay attention. Conjoint collateral. So it can be open vascular bundle, it can be closed vascular bundle. So how can you remember this? Closed. I remember it like this. It is closed for secondary growth. It will not allow the secondary growth. And how is it possible? Because it doesn't have cambium. No cambium is there. No cambium is there. Now here it is open. Cambium is present. Cambium is present. So how can we make it like this? See this? So this is xylem, this is xylem, this is phloem. In between this white colored is cambium. So if cambium is present, secondary growth is possible, right? So it is open for secondary growth. That is how I remember. So if it is open for secondary growth, what is required? Cambium is required. For secondary growth, what is required? Cambium is required. So that's how I write it, okay? And this one is closed for secondary growth. Take. Right, so bacha, bicollateral is also something, right, and it is the characteristic of Cucurbitaceae family. I'm writing this, the spellings here, Cucurbitaceae, A-C-E-A-E. I'll share this PDF in our official Telegram group. You can even join my Telegram group, Love Biology by Ambika. Take, so xylem is there. So bicollateral mein, you will have xylem here, and both the sides will have phloem and phloem like this. It is the bicollateral. Concentric vascular bundles are also there. If xylem is in the surrounding, phloem is in the center, amphivessel. If xylem is in the center, phloem is surrounding, amphicribral. So in our syllabus, that's something we have to remember that I have already told you. Okay. So here you can see the NCERT, radial vascular bundle in the case of roots. This is the closed vascular bundle. Where will we see this? In the monocots. Because monocots, they don't show the secondary growth. Monocots like grasses, take a gymnosperms, even your dicot stem, dicot root, they show the secondary growth. But when it comes to the conjoint or collateral type of vascular bundle, they are in the stem only. This is the open type. This is the open type. Is that clear? Is that clear? Is that clear? Chaliye. So now quickly revise the sections. Now in the sections, see. Now in the sections, how to complete that? First of all, it will be epiblemma and also known as pyliferous layer. And of course, why? Because no cuticle is there. And when it comes to the appendages, we know that root here will arise that will help in water absorption. Now here in the epidermis, TK cortex is there. And I told you endodermis is what? Endodermis is the innermost layer. Now regarding endodermis here, we need to remember something, that in the endodermis, the radial walls, they are having what? They are having the Casparian thickenings, Casparian strips. Casparian strips. And these Casparian strips are what? They are water impermeable, right? They are water impermeable. Now, transport in plant is deleted from our syllabus. Otherwise, we used to discuss apoplast, symplast. So, water impermeable, kyun? Because suberin etc., they are deposited. Now, when you look at the dicot root, here 1, 2, 3, 4 xylem, here you have the phloem as well. So here it will be diarch to tetrarch or hexarch. Matlab it will not be more than vascular bundles, right? They are not more than 6. Take. So how to remember that? Baki ye dekho quickly. Right, just keep the diagrams in your mind. Here the revision is very easy. Now we will take 15 minutes to finish these sections. Right, we will take, yes, 15 minutes. We have to speed up now. You told me next chapter, plant growth and development, we will go for 1 hour, right, maximum. Right, because I have mentioned all the points. I'll share the PDF. There you have to revise the things only. We don't have much there. Then photosynthesis and respiration is going to take the time, right? If you are comfortable, we can directly start with the respiration because we have to focus on certain tricks only there. Okay, and even for the photosynthesis and respiration, I'm going to put the short revision sessions as well. Okay, I am going to put the short revision sessions as well. Just here. And again, I'm telling you, do check this. Do check this 20 days revision plan as well. Baki. So these are the short video sessions, right? I'm not getting your sufficient love. So that's why I didn't post the next one. So if you want, right, if you want the sessions like this, do let me know. So I have planned the biotech short sessions, the photosynthesis short sessions, the respiration short sessions, and the new NCRT the new angiospermic families as well. And moreover, right, moreover, that is not the only thing that we have. Cockroach and frog, I know I have to post it in the recorded format. So of course the NEET is there, so we'll keep posting. Don't worry about that. Okay, don't worry about that. So make sure you mention it in the comment section that yes, we want such kind of session. So I'll put the another one. And do not forget to check this revision plan as well, people. Okay, chaliye. So dicot root. So as I said, epiblemma. Okay, unicellular root here, done, simple part. Now cortex will have several thin layered parenchymatous cells, right? And here the cortex is having the intercellular spaces. So now when you're talking about the endodermis, it is single layer barrel shaped cells there. What kind of cells are they? They are the barrel shaped cells. They are the barrel shaped cells, and in the endodermis you will not see any. Right, you are not going to see any kind of intercellular spaces, and the tangential as well as radial walls will have Casparian strips. Okay, right, Casparian strips, because suberin is deposited there. Already discussed it with you. So after this next to this endodermis, you will have pericycle. You will have pericycle like this. Now, bache, remember one thing. You have the xylem also, na? Exact arrangement, xylem also here like this. So the pericycle part, right, like the part where you know this xylem is facing the pericycle cells, these cells are also known as passage cells. These cells are also known as passage cells. So next to endodermis lies a few layers of thick walled parenchymatous cells. They are the pericycle. This one is pericycle, this one is endodermis. Remember the sequence: epidermis, hypodermis, cortex. Innermost layer of cortex is endodermis, then pericycle is there. Okay, so the sequence here is important. Okay, so initiation of lateral roots and vascular cambium during secondary growth take place in these cells. So basically they are saying that this pericycle is responsible for secondary growth, or you can say that secondary root. Just we make the primary root like this, then secondary roots are there. So they will arise from the pericycle. Okay, they will arise from the pericycle. Okay, so lateral roots arise from pericycle, and that's why the lateral roots, their development is considered as endogenous. Right, because they are arising from inside, from the cortex part. Okay, and along with that, bache, I already told you, right, for the secondary growth also, this is the one that will form secondary meristem. So this line is important, even if the secondary growth is deleted, but this part is important. Baki pith is small and inconspicuous. So in between xylem and phloem, conjunctive tissue is there. Okay, so that will also help in the secondary growth. So there are 2 to 4 xylem and phloem. So diarch to tetrarch it is, right? So cambium ring will develop between the xylem and phloem when there is the secondary growth, because it is the dicot root. So students, in dicot root, in dicot stem, secondary growth is there. Okay, so I will show you the structure. So see this. So here you have the pericycle cells. Matlab this one is, let's suppose this one is next to the endodermis is your pericycle. So the cells of pericycle which are facing the xylem, the cells of pericycle which are, the cells of xylem which are facing the pericycle cells, these pericycle cells will known as passage cells. And in between this xylem and phloem, right? In between this xylem and phloem, see this. This one is what? This one is the conjunctive tissue. Right? This one is what? This one is the conjunctive tissue. What's that? This one is the conjunctive tissue. Is that clear? Is that clear? So that is something you have to keep in your mind. And when it is the monocot, you remember na, phloem parenchyma, it is absent in the monocot, most of the monocot, remember this. So monocots may, it will be polyarch. There are so many. It is not diarch to tetrarch, there are so many, polyarch. So that is the one way to differentiate it immediately. Okay, so here there will be no secondary growth. So you can see epidermis, cortex, endodermis, pericycle, vascular bundle, pith, everything is there, right? So here in the monocots, you have more than 6 xylem bundles, right? In the monocot root. Pith is large and they do not show any secondary growth. Already discussed. I told you, the sections are very easy. Now let's come to the dicot stem. Look at it. Dicot stem. Look at it. Here cortex is well differentiated. Cortex is well differentiated. Right. Here you will see hypodermis, ground tissue is well differentiated. You will see hypodermis, you will see cortex, endodermis, pith, medullary ray, pericycle. So now when you look at this part, look at the pith part. It's conspicuous. Now this is your dicot stem which will undergo secondary growth. Please have a look. Here I'll mention one more thing. These are the vascular bundles that you have. Can we say that here the vascular bundles are present in the form of a ring? Can we say that here the vascular bundles are present in the form of a ring? Like when you look at this part, so here it is the epidermis that you have, number one. Below epidermis, it is collenchyma. So here we say that hypodermis is collenchymatous. What do we say here? That the hypodermis is collenchymatous. Hypodermis is collenchymatous. Next to that, cortex will have parenchyma. Endodermis is there. Here, endodermis is known as starch sheath. What do we call it? We call it as starch sheath. Right? Right? No Casparian strips. Then pericycle. Pericycle here is sclerenchymatous. It is in semilunar patches. Then phloem, cambium, then metaxylem and protoxylem, then pith is there. That's what we have. Okay, that's what we have. Ye dekho. Collenchyma, these are the parenchymatous cells. You have the endodermis, which is also known as starch sheath. Then this pericycle here is sclerenchymatous. It is in the form of semilunar patches. So here you have your phloem, cambium, then metaxylem, protoxylem. So this arrangement means endarch arrangement. So just have a look. Just have a look, please. Okay, so this is the diagram. Okay, so now when you look at this rings, right, they are the rings, and I told you here secondary growth will be there, girth will increase. So students, in between this vascular bundle, you have the cambium there. You have the cambium there. What do you have? You have the cambium there. This cambium which is present in between, it is also known as intra, mane within, intrafascicular cambium. Do you know that? It is also known as intrafascicular cambium. It is also known as intrafascicular cambium. What do we call it, people? We call it as intrafascicular cambium. Is that clear? Is that clear? And it will not form due to dedifferentiation. It is there from the starting only. So that is why it is also known as primary lateral meristem. Matlab it is lateral. Lateral in position it is. It will help in that secondary growth. But it is primary in origin. Matlab it formed during the primary growth only. Now in between these vascular bundles, you have the medullary rays, right? These are the medullary rays that we have in between. So the cells here will become, right, de-differentiated, and they will start the division again. And that is how this ring will form here. That is how this interfascicular cambium will form here, right? That is something we study in the case of secondary growth, right? Right. So that's how you people can make it. So this pinky, it is your interfascicular cambium, and it forms due to dedifferentiation of the primary permanent tissue, right? It is formed due to dedifferentiation of the primary permanent tissue, right? It is due to dedifferentiation of the primary permanent tissue. So it is the interfascicular cambium, and it is also having the intra. So this is intra, this one is inter. This is the complete cambium ring. This is the complete cambium ring. Are you getting my point? This is the complete cambium ring. Okay. So here, right? So you people can see this here. So it is covered with a thin layer of cuticle. It may have trichome and fused stomata. The cells arranged in multiple layers between epidermis and pericycle, and it consists the cortex. So here you have hypodermis that is made up of collenchyma. Then you have the cortical layers having parenchymatous cells having intercellular spaces. Then you have the endodermis. So endodermis is rich in starch grain. So I told you it is the starch sheath. Here you will not talk about that Casparian strips. Then pericycle is also there, students. Pericycle here is what? Pericycle here is what? This is present on the inner side of the endodermis and above the phloem. And it is in semilunar patches, and it is sclerenchymatous. Remember it like this. That's why I specified that. Okay, that is why I specified that. Okay, so radially placed parenchymatous cells are there in between the vascular bundles. So there is a ring, right? So each vascular bundle is conjoined, open, and within dark protoxylem. Okay, so that is what we have here. Okay, that is what we have here. Okay, so Baki, here you have the monocot stem, and here you will see that ground tissue is not differentiated. If this is the monocot stem, vascular bundles are just randomly scattered here. They are randomly scattered here. They are randomly scattered here. Are you getting my point? Are you getting my point? So do focus on this thing, right? Phloem parenchyma is absent here. The vascular bundle in the center, they are large. The one that are on periphery, they are small. The one that are on periphery, that they are small. That's it. So you can compare them. You can use these notes. Baki beta, this is your dorsiventral, right? Dicot leaf. You know that here two sides are different, right, bache? Right. So it is the upper epidermis. It is the upper epidermis, so upper side will have palisade parenchyma. So here you will use the word mesophyll. This side is spongy parenchyma. Okay? So upper epidermis is there. This is the lower epidermis, right? Bundle sheath cells are surrounding the xylem tissue. So this is not right. So this is what we have here. So cuticle, upper epidermis, palisade. That's it. So here in dicots, it is dorsiventral, and in the monocots, both the sides are equifacial or isobilateral leaf is there. So just compare it, revise it, and dark arrangement is there, right? So that's it, that's all. So this is about the leaf revision. That's it. So now let's start the plant growth and development. After finishing plant growth and development, bache, I will give you the break. Plant growth and development is just for 1 hour. I have made the short notes as well. We'll revise it very quickly. So are you ready for that? So do you need a break? What will you do with the break? Break will break that flow. है ना? Break is going to break the flow. After this chapter, let's have a break. After this chapter, let's have a break. Then after that break, let's start photosynthesis. So basically, see, in like, chalo, let's consider 5 hours. In 5 hours, we have completed 2 chapters. And in that 5 hours, 40 minutes ka break was there. Right? 40 minutes ka break was there. So we have completed 2 chapters. The speed is good. If you think, ki, nahi madam, finish it in 1 hour. Not possible, beta. We have to discuss few things, na. ठीक. So plant growth and development. Plant growth and development. So growth, you know that it is the fundamental and conspicuous, right, characteristic of the living beings. Remember, growth is not a defining feature. It is the characteristic feature, right? When we are not talking about the internal growth in general, when we are saying growth, it is not a defining feature. It is a characteristic feature. It is an irreversible permanent increase in size of organs or its part or even of an individual cell, right, of the organ or the parts of a living organism. Okay, so growth base due to the metabolism, and here the anabolism, that is build up, should be more, right? Build up should be more. Okay, so anabolism means the reaction in which there is a build up, energy stored there. Catabolism, where breakdown is there, right? So expansion of leaf is also the example of growth here, right? So this is simple. Hypocotyle, epicotyle, the question they don't ask from this part. So growth I told you. So next thing that you have to remember is, in the case of plant, the growth is indeterminate. Why, beta? Just now we discussed about the meristematic tissue, right? We have to apply the same knowledge here. That is why I'm telling you that let's finish it in one go. What say people? Let's finish it in one go. What say people? Right? Because these 2 chapters are linked. So let's finish it in one go and then let's proceed. What say? Okay. Okay. Right. So, because of meristematic tissue, the growth is unlimited. It is throughout the life. It is throughout the life. So, product will lose the capacity to divide and such cells will make up the plant body. So, simple. Look at this diagram. The tip is showing root apical meristem. Here, shoot apical meristem, root apical meristem. Vascular cambium is the example of your cylindrical meristem. So, it is on the lateral side and it is helping in increasing the girth. Just now, we have discussed that. So, because there is meristematic tissue, the plants will grow continuously, right? New cells being added continuously. That's why it is indeterminate and open form of growth. What type of growth it is? It is the open form of growth. It is indeterminate one. It is the open growth of open form of growth. So you know that when there is the apical meristem, it is helping in primary growth. It is helping in elongation of the plant. Okay. When you people talk about the lateral meristem, that is vascular and cork cambium beta, that help in increasing the girth. Already discussed. Okay. So growth here is indeterminate. That is what I told you. Okay. So right. So meristem have the capacity to divide and self-perpetuate. Right. Self-perpetuate. Okay, beta. Right, bache? So they'll keep on adding the cell. So that's why the growth is open type. So that is what we have discussed. Now, next part that we have to discuss is the, right, growth is measurable. Yes, growth is measurable here. Again, let's have a quick revision. Is growth measurable here? Sorry. Arey. So is growth measurable here? Yes, bache, at cellular level, because of the increase in protoplasm. Because of the increase in protoplasm. Right. So we cannot measure it directly. We cannot measure it directly, but yes, we can focus on the number. We can focus on the cell size. We can focus on the length. We can focus on the surface area and fresh weight, dry weight, and volume. So when it is the number, so root apical in the case of maize, right? In the case of maize, right, root apical meristem can produce more than 17,500 new cells per hour. right? So in watermelon, cell size can increase up to 3,50,000 times. Then growth of the pollen tube, it is showing the increase in the length. Right, it is showing the increase in the length. Then surface area in the case of leaf, and dry weight, fresh weight, and volume is there. That is how we can measure the growth. So this information is from the NCERT only. So do mark it. Okay, do mark it. Then now we have the regions. Even in the case of root, if you people remember, in the morphology, that is something we have to discuss now. I'm discussing it here only. In morphology part, that is something that we discuss now. Regions of root. So here in the case of root, this is the apex, this is the tip, and this is the base. So this is a tip, and this tip is having root cap, because roots, they need to, penetrate deeply into the soil, right? So the root cap, it is providing the protection. So here you will have the meristematic zone. I already told you when we started the chapter that what are the characteristic feature of the meristematic cell. So this is the meristematic zone. Next to it, beta, here you will see elongation zone. So when you talk about the elongation zone, here they help in increase in length. They help in increase in length. And let me tell you, right, let me tell you that here you are going to see, right, they help in elongation zone, this help in increase in length, and here there will be increased vacuolation. Here there will be increased vacuolation. So it helps in increasing the length of the plant. So here vacuoles are more. Okay, these are the regions of the root. Root cap, then meristematic zone, या meristematic region, then elongation zone, elongation region, where the cells are elongating. The expansion of cells is there. So of course, that's why the plant is increasing its length. That is why plant is increasing its length. And here you will see vacuolation will increase. So already in the anatomy part, we have discussed this topic. So that is why I'm not touching it here. Okay, that these are the properties, this and that. Already we have covered it. So why to revise or why to repeat it again? है ना? Why to repeat it again? Yes or no? Yes or no? So simply that's why I'm mentioning the important points here only. Okay? Okay? Done, beta. So here in the phase, it is the phase of elongation, beta. Cell enlargement is there, right? Cell enlargement is there, yes or no? Cell enlargement is there, new cell wall. So increased vacuolation, cell enlargement, new cell wall formation, या material deposition is there. So secondary cell walls will form. So yes, the characters are important here. Okay, new cell wall deposition is there. Okay, so deposition should be the word here. You are talking about the secondary cell wall, which will show accretion, accumulation of material, yeah, deposition of material. ठीक? Okay, right? So basically here accretion is also there, and beta here there is the enlargement of the cells. Okay, so new material will also deposited. New material will also deposited. ठीक? Done, beta. And then in the cell maturation, so this is the last one. Maturation zone. It is the maturation zone, and here you will see you will see root here in the maturation zone only. Where will you see the root here? You will see it in the maturation zone only. And here, bache, basically this is the maturation zone, means cells are differentiated. Cells are differentiated. They have a permanent function now, right? What do they have? They have a permanent function now. Okay, okay, just have a look here. Have a look here. So you people can see this. So there are meristematic, elongation, and maturation zone. Okay, so these cells are constantly dividing both at the root apex and shoot apex. And yes, they can ask you the sequence from base to apex. What is the sequence of the zones? So near to the base you have meristematic zone, then elongation zone, then maturation zone. Firstly maturation zone near to the base, then elongation zone, and then there is the meristematic zone. Then there is the meristematic zone. So it is from base to apex. So if somebody will ask you, somebody can NTAs, if NTA will ask you the sequence from apex to base, Apex is your tip, na? This is your base, huh? Then it will be meristematic zone, elongation zone, then it will be maturation zone. So yes, from morphology, we have seen the question like this. Done? Done? Okay, so the cells in this region are rich in protoplasm, large, conspicuous. You are talking about the meristematic zone. They are of primary in nature. Already I have mentioned in the anatomy part. Okay, they are thin and cellulosic with abundant. Abundant means many plasmodesmatal connection. What is this plasmodesmatal connection? They are the kind of gap junctions in the plant. They are basically, these are the kind of junctions, continuous cytoplasmic junction in between the plant cells. They are traversing all that, layers, middle lamella as well. So it is the continuous cytoplasmic connection in between the plant cell. That is the plasmodesmatal connection. Like in the animals, we talk about the gap junctions. Here we are talking about the plasmodesmatal. Right, here we know about the plasmodesmatal connection. So P for plant, P for plasmodesmatal. That's how you people can remember. So cells which are just away from the tip to the meristematic zone, it represents the phase of elongation. So increased evacuation, cell enlargement, new cell wall deposition are the characteristic of cell. Now further away from the apex, more proximal to the phase of elongation, it is the portion of the axis which is undergoing the phase of maturation. ठीक? Right? So here the cell will have maximum size in terms of wall thickening and protoplasmic modification. That's it. So see in the notes, you will get these points. Now come to the growth rates. So beta, growth rates, again, it is also a very simple topic. It is also a very simple topic. The increased growth per unit time is the growth rate. What is it? It is the increased growth per unit time. What is it? It is the increased growth per unit time. Hanji, what is the word here? What is the word here? Increased growth per unit time. Yes? Done? Done. So there is one question a student is asking, that what is the difference in the protoplasm and protoplast? Beta, it's very simple. Protoplasm is actually the living matter. When you are talking about the protoplasm, you are talking about the living matter of a cell. जैसे कि genetic material plus cytoplasm. Genetic material plus cytoplasm, protoplasm. Then protoplast. When you are saying protoplast, so what exactly this protoplast is? From a cell, right? When cell wall is removed, right? From a cell, when cell wall is removed, from a cell, when cell wall is removed, it is removed, cell wall is removed, then it is a kind of cell. We are calling it as protoplast. Its cell wall is removed. Its cell wall is removed. ठीक है बेटा, that is the difference. बाकी growth rate, it is the increased rate growth per unit time. Increased growth per unit time. Per unit time. Right? जैसे if I'm saying that in 1 hour, how many physics questions are you solving? Right? Right. So here, if I'm talking about the growth, I have specified that in per unit time, right? What is that increase? Okay. So mathematically, if we have to express it, arithmetic growth and geometric is there. Beta, when it is arithmetic, it's like that 1, 2, 3, 4, 5, 6 types. When it is geometric, it is like that 2, 4, 8, 16 like this. Basically, in arithmetic growth, what's going to happen? This is your one cell. This cell will divide into 2. Out of that, one cell is like, ki I will divide further. And another cell is like, that I am not interested in dividing further. I just want to get matured. Okay, fine. Now this cell will divide again. It will again form 2 types of cell. One cell is like, I will just grow, I'll just mature, and another cell is like, okay, I'll divide. So here you got 1 cell, 2 cell, 3 cells, like this. But in geometric growth, cell, divide 2 daughter cells, they'll further divide 4 daughter cells, and so on. Okay, and so on. So that is what arithmetic growth is, and this is the picture that we have. This is the image that we have in NCERT. Okay, so here in arithmetic growth following mitotic cell division, only one daughter cell will continue to divide and other will differentiate and mature. So it's DM. Firstly differentiation, then maturation. So a root elongating at a constant rate is an example. A root elongating at a constant rate is an example. It is an example. Done? Any doubt? Sure? Okay. So, on plotting the length of the organ against time, a linear curve is obtained in this case. This is important. So, mathematically, LT is equals to L naught plus RT. So, length at time T, length at time 0. R is growth rate to the elongation per unit time. What is this R here? It is the growth rate to the elongation per unit time. It is the growth rate to the elongation per unit time, right? Yes? Okay? It is the elongation or the growth per unit time. That is the arithmetic growth that we have. Any doubt here? Any doubt here? Right? The curve is linear here. Okay, so R is equals to growth rate to the elongation per unit time. Fine, to the elongation per unit time. So this is it. So x-axis will have time, y-axis will show the height of the plant. Now, next point, geometric growth, geometric phase. So you know that here it is going to be sigmoidal. It is going to be sigmoidal, S-shaped. So x-axis is there, y-axis is there. So initially beta lag phase, initial growth is slow. Then log or exponential phase. This one, this is initial one. Let's suppose this lag phase, log phase, because resources are limited here. Then it will be stationary. Then it will be stationary. Lag, log, stationary. Lag, log, stationary. So what is going to happen? Both the progeny cells are undergoing mitotic cell division. So initially it is slow. So then one cell is forming 2, 2 is forming 4, 4 is forming 8. Then comes the stationary phase. The growth will slow down due to limited nutrient supply. Due to limited nutrient supply. Is that clear? So it is sigmoid or S curve. Sigmoid or S curve. So x-axis is showing us the time, right? X-axis is showing us the time and y-axis is showing us the size and weight of the organ. The size and weight of the organ, right, bache? So lag phase is there, log phase is there, and stationary phase is there. Okay, okay. So W1 is equals to W naught ERT. So in the paper they can ask you what this E is, yeah, what this R is. So it's the final size. Here you are talking about the weight, height, number, etc. W naught initial size beta. When at the starting, what is the size? After it is the final size after the growth. R is the growth rate. And here when you are talking about the R, it is the growth rate to the elongation per unit time. So do not confuse there, right? Do not get confused there. So then T is time, E is the base of natural logarithm. In the ecology also we discuss it now. Sigmoid growth, Verhulst-Pearl logistic growth curve model. So R is relative growth. So basically we have two growth rates. One is absolute, another is relative. One is absolute, another is relative. One is absolute, another is relative. So relative growth rate mein you are relative, relation, right? You are comparing two things. It is measure of the ability of the plant to produce new plant material. It is also the efficiency index. So relative growth rate mein we are checking that what is the ability of that plant, right? Right? How much growth it has done, Okay, so it is the efficiency index. So final size always depends upon the initial, right? What should be the final result? It depends upon this. जैसे your preparation, मतलब how well you have when you talk about your exam or your final result, it depends upon your preparation na. So the whatever you are going to perform in that exam, it depends upon your preparation. Same is the case here. So if we need to check W1, it depends upon the W0, right? W0. So here relative growth rate is considered. The R is the relative growth rate, and it is the measure of ability of plant to produce new plant material. So it is the ability, measure of ability of the plant to produce new plant material. Okay beta, to produce new plant material, and this is the efficiency index. So hence the final size of W1 depends upon the initial size, right? So here you have the growth comparison. Two leaves are there. So in the case of absolute growth rate, so it is that, it is written like that, na, measurement and comparison of total growth per unit time. So what is that total growth per unit time, right? Absolute measurement and comparison of total growth per unit time, measurement and comparison of total growth per unit time. So you can even write down total growth over unit time. That is how you can write it. Total growth to the unit time. Total growth to the unit time it is. What is it beta? Total growth to the unit time, right? Right? What is it? Total growth to the unit time. Absolute growth rate. Then comes the relative, right? So here the growth of the given system per unit time expressed on a common basis, per unit initial matter. So how can you write it? Growth in given time period to the measurement at this time. Growth at given time period to the measurement at the start of that growth. So basically relative it is na, you are comparing what was there in the starting and what somebody has done in the given time. So it is actually the absolute growth, and then you are also considering the initial one. That is why we are saying, when we were discussing the geometric growth rate, right, when we were discussing that part also, right, I told you na that initial will depends, the final will depends upon the initial. So that is how you can compare it, because here also we were talking about the relative growth rate, right? Relative growth rate. So efficiency index we were considering. So basically it depends upon the, right, matlab jaise let's suppose today somebody is saying ki after attending the lecture, right, now my marks, they are jumped from 200 to 300. So it depends now that initially, right, initially they were at 200. Maybe there is somebody who will be at 100 marks. After this lecture your marks will jump to 200. So that is something we have to consider, that from where somebody has started. So here in this particular case we are saying growth at given time period to the measurement at the start. At the start of what? Of course at the start of time period. So this is the relative one, and this is the absolute one. This is the absolute one. So here if I talk about the absolute growth, total growth in unit time, in both the leaves it is 5 centimeters square. Here also it is 5 centimeters square. So now if I have to consider the relative growth rate, relative, so we have to consider the starting also. So absolute growth rate is 5, starting mein it was 5. We have to multiply it with what? Then, because we have to check this na, 100, right? So it is the relative growth rate. So growth of given system per unit time to the initial parameter, and yes, of course you have to multiply it with 100. So you are going to multiply it with 100. So here it is 100% for leaf A. Now in the case of leaf B, right, so growth was 5 but initially it was 50. So 5 into 50 multiplied by 100, so it is going to be 10%. So of course this leaf is growing faster. Okay, this leaf is growing faster. Is that clear? Is that clear? The question can come from this part, right? From growth rate thing the question can come, so please tell me. From growth rate thing the question can come, so please tell me. If there is any doubt, just have a look. Just have a look. Done? Any doubt? Chaliye, let's move to the next part. So quantitative growth ka comparison, done and dusted. Condition for growth, very easy beta, that water, oxygen, nutrient, temperature, light and gravity is required. Now when it comes to the concept of differentiation, de-differentiation and redifferentiation, that's why after anatomy I always start this chapter, because this is what we have discussed. So when the cells derived from the meristem, right, they differentiate and they mature to perform specific function, differentiation. So during differentiation, you know that structural changes will occur in their cell wall, in their protoplasm, right, and it depends upon their position as well, right. So just say here they are giving the example that if we need to form the tertiary element, tracheids and vessels, they need to lose their protoplasm, they need to develop strong elastic lignocellulosic secondary wall so that they can carry out the water. So matlab, this is something that we have, right, as per their function. Then let's suppose the differentiated cells, then again they have lost the capacity, to divide, and when they regain that capacity, de-differentiation, right. So interfascicular cambium, cork cambium, then de-differentiation, right. So secondary meristem, we will get secondary meristem after de-differentiation. So this is something that we have already discussed, this is something that we have already revised, so I am not going to discuss it again, right. But yeah, this table is important, okay, this table is important. So here you can simply see that development is a term that include all the changes, development is a term that include all the changes, right, that include all the changes that an organism can go through during its life cycle, from germination of the seed to senescence, from germination of the seed to senescence. So it is the sum of growth and differentiation, right. So what is development? Sum of growth and differentiation. So it includes all the changes that an organism will go through, right, that an organism will go through, right, during its life cycle, from germination of the seed to senescence. So meristematic cell, when I'm saying plasmatic growth, means its protoplasmic growth is there. Matlab, when protoplasm is growing, the cell will also expand. It is understandable. There is no need to cram this portion. There is no need to cram this portion. It is understandable. So meristematic cell, it is showing that plasmatic growth, right, that protoplasm, that cytoplasmic growth, which will result in expansion. Of course, elongation is there. Then differentiation or maturation. Finally, the mature cell, it will undergo senescence. The death will be there. So plasmatic growth, and again the cell division will occur. ठीक बेटा, cell division will occur. बाकी plasticity. So plasticity is simple. It is the ability. It is the ability of plants to follow different pathway in response to environment or phases of life. मतलब how a plant will react to different phases of life or to the environmental changes, that will come under the plasticity. One example out of that is heterophily. जैसे cotton में, coriander में in different life phases it is having different leaf shape. Okay, that is the example. जैसे leaves of the juvenile plant are different in shape from those in the mature plant. Now in the buttercup, which is also known as ranunculus, right, so leaf produced in air, matlab which are not under water, right, they will have complete lamina, and the one which are under water, right, they are dissected one. What are they? They are dissected one. They are dissected one, right bache, they are dissected one. Is that clear? So again the example here is important. Baki beta, plant growth regulator. Okay, scientists, their discoveries, too much information is there, but again I will just conclude it with the most important points, otherwise we will not be able to finish the complete structure. So if we really want to finish it, we have to increase our pace, right? So see, plant growth regulators they are. So here we have inhibitors. So firstly, we have the growth regulators. They are going to regulate the growth. So they are the promoters as well as the inhibitors. They are the promoters as well as the inhibitors. The one that will promote auxin, gibberellin, cytokinin. They are the one that will promote the growth. Inhibitor mein ethylene is there and abscisic acid is there. So ethylene can be in both the category, right? It can be the promoter as well, but mainly its functions are just like inhibitor. Okay, okay, right bache, right, right, yes. So small simple molecule of diverse, so indol compounds, indol 3 acetic acid will give you auxin, adenine derivatives, right? N6 it is. It is N6 furfuryl aminopurine kinetin. Derivatives of carotenoid is your abscisic acid. Terpenes are gibberellic acid. Gas is methylene. Again, beta, it is important, but it is totally memory based. Okay, it is memory based. It is something that you have to revise. Okay, it is something that you have to revise. So just have a look, right? Just note down quick. You write it down, please, quickly. Do it quickly. Yes, everybody, do it. Done? Just a minute. Okay, chalo, let's go. So this part is done and dusted. Now the next part. So auxin, chemical composition and other names. Auxin, chemical composition and another names. So people, you can see this part very clearly, right? These PDF notes will be provided to you in our Telegram group, and you people can just have quickly revise it. Okay, so from this part, these kind of questions are important. Just say, see, IAA, indole 3-acetic acid, indole butyric acid, they are the natural, right? What are they? They are the natural auxins. 2,4-D, dichlorophenoxyacetic acid. This is the, right, even 2,4-T, trichlorophenoliacetic acid. They are the synthetic auxins, and this one is also the herbicide. Yeah, you can say that VD side. Okay, it will destroy the dicot weeds. VD side it is. Okay, then when it comes to cytokinin, it is the adenine derivative N6 purpuryl aminopurine. So zeatin is the natural one. So from the word zeatin, just remember Z-mase, right? From the word zeatin, just remember Z-mase. Then kinetin, not found naturally in plants. Gibberellin, terpenes, they are GA1, GA3, there are so many. Then ethylene gas, ethylene, that is used in the aqueous solution. Then abscisic acid, the stress hormone in the case of plants. So now when we talk about the discoveries, okay, so again here we have the, quickly we can revise it. So you can see auxin, FW went, right? The first, it was isolated from human urine, right? Who isolated it? Went, right? So when it comes to the discovery, so isolation is from went from the human urine. So Darwin and Francis Darwin and Charles Darwin. So in the case of canary grass, just pick up the main points here. Right now, this is what, right? This is what you need to focus. This is what you need to revise. Trust me on that. Then cytokinin is there. So kinetin from the autoclaved herring sperm. Right. So what is the word here? It is the kinetin from the autoclaved herring sperm. Right. Herring sperm, it is a fish sperm. Okay. DNA, from its DNA it was isolated. And zeatin, it is from the extract of vascular tissue, yeast extract, coconut milk and the corn kernels. Right. Bache, so this is something that you have to revise very, very, very quickly. And if you have completed it, right, or if you have revised it once, then go for it. Otherwise, do not touch it. Literally, I'm telling you, do not touch it. Focus on the function of the hormones. Okay. Then focus on the function of the hormones. Okay. Okay. So that's something you have to keep in your mind. Understood? Understood? Then after that, so who discovered it? And yes, whenever you talk about the discoveries, just here it is like right on the accidental hormones. So we use the word serendipity. What is the word that we use here? We use the word serendipity here. What is the word that we use here? We use the word serendipity here. Serendipity means here they all are basically the by chance discovery. What's that? It is basically the by chance discovery. Okay. It is a by chance discovery. Then Skoog and co-workers are there. So internodal segments of tobacco stem. The calyx proliferated only when the auxin was supplemented with with cytokinin. Again, accidental discovery it was. Then gibberellin, sterile filtrate of the fungus Gibberella fusiculae. Remember? So Kurosawa, fungal pathogen Gibberella fusiculae, it was causing Becane disease, right? The foolish seedling, that's the point. Then ethylene is the word, volatile substance released from ripened roots. Okay, so HH cousins, right? They discovered it, and then abscisic acid, stress hormone. So it doesn't mean that it is going to give stress to the plant. It simply means that when you people talk about the stress hormone, it will be released when the plant is under the stressful condition. Let's suppose there is a water stress, right? And water scarcity is there. So abscisic hormone will release. Abscisic hormone will make sure that transpiration is not there, right? Why? Because it has to make sure that plant survive in such kind of conditions. Okay. Okay, so when it comes to the discovery in this way, very quickly we have to revise. Now, auxin. Bache, it shows the polar transport. I'm giving you the main points right now. Polar transport. And when it is auxin, A for apical dominance. Right, that is the main function here. A for apical dominance. So, beta, in this case, apical bud dominates the growth of lateral bud. Right, matlab when the apical bud is present, the lateral buds will not grow. So of course, if we will decapitate it, then the lateral buds will grow. So this is something which is very useful in the hedge making. Okay, for tea plantation and hedge making, this is useful. So in cell division and cell enlargement also it is helping out. So mainly when cell elongation is there, so it helps in cell division. So auxin also controls the xylem differentiation, right? It is also important. Now, rooting on the stem cuttings, right? In tissue culture, when it comes to the rooting, when it comes to the growth of the root, when it comes to the growth of the root, so then in that case also auxin is used. Okay, then abscission, right? You know na abscission, जैसे IAA, indole 3 acetic acid, naphthalene acetic acid, they prevent premature abscission. Like let's suppose there is a plant, so it should not fall down from the tree prematurely, right? So premature abscission of plant organ will be prevented by it, right? But yeah, it will promote that in the case of mature leaves. Ki mature, now mature leaves and fruits, you are mature, you fall down, but not for the young organs. Okay, then flower initiation, that is also somewhere there in the ethylene, so do related. So when it is the flower initiation, when it is what, when it is the flower initiation, so auxin promote flowering, auxin promote flowering in pineapple and lychee plants, auxin promote flowering in pineapple and lychee plants. Okay, then parthenocarpy is there. Then parthenocarpy is there. So seedless fruits can be produced by the spray of indole 3 acetic acid. So seedless fruits, okay, parthenocarpy, right? Without fertilization, fruit will form. So if without fertilization, fruit will form, then it will what? It is the seedless fruit. It is the seedless fruit. It is the seedless fruit. Okay, then select. So 2,4-D, just now I told you it is a VD site. It will kill the dicot weeds. That is also PYQ. So these are the things. So apical dominance, rooting, do not forget about this part. Now let's move to the next part, and that comes the gibberellin. G for gibberellin, G for growth hormone. Even its discovery is also serendipity. So whenever you talk about the plant growth regulators, their discovery is serendipity. Their discovery is a by chance discovery. What kind of discovery it is, bache? What kind of discovery it is? It is the by chance discovery. Okay, what kind of discovery it is? It is the by chance discovery. Clear? Clear? Are you sure? Are you sure? So in the case of gibberellin, right? Remember bolting. This is the word that you have to remember in the rosette plant. So stem या फिर internode elongation is the main function here. So I have started it with the main functions only. So internode elongation, right? What is the main function here? Internode elongation is the main function here. So gibberellin induce internode elongation just before the flowering in rosette plant. Rosette plant where, you know, leaf is there, internode is very short, just like your beet, just like your cabbage. So this is the bolting effect. Important phenomena it is. Okay, then after that there is the elongation of genetically dwarf plant, right? So when gibberellin is applied, these plants, they'll increase their height. Then seed germination mein, right? So remember, your gibberellin and ABA, right? They are antagonist, opposite to each other, right? Gibberellin and ABA. So seed germination mein gibberellin will induce the synthesis of hydrolysing enzyme and it helps in seed germination, and ABA will promote the seed dormancy. So that is why your gibberellin and abscisic acid, they are antagonist. They are antagonist. Their function is opposite to each other, right? They are antagonist. Is that clear? So gibberellin induces the synthesis of hydrolyzing enzymes like alpha amylase, lipase, protease in seeds, and these enzymes help in seed germination. So they help in increase the length of grape stalk as well as elongate and improve the apple fruits. Okay, so GA, gibberellic acid, it also helps in the malting. Okay, right? So increase in the height of sugarcane plant, that is a repeated PYQ. So all these statements, right, you need to revise it. So you can just use this PDF, right? You can use this PDF and you can just simply revise these topics. Okay, okay, right? So yield of sugarcane will be increased up to 20 tons because it will increase the height. Baki delay the senescence, but when we talk about the senescence, mainly for delaying the senescence, we go for the cytokinin, right? We go for the cytokinin. Is that clear? Is that clear? So in plants or the fruits, right, when we keep them on the tree longer as to extend the market period, we can apply the gibberellin acid. So spraying juvenile conifers with gibberellic acid will speed their maturity period, and the early seed production will be there. Then cytokinin, let's discuss about the cytokinin, then we are about to finish it. After that, we'll start the photosynthesis. But yeah, when it comes to photosynthesis, let's have a break first. Okay, let's have a break first. So cell division, cytokinin, cell division, it is main function. And here you will discuss Richmond-Lang effect. So what is that Richmond-Lang effect? So that Richmond-Lang effect is the delay of senescence. What's that? It is the delay of senescence. Okay, that is the Richmond-Lang effect. So cell division, main function, morphogenesis. So my dear students, what is the meaning of genesis? Genesis means formation. What is the meaning of genesis? It means formation. Morph means form. Morph means form. So morphogenetic changes, जैसे root and shoot development, it is induced by cytokinin and auxin in presence of tissue culture. So when there is low cytokinin but auxin is high, roots will differentiate. But when there is high cytokinin, low auxin is there, shoot will differentiate. It is important. Okay, it is important. Understood? It is important. Understood? Pakka? Next. So this helps in overcoming apical dominance, or you can say that it counteracts the apical dominance. So delay in senescence, Richmond-Lang effect, baki beta. So promote lateral shoot growth and adventitious shoot formation. Then comes the ethylene. So whenever you talk about the bioassay of ethylene, whenever it comes the ethylene, so always remember triple response. What you need to remember there? You need to remember triple response, right? Right? What you need to remember? Triple response. Okay, so triple response means let's suppose the seed is germinating but a hurdle is there, right? So basically there is the swelling of the horizontal axis, right? Then apical bud formation is also there, right? So basically what will be there? What will be there? You can have a look. First of all, this ethylene is very important for the ripening of fruits. Okay, so banana, apple, tomato. So nowadays even ethylene, this is important. Ethylene, this is important, which is CEPA, 2-chloroethyl phosphonic acid. It is used commercially for this purpose. So it increases the respiration rate during the ripening of fruit, and then the respiratory climatic conditions will be there. So stimulation of senescence. So that is its inhibitory effect. So it stimulates the senescence and abscission of flower and fruit, right? So flowering and synchronizing fruiting in pineapple. So related to pineapple question was also there in auxin. So do revise it. So triple response mein apical hook formation is there in dicot seedling. Apical hook formation, swelling of axis is there. That is horizontally. Horizontal growth of seedling is there. So this is what we consider in the triple response. So whenever you talk about it, discuss the ethylene, and this is also the bioassay of ethylene. Okay. This is also the bioassay of ethylene. What's that? Bioassay of ethylene. Okay. Okay, and then promotes the root growth, stimulate the formation of root hair, help the plant to increase their absorption surface. Baki femaleness in cucumber will be promoted by this, and it will break seed and bud dormancy, right? So it breaks that dormancy. Okay, so it promotes rapid, and when it comes to deep rice water plants, ye dekho, again PYQ. So it is totally memory based. If you think madam will do, I can explain it, right? But it is of no use, beta, because it is a marathon. We have to focus on the most important topics here, right? If let's suppose if this chapter is weak but other chapters are well prepared, then you can just watch my game of NEET session as well. That is also for 3 to 4 hours. Okay, for this particular chapter I'm talking about. So ethylene will promote what? It will promote the rapid internode or petiole elongation in deep water rice plants. That helps the leaves and the upper part of the shoot to remain above water. That's important. Okay, that's important. Done. Then abscisic acid, also known as stress hormone. So whenever plant is under the stressful condition, it will release so that it can help that plant to survive in that stressful conditions. That is the agenda basically, okay? That is the agenda basically, okay? Okay, so first thing is it induces senescence and abscission, right? So how? Because it will increase the activity of cellulase and pectinase. Then it will induce bud and seed dormancy, right? It increases the bud and seed dormancy. Then in seed development, maturation and dormancy, it is having the role. It induces dormancy, right? So that plant can survive in unfavorable condition. It closes the stomata if water plant is under water stress. So it is the antagonist, the opposite of GA, right? So this is all about the bioassay. Then we will go for the photosynthesis part, right? So photosynthesis mein one question will be there from the experiments. Baki beta, after experiments we have to jump directly to the chloroplast structure and photosystems. I'll tell you about photosystems, chromatographs and all, because if you have completed it earlier, then it will be very easy for you to remember that. Otherwise, I'll touch the basics. I'm not saying I will not. So in this way, we have to revise it, okay? So we have to speed up. So let's have a break for 15 minutes. That's fine. That's fine now. Am I audible? I hope I am. Okay, so let's start the photosynthesis, or do you have any other demand? What do you want me to start? So beta, basically we are taking these sessions for you people only. Okay, so I really want you people to revise. Okay, and even if let's suppose there is a chapter which you think in the marathon ma'am finished like we discussed very quickly, said then you people can do one more thing na, then you people can just watch my like detailed lectures. You can watch my last year ka marathon, right? That will be sufficient enough for you. Okay, trust me, that will be sufficient enough for you. So we are here for you people. We want you people to get good marks. We want you people to work hard, because see, it doesn't matter how well I teach, but if you people will not revise, if you people will not solve the questions, it will be of no use. Okay, so my throat is literally, it is in a very, very, very bad condition, and I don't know how am I going to manage it further, and how am I going to manage my next marathon. Okay, but anyhow, let's go for that. So even this NEET time, it is not just hectic for you, it is hectic for us as well. So let's start the next chapter, that is photosynthesis. So students, if you are thinking that what about the chapters like biological classification, or what about the chapters like living world, from living world one question will be there, right? You can watch my one shot for that if you want to, but I really want to give you the suggestion that if you have completed the syllabus, then go for that. Otherwise, do revise the most important topics first, because that is going to be very simple for all of you. I'll be very honest about it. Number one. Number two is, now plant kingdom, I know from so long we didn't revise it, so I started with the plant kingdom. Anatomy done, your plant growth and development done. In plant growth and development, memory based topics are there. Yes, we have to finish it. So basically within 6 hours we have finished 3 chapters, and all of them were important. Okay, so now we will start with the photosynthesis. So if you are studying this topic for the first time, then yes, you are going to find it difficult. But if let's suppose you are doing it for the third time or second time even, then you are going to find it easy. So we will pick up the most important things right now, and we have to speed up, because I really want you, honestly, I'm telling you that no doubt you must be thinking, nahi ma'am, your energy level is quite right, your energy level is good, but honestly speaking, today I'm tired, I'm genuinely tired, right? You can see this thing from my eyes as well, that genuinely I'm tired, but yeah, we'll keep on going. Okay, we have to revise the things. Okay, so now please listen to me. So let's discuss the photosynthesis. Let's pick up the most important part, then let's go for the respiration, where we have the tricks as well. Okay, and my main agenda is to complete it till respiration only, right? I'm not saying the other chapter is cell cycle. I have repeated it multiple times in top 50 concept videos. We started with the cell cycle, you can follow that. I have posted a short video where we have revised the cell cycle in less than 20 minutes. You can follow that, because it has covered all the PYQ. Cell chapter, I think that is also I have revised multiple times, especially the cytoskeletal elements, because I know students miss it. Okay, and now when it comes to the biological classification, fungus part is important, and even in my short video series session, I will take care of that fungus part too, and the families part too. So now when it comes to morphology, so I believe from so long we didn't revise the morphology, right? From so long we didn't revise the morphology, right? Yes or no? From so long, this is what I'm thinking. Don't worry, even I'm worried about it. From so long we didn't revise morphology. Am I right? Wow, ma'am, वो dark circles भी beautiful लग रहे हैं. First of all, अच्छा, who's it? Obito. So you have confirmed it that I have dark circles, right? Ma'am, if you're tired, please stop. Meaningless session is not fruitful. What is the meaning of meaningless session? Even if I'm tired, I'm going to continue it. You know it very well, right? Right. My sessions are never going to be meaningful. Meaningless, sorry. My sessions are never going to be meaningful. My sessions are never going to be meaningless. Right, so chill, okay, right? So yes, even it's visible, ma'am. Dark circles are visible. Oh my god, so you want me to put makeup? You people want me to put makeup? No, I sleep, but from last few days, it's quite hectic. Okay, so all I'm saying is let's discuss the photosynthesis and respiration part. And yes, even I think from so long we didn't revise morphology. So families part I'll pick it up separately. So I'll make short videos for morphology as well, right? I'll make, because it is completely memory based. Okay, so I'll make the short videos for morphology as well. Firstly, let's try to finish it today only, right? Let's try to finish it today only. So I need your support. Be there with me, right? Let's continue. Okay, so I'll try to discuss it today only, but if not, I'll put the short videos. Okay, I'll put the short videos. Okay, chaliye. So let's start the photosynthesis. So photo means light. Very simple it is. Photo means light. Photons. Synthesis means formation. So you know that in the presence of light, there is the formation of food. So here there is the formation. Here there is the build-up. You are going to make something. You are going to make something. So can I say that it is an anabolic reaction or anabolic process? It is, of course. It is the anabolic process, because here what is happening, your carbon dioxide and water in the presence of light, right? Of course, chlorophyll is required, right? Enzymes are required, and it is going to make C6H12O6. That is your glucose, and the oxygen will be released, isn't it? The oxygen will be released, isn't it? Yes or no? The oxygen will be released. Yes or no? Yes or no? The reaction is not balanced, but this is what we discuss in the photosynthesis. So we have to start with the experiments first. Okay, so from experiments, one question can come. So it is PIS T cartoon network RHK. What is the trick that we are going to follow now? What is the trick that we are going to follow now? It's PIS T cartoon network RHK. It's PIS T cartoon network RHK. Joseph Priestley, Jen Ingenhouse, Julius von Sachs. That is my way to remember that, right? It's PIS T cartoon network RHK. It is PIS T cartoon network RHK. So you can repeat it as well. So it is PIS T cartoon network CN RHK. So basically, firstly, let's start with the scientist name, right? So we have Joseph Priestley. We have Joseph Priestley, right? We have Jen Ingenhouse. We have Julius von Sachs. We have T.W. Engelmann, right? Here you will talk about the first action spectrum. Cartoon network is basically your Cornelius Van Nile, right? He is basically your Cornelius Van Nile, who has given the what? Right? It is Cornelius Van Nile, and then it is Ruben Hesed Cumming. It is Ruben Hesed Cumming. So these are the scientist name. So let's start with the first experiment. So Priestley 1700. So in this sequence you have to remember it. In this sequence you have to remember it. Right, bache? Right, bache? So plants, they restore their air that is removed by breathing animals and burning candles. So in 1774 he discovered the oxygen. So here you guys can see it is the quick revision. Okay, it is the quick revision of all the experiments, and that is why we are here. Right, that is why we are here for this particular marathon. So basically the point is, basically see, when you talk about the first experiment here, you all can see this. Yes or no? You all can see this. Just basically, what are we talking? We are firstly talking about the moles half-leaf experiment, right? What is the first thing that we have to discuss here? We have to discuss the moles half-leaf experiment. Yes, bache. What is the first thing that we have to discuss here? Everybody, please. Everybody, please. So what is the first thing that we have to discuss here? It is the moles half-leaf experiment. Okay, I'm writing it here. It is the moles half-leaf experiment. Moles half-leaf experiment. This is the first experiment that we are studying here. So when you look at this particular experiment, right? When you look at this particular experiment, so we are talking about the starch formation in two leaves, right? Starch formation in two leaves. So firstly, there is a variegated leaf, right? It's a kind of multi-layered having different colors in that, and it is partially covered with a black paper. So see, let's suppose if this is a leaf, a part of it is covered with the black paper and rest of it is exposed to the light, right? So when you test these leaves, right, for what? For the presence of starch, right? So basically we will get it in the green part only. We will get it in the green part only. Are you getting my point? Are you getting my point? Yes? Yes? Okay, and this is not, this initial experiment is not Moll's half-leaf experiment, that is the next one, right? So firstly, this is a general thing that is telling us about the starch presence. That is telling us about the starch presence where? In the green parts. Now, after that, we have the Moll's half-leaf experiment. This one, right? So it means that I need a cup of tea, है ना? Cup of coffee rather. So Moll's half-leaf experiment it is. What is that? Moll's half-leaf experiment it is. So when we are talking about Moll's half-leaf experiment, then in that half-leaf experiment, earlier one was normal experiment where we are just showing that green part, right? Where we are just showing, what are we showing? Where we are just showing that it is the, yes people, that it is what? It is that green part where the starch formation is there. Now next, in the next case, what are we going to do? A part of leaf is enclosed in a test tube. That test tube is having KOH. What that test tube is containing? What that test tube is containing? That test tube is having KOH. What that test tube is containing? KOH, right? Right? Yes or no? So it is potassium hydroxide. It is potassium hydroxide. So it is basically a CO2 absorbent. It is going to absorb the CO2. What is the role of KOH here? It is going to absorb the carbon dioxide. So here the part is, the part of leaf is enclosed in a test tube and that test tube is having the KOH. So as I said, it is the CO2 absorbent. It is the CO2 absorbent. So it will soak cotton, right? It will absorb the CO2 and other half is exposed to the air. Like a part of leaf is there in a flask where KOH is there, another part is exposed to the light. And when you will check the starch, where are we going to see this? The part, right? The part which were not there in the flask, which were outside. So basically this shows that CO2 was required for the photosynthesis. Now, when you talk about the Priestley's experiment, right, this is what we have to remember in my notes, right? Everything is in a very brief manner, it is explained there. So here you can see in the bell jar experiment, right, the candle, the mouse will die, the candle is not burning. Then here we have to put a mint plant. Why are we talking about the mint plant here? Because this mint plant is a sciophyte. What's that? Sciophyte means it can grow. It's a shade plant. In simple words, it is what? It is a shade plant. In simple words, what is it, beta? It is a shade plant. That's why we are using it here. This is one of the explanations. Okay. So in first experiment, what happened, bache? In first experiment, what happened? Priestley observed that a candle which is burning in a closed space, that is in a bell jar, it will get extinguished. It will get extinguished. Why? Because a mouse, and of course, because of that, the mouse will soon suffocate. So a burning candle or the animal, they are breathing that air. They are somehow damaging the quality of that air. Okay, they are somehow doing what? They are somehow damaging the quality of that air. What are they doing? They are somehow damaging the quality of that air. So when you put the mint plant there, right, the mouse will stay alive. So he hypothesized that plants will restore the air, whatever burning animal and burning candle has removed. So here in the notes itself, only that keywords are given. Now after Priestley, JPGI, Jen House experiment will be there. So what he did, he tried it with the similar setup as used by the Priestley, but by placing it once in the dark and once in the sunlight. He placed that setup in the dark as well as in the sunlight, and he showed that sunlight is essential to the plant process. Okay. So in his experiment, what he did, an aquatic plant that showed in the bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Okay, while in the dark they did not. So in House experiment, in that particular experiment, the aquatic plant, right, which was there in the bright sunlight, it was showing the bubbles. That bubbles were showing the oxygen, right? So he showed that it is only the green part of the plant that could release the oxygen. Okay, that could release the oxygen. So in 1854, Julius von Sachs, next experiment, JPGIJS, right? So what they did, that production of glucose will occur when plants grow, right? So glucose is stored as starch, and he was like, there is a green substance. He was not able to figure out that it is the chlorophyll at that point of time, but it is the chloro. Now we know it as chlorophyll. So in the green parts, right, the special bodies are there. Basically, there we have the glucose. This is what we are trying to say. Now, next experiment is very important. And yes, this is something we need to remember, because here you are talking about the Here you are talking about the first action spectrum. So here we are talking about what? We are talking about the T.W. Angelman. T.W. Angelman. What he did? He used a prism. Because this prism can split the white light into its different components. Because this prism can split the white light into its different components, into its spectral components. Then he illuminated a green algae. So let me show you this diagram. So this is the diagram. So when this white light will cross this prism, it will split into its different components. Now this is the green algae. So what he observed, that in certain regions, and here in the water, in this medium, your aerobic bacteria is there. So if I am saying aerobic bacteria, so it's simple. My dear students, if I am saying aerobic bacteria here, means they need oxygen. What is the meaning of that? It means they need oxygen. So of course they observe that in the certain regions, like let's suppose this is the algae here, right? So in certain regions, the aerobic bacteria, they are gathered. So let's say, and that regions are basically your blue light and the red light. Blue light and the red light. So of course, why are they gathered here? Because these are the regions where the photosynthesis is there, the oxygen will be released. So here, the bacteria were used to detect the sites of oxygen evolution. So it is cladophora. So remember, cladophora is a green algae. Today itself we have revised it. So basically we have to detect the sites where oxygen is evolved. So bacteria, they accumulated in blue and red region of the split spectrum. So this is the first action spectrum. Beta, when we say action spectrum, right, it will tell us about the rate of photosynthesis. It will tell us about what? It will tell us about the rate of photosynthesis. It will tell us about what? It will tell us about the rate of photosynthesis, right? So it resembles roughly the absorption spectra of chlorophyll A and B. Okay? Okay? Understood? Are you sure? Are you sure? Next experiment, right? So next experiment is a very important experiment. This is Cartoon Network, right? What you have to remember CN, Cartoon Network. So Cornelius Van Nile. What we have to remember, Cornelius Van Nile. That is what we have to remember, okay? So here in this experiment, so they are saying that by the 19th century, the key feature of plant photosynthesis were known. Right, the plants could use light energy so that they can make the carbs from carbon dioxide and water. Right, they can make the carbohydrates from CO2 and water. So this is the equation. Now here in this equation, the CH2O, please do not make the mistake here, students. It is showing carbohydrates. Right, if this equation is there in the paper and if somebody will ask you what is the CH2O, what are they talking about? They are talking about the carbohydrates. Okay. Okay, everybody wake up. Wake up. Yes, please wake up. So, a milestone contribution to the understanding of photosynthesis was that made by a microbiologist. Now, here, beta, what are they saying? First of all, you have to remember what Cornelius Van Nile, what he studied purple and green sulfur bacteria. What he studied the purple and green bacteria. What he studied the purple and green bacteria. And he saw that a light-dependent reaction in which hydrogen from a suitable oxidizable compound. This line is very important. In which hydrogen from a suitable oxidizable compound. Right? Right? Hydrogen from a suitable oxidizable compound will reduce the CO2 to carbohydrate. So ultimately we are saying that it is the hydrogen, right, from a suitable oxidizable compound that is going to oxidize the CO2. So basically we are getting the carbohydrates after the after this. What? After the, basically we need the oxidizable compound that can reduce the CO2. So when CO2 is reduced, what will we get? We'll get the glucose. Now when you look at this particular reaction, here they are showing 2H2A, which is this A can be anything. If this A is O, matlab O2, oxygen, like if this A is O, so here oxygen will release. If this at the place of this A, if S is there, if at the place of this A, if S is there, if at the place of this A, S is there, then sulfur will release. So basically, initially it was believed that this oxygen, right? Initially it was believed that this O2 is coming out from CO2, which is false. Which is false. This oxygen is not coming out from CO2, rather it is coming out from the water. So basically in the photosynthesis, overall I'm telling you what is going to happen. Your carbon dioxide will be reduced and this compound will be oxidized. So oxidation reduction is going on when you are talking about the photosynthesis. Redox reaction is going on. So here they are saying, right, they are saying what is happening. 2H2A, CO2, right, 2A plus CH2O plus H2O. So A is released, right? So this is what you have to keep in your mind. So H2O is the hydrogen donor and is oxidized to oxygen. So it is the H2O which is the hydrogen donor here. So if at the place of H2O, if we have H2S, then hydrogen donor will be, right, it is the hydrogen donor for purple and green sulfur bacteria. So sulfur will be released at the place of oxygen. Right, at the place of oxygen. So what is inferred? So he inferred that the oxygen evolved by the green plant, it comes from H2O, right? But it was later proved. So now the last experiment that I'm talking about of Rubin, Hasid and Kamin, the scientist name is not given, but they experimented. Okay, but what they did, bache, but they experimented. Okay, they experimented. So they suspended chlorella. Right, they experimented on what? They experimented on chlorella. So they suspended chlorella, you understand, green algae, in a water medium having radioactive isotope, having what? Having C. So they suspended the chlorella in a medium where the water is basically radioisotope of water is there. Right, radioisotope of water is there. What is there, bache? What is there, beta? So radioactive heavy isotope of oxygen is there. Just normally it is O16. Normally it is what? Normally it is what? O16. But they suspended that chlorella in a medium where it is the radioactive. Right, where it is the radioactive, where it is what, beta? Where it is the radioactive heavy isotope of oxygen. Radioactive heavy isotope of oxygen it is. And when you are talking about the radioactive heavy isotope of oxygen, you are talking about the O18 here. Now with the help of their right, now with the help of their mass spectrometer, right, they checked it and they just checked that the oxygen that was evolved, that was having O18. So that is how they proved that from the water it is the oxygen in photosynthesis is coming out from that oxidizable compound. Okay, okay. So Rubin, Hasid and Kamin, they suspended chlorella in a water medium where radioactive heavy isotope of oxygen is there, that is O18. Right, then with the help of mass spectrometer, they checked it out and they were like, the oxygen which is released, okay, the oxygen which is released is O18. So it indicates that oxygen is coming out from the water. This part is clear? This part is clear? Are you sure? Now, where does this photosynthesis will occur? So, of course, you have to talk about the chloroplast here, and overall, you have to talk about the plastid here, right? What we have to discuss here, we need to talk about the plastid here. So, basically, if I talk about plastid, you know that there are three types. Let's have a quick revision of that as well. So, we have the chloroplast there, right? We have the chromoplast there. We have the chloroplast there, we have the chromoplast there, and then we have the leucoplast as well. Leucoplast as well. So they all are developed from a proplastid, right? They all are developed from proplastid. And let me tell you, this proplastid, it can be, thank you, it can be converted, right? It can be converted. So see, with us, even our families, you know, they are not sleeping, they are also up because they are also waiting for us, है ना? So we are basically because we are also taking the marathons late night, and this is the demand right now, this is the need basically, and we want you people to study it because during night time, you know, it's quite silent kind of environment. So of course, right, it's good for you as well. And if you will complete the topic now, you can take a sound sleep, then you can wake up, then in the evening you can revise and you can go for the next session as well. So our families are also waking up with us and they are also helping us and supporting us. So please do revise it, do study it, and please, I don't know why, like, yeah, it is a human tendency that we wake up at the last moment. So we are teaching from the starting of the session, but now students are sincere, right? It's coffee today because I really need to wake up. Okay, chalo. So chloroplast, chromoplast, leucoplast is there. So from chloroplastid they'll form. So when it comes to the most common plastid, so this is what we have. Okay, so when it comes to the most common plastid, this is what we have. Okay, okay, so leucoplast, you know that here it is for the storage only. Remember, storage only. Myeloplast, elioplast. Do you remember that? Eleuroplast, myeloplast, elioplast. Okay, so let's discuss the chloroplast first, right? And that is what is required right now. Okay, so last year a question was there and many people were like, controversial question. There was nothing like that. No, last year basically. Yeah, last year, I guess, last year. Chalo, so this is chloroplast. First of all, it is double membrane bound organelle. Without any doubt, it is also a double membrane bound organelle, right? Double membrane bound organelle, and it is semi-autonomous. What's that? And it is semi-autonomous. What's that? It is semi-autonomous. Is that clear? So when it is the double membrane bound organelle, so of course this one is the outer membrane, right? 110%. This one is the outer membrane, and here you have the inner membrane as well. Let's go for the short forms now, right? And in between these two, either you can call it as intermembrane space, either you can call it as intermembrane space, or we can also call the same as periplastidal space. It is also known as periplastidal space. So intermembrane space and periplastidal space is same. Now here you know that it will have 70S ribosome, right? And it is also having the circular DNA. That's why we are calling it as semi-autonomous, right? It is not completely dependent on the nucleus only. Then here, these are the kind of structures which we need to understand, people. Okay, these are the kind of structures which we really need to understand and which we really need to revise. Something like that. Yes. So here, this is stroma that we have. This matrix here is stroma, and in this matrix, students, the dark reaction will occur. We'll tell you what dark reaction is. Now look at this part. Look at this part. See, inner membrane and outer membrane is there. Remember, outer membrane is more permeable. Matlab, it is having pores. Porins are there. It is more permeable. Inner membrane is selectively permeable. Inner membrane is selectively permeable. Outer membrane is more permeable. Inner membrane is selectively permeable. Here the matrix of this chloroplast is known as the dark reaction, and here you will have the enzymes, all the enzymes for this dark reaction. Now look at this particular part. These structures. This is a granum. What's that? It is a granum. Granum is singular, plural is grana. This in between, it is intergranal thylakoid, or it is also known as fret lamella. It is also known as stroma lamella. Okay, you can call it anything. Done. So that's how you can see. This is what you have. Right? Right? Or see, so this is granum. Singular is granum, plural is granum. So here, when you look at this part, right, just focus on this thing, this sac-like structure, which we call it as thylakoid. Here within it, it is the lumen of thylakoid. So this thylakoid is a membrane bound structure. We have the membrane here. So it is the membrane bound structure. What kind of structure it is? It is the membrane bound structure. Now students, when you look at this membrane, right, we have to discuss it as well. So of course, that point is same, that there will be phospholipid bilayer and all. And here you will see that certain protein complexes are present. What do we have here? We have certain protein complexes here as well, right? That we have to discuss. So this is what we have right now, right? This is what. So basically all these thylakoids which are membrane bound, they will pile over each other, right? They'll stack over each other and they are forming the granum. Right, they are forming the granum. So when you talk about the thylakoid, when you talk about this thylakoid membrane, right, when you talk about this thylakoid or when you talk about this thylakoid membrane, you will see that here the light reaction will occur because protein complexes are present there, right? So imagine it like this. This is your granum. In its surrounding, of course, stroma is there. Even if I'm picking up one thylakoid, this is the lumen, in the surrounding stroma is there. So we will see, we will read how that proton gradient will be created and how, right, how that proton gradient will be created and how further, you know, the movement and everything will be there here. Is that clear? Is that clear? So outer membrane is more permeable, inner membrane is comparatively selectively permeable, right? So stroma, I told you about stroma as well, right? Bache, the enzymes are present. We'll discuss which enzymes are there, but I hope this part is clear. Now, here, right? So this is your one granum. Here you have your another granum. And as I said, you people are having intergranal thylakoid in between. You people are having intergranal thylakoid in between. What do you have? You have intergranal thylakoid. It is also known as stroma lamella. It is also known as fret lamella. Fret lamella. So yes, we have to remember it as well. Okay, so now let's talk about the layers here. Okay, let's talk about the layers here. So when you are talking about this thylakoid membrane, right, when you are talking about this thylakoid membrane, right, this is important to remember. We will discuss it now as well. So you will have appressed membrane, yeah, appressed part and non-appressed part. So what do we have here, bache? We have the appressed part as well as the non-appressed part. Appressed part as well as the non-appressed part. So appressed part. So it is basically inside the membrane. Just appressed part. Like the partition membranes there. I'll show you the other diagrams as well. So it is basically also the partition membrane. Right? It is present between thylakoids. And of course, if it is the non-appressed part, right, it is the marginal one. Am I right? It is the marginal one, right? So marginal one are the stroma lamella and the surface of granum, right? So these non-appressed are the basically they are the marginal one. So they are basically surface of granum or even the fret lamella. That is what we have. Okay, that is what we have. Okay, that is what we have. Okay, right, just keep it in your mind. Right, so look at the chloroplast here, look at their positions here, right? We will discuss it as well, but I'll stick to the important topics. Okay, chaliye. So this is the appressed and the non-appressed part here. Okay, so you can see appressed part, a partition membrane. In between, non-appressed part will be the exposed part. It is the marginal part. Okay, it is the marginal part. Yes, you need to understand that, right? Just this is the stroma thylakoid, non-appressed regions, end membranes. So appressed is basically non-appressed is this outer one, right? Exposed one, this one, and appressed one is inner one. So this is what you have to remember, right? This is what you have to remember. Baki, when it comes to the chloroplast and the light intensity, you know that the chloroplast will change its position. Just say, for an example, if there is the high light intensity, then what will happen to the intensity? So what will happen to the chloroplast? So chloroplast will be, yes, parallel to the incident light. It will be parallel to the incident light. It will be parallel to the incident light. Now, after that, if there is low light intensity, if there is what? Low light intensity, in that case it will be perpendicular. It will be what? It will be perpendicular to the incident light. It will be perpendicular to the incident light. Are you getting my point? Are you getting my point? Are you getting my point? Right. Then if there is the moderate light, right, moderate amount of light it is, then the chloroplast distribution will be random. Then chloroplast distribution is random. So yes, even this is something that you all can note down. Okay, this is something that you all can note down. Chaliye. So oppressed part, non-oppressed part, the diagram part clear? The diagram part clear? I hope it is clear to you. So one thing that I would like to mention here, and that I believe is important as well, and students should know about it, and that is your PS1, photosystem 1, is mostly there in non-oppressed part and stroma lamella. They are having this, right? They are having this. Now, PS2. Now, the PS2, it is there in the oppressed part. Where are you going to see this? In the oppressed part. So we will discuss it as well, right? We will discuss it as well. Is that clear? We are going to discuss it as well. So I believe up to this part, all clear. So you can see the thylakoid membrane here. You can see the lumen here. Surrounding me, there will be the stroma. And here you can see the protein complexes which are located. And here the light reaction will occur. And that is something we need to understand. But before that, let's focus on the pigment part. Okay, let's focus on the pigment part. Is that clear? Let's focus on the pigment part. First of all, when you are talking about the photosynthesis, we need to focus on PAR, P-A-R. Just overall, if I talk about the light, overall, if I talk about the light, right? If I talk about the light, so you know that 8 types of radiations are there, and it depends upon the wavelength. And it depends upon the wavelength. So basically in that light, we have to focus on PAR, and that is somewhere in between 400 to 700 nanometer. So PAR is what? It is photosynthetically active radiations. Yes, students, everybody, the energy level should be high, right? Energy level should be high. Have a cup of tea or coffee, but not too much. It's not good for health, right? Just have a cup of coffee or tea and please focus here, right? Right? So PAR, photosynthetically active radiation, that is somewhere in between 400 to 700 nanometer. That is somewhere in between, right there, that is in between 400 to 700 nanometer. Is that clear? That is 400 to 700 nanometers, right, bache? So yes, this is what we need to understand. Now, look at these graphs. They all are very important here. So what are they saying? They are saying, they say we know that leaf pigments are there. Right, if we try to separate the leaf pigments from any green part, how can we do it? With the help of paper chromatography, again a PYQ. So if we want to separate the pigments from a leaf, what can we do? We can go for the paper chromatography. What can we do? We can go for the paper chromatography. So it is basically a separation. It is basically the separation of leaf pigments, and the color that we see in the leaf is not due to the single pigment, but there are many pigments. There are many pigments, right? There are many pigments, isn't it? So we have the chlorophyll A, we have the chlorophyll B, xanthophylls are there, and carotenoids are there. So if you are, okay, if you are, right, if you people are reading this chapter for the first time, right, then please note it down, because from this part the question can come. And of course, you know that what else you need to watch for the quick revision. This is the video for the quick revision. This is the video for the last ultimate day plan. Do not miss it out. And other than that, right, other than that, you people can also see that the quick revision sessions are also there, right? So same I will post for the photosynthesis, same I will post for the respiration as well. Okay, so here you can see all the things are clearly mentioned here, right? So just focus on the revision. Okay, bache? Just focus on the revision. Are you getting my point? Just focus on the revision here. Take. So do not miss it out. Now come back here. So paper chromatography part is there. Paper chromatography part is there. Yes or no? Paper chromatography part is there, right? And here chlorophyll A will give color in bright or blue green, right? In the chromatogram, yellow green, sorry. Bright or blue green, yellow green, and yellow only when it is the xanthophyll. Carotenoids mein yellow to yellow orange. Very, very important information. It is very, very, very important information. Okay. Okay. Yes. Done. Done. Are you sure? So their colors are important. Let's focus on their structure as well. So whenever you talk about the chlorophyll, yes, we have to read about the chlorophyll A and the B, right? First of all, just have a look. So basically, students, the central metal ion, right? When we talk about the central metal ion, it is going to be magnesium. That is very important. Right, the central metal ion is what? The central metal ion is magnesium. What is the central metal ion here? The central metal ion is magnesium. So other than that, when you look at the structure here, right, firstly let's discuss the structure of chlorophyll, then we will discuss the another point. So basically here you will see that there will be a head and there will be a tail. Overall, this is the kind of structure that we see, right? So here phytol tail is there, right? Phytol tail is there, and bache 4 pyrrole rings of so this is the phytol tail and porphyrin head. That is the terminology that we use. So porphyrin head is there and phytol tail is there. What do we have here? Porphyrin head is there and phytol tail is there. Porphyrin head is there or phytol tail is there. So that is what we have. That is what we have. Porphyrin head and phytol tail is there, right? So in this porphyrin head, what do we have? Right, it is having squarish tetrapyrrole ring. Square like, yes, squarish tetrapyrrole ring, right? Squarish tetrapyrrole rings are there, right? Then now I'll speed up as well, right? So when you look at these tetrapyrrole rings, magnesium is the central metal ion. So you will see this is the phytol part, and here this is your first ring, this is your second ring, this is your third ring, this is your fourth ring. Okay, so as I said, the central metal ion is your magnesium. Which one is the central metal ion? It is your magnesium. So here you will see that there is methyl group at the third position, right? This is the second ring, the second pyrrole ring. And at third position, second pyrrole ring mein, at third position CH3 is there. At third position CH3 is there. So it is in chlorophyll A, but in chlorophyll B, this CH3 is replaced by CHO. That is, aldehyde will be there at the place of methyl. Do we need to remember it? Yes, of course. We need to remember it. Okay. Yes, of course, we need to remember it. So chlorophyll A is having that CH3 group. Chlorophyll B is having CHO. Chlorophyll B is having CHO here. Is that clear? Is that clear? Right? So it is going to be, oxygen is going to be 5, N is going to be 4, and then it will be Mg. Then it will be Mg. So it is in second pyrrole ring. At third position CH3 is there. Now this is the formula of chlorophyll A. Okay, I'm not telling you to remember, but at least you should know CHON and Mg is there. And if it is the chlorophyll B, because at the place, carbon number will be same, but at the place of HO you have CH3, so 2 extra hydrogen will be there, so H will be 70. Yes, so chlorophyll B mein nahi, chlorophyll, in case of chlorophyll B it is yes CHO, but in chlorophyll is CH3, so basically 2 hydrogen are less, so C55, H72, but yeah, there will be one extra oxygen, so that is how it is going to be. So this is the formula that we have here, right? So this is for the chlorophyll B. So I am not saying to remember the formula, but yeah, this part is important when it comes to the structure of the chlorophyll, right? This part is important when it comes to the structure of the chlorophyll. Is that clear? Right? So when there is the chlorophyll, so protochlorophylls are going to form the chlorophyll, right? So when there is the chlorophyll synthesis, that is again a PYQ, right? But yeah, now mineral nutrition part is deleted, but still you should know about it. So when you talk about the chlorophyll, when you talk about what? When you talk about the chlorophyll, when you talk about what? Chlorophyll. So basically succinyl coenzyme A, and this is the amino acid that is used here, that is glycine, right? So this can be asked as a question, and it will form what? It will form protochlorophyll. It will do what? It will form protochlorophyll. It will form protochlorophyll. What will it do? Protochlorophyll. Now, of course, light is required, right? For the chlorophyll synthesis, light is required. If light is not there, chlorophyll will not get synthesized. And other than that, iron, Fe, is also required when it is the biosynthesis of the, right? So reaction is catalyzed by iron. Fe is required. So Fe is required for biosynthesis of chlorophyll. It is required for biosynthesis of chlorophyll. So if I talk about the central metal ion, that is magnesium. If I talk about the central metal ion, that is magnesium. But when we talk about the biosynthesis, so for the biosynthesis, what is required, people? For the biosynthesis, what is required, people? For the biosynthesis, iron is required. Okay, for the biosynthesis, iron is required. So that is something you have to keep in your mind. Okay, that is something you have to keep in your mind. Okay, so this is for the chlorophyll, right? This is for the chlorophyll. Baki, you know that the, so this is the chlorophyll A and chlorophyll B as well. Okay, so here you have the different pigments. Now of course we have the carotenoids also, right? TK chlorophylls are there. These are the pigments that are going to absorb the light, right? But we have other pigments also, just like carotenoids, right? This is what we have discussed now, ki carotenoids are there. So let's discuss about them. Take, let's discuss about them. So first thing is what? First thing is carotenoids. First thing is what? First thing is carotenoids, right? It is the carotenoids. So first of all, they are lipid soluble. What are they, students? They are lipid soluble. They are not water soluble, lipid soluble. A water soluble is your anthocyanin, right? So carotenoids are lipid soluble, yellow to yellow orange colored pigment on, right? So it is yellow to yellow orange colored. What's that? It is yellow to yellow orange colored. Yes, bache? Yes, bache? Yes or no? Yes or no? So it is basically chemically they are terpenes, right? Chemically terpenes, and they are most stable. Okay, so first point, second point, third points in the form of points. You should make the notes, bache. So in the case of carotenoids, right, they absorb light energy, they'll transfer it to the chlorophyll A. So basically it is an accessory pigment, right? Where do we put it? We put it in the category of accessory pigments. Okay, we put it in the category of accessory pigments. Why? Because see, right, that whenever you will talk about accessory pigments or the shield pigments, so basically what is going to happen, bache, that chlorophyll, okay, sorry, carotenoids, actually, bache, they protect the chlorophyll from the photooxidation. They protect the chlorophyll. So carotenoids, they protect the chlorophyll from photooxidation. Photo means light. So in the light can even oxidize the chlorophyll, but that photooxidation is prevented by carotenoids. Why? Because they pick up the nascent oxygen by doing what? By picking up nascent oxygen and converting it into harmless. Right, so carotenoids, what will they do? They will convert it into the harmless 1. So they are known as shield pigment. So this is something we need to know while understanding the photosystems. I know now I am going slow, I have to speed it up. So you can write it down. So they can pick it up into harmless molecular stage. So they are known as the shield pigments. Is that clear? Is that clear? So we have the examples here. Your carotenes and xanthophylls are also the kind of carotenoids, right? So when you talk about the carotenoids, you have the examples. Jैसे we have the carotenes and we have the xanthophylls. Xanthophylls, yellow, right? Yellow. Dirty fellow. So carotenes mein we have the example alpha, beta, gamma carotenes. So here we are mainly focused on this part na, beta carotene. So beta carotene, your beetroot color is due to this beta carotene. Yes, beetroot color is due to what? Due to this beta carotene, isn't it? Isn't it? So basically in the plants, the different colors, even the carrot is also having the beta carotene. Sorry, is that clear? Is that clear? So one is xanthophyll is yellow. So when you talk about the yellow one, so you have to discuss the lutein. So this is yellow orange. So here you have lutein, right? That autumn foliage, autumn leaves, right? They are yellow because of that. Your fucoxanthin, right? Your brown algae is brown in color. Your brown algae is brown in color. So it is also due to this. It is also due to this. Is that clear? Is that clear? So I told you what accessory pigments are. I told you this beta carotene, it is there in the carrot as well. And this beta carotene is also precursor of vitamin A. Remember, it is also the precursor of vitamin A. Okay. Okay, so this is about the pigments. They are going to absorb the light. So when you look at this part, okay, I'm not going to write the definition now. So here we have two graphs. One, see, basically when it is PAR, it is in between 400 to 700 nanometer. So basically you are talking about the blue light here, which is very effective for photosynthesis, and red light. So if we have to compare blue and red light, it is actually the red light which is better than the blue light, and green light is least effective. You know, na, plants, they are going to reflect it. So now when you look at these spectrums, so you have two things. This one is the absorption spectrum. This one is what? This one is the absorption spectrum, right? Which spectrum it is, bache? Absorption. And this one is the action spectrum. One is your absorption spectrum and another one is your action spectrum. One is your absorption spectrum and another one is your action spectrum. These are the two words that we have here, absorption and action. So absorption means, like, it will tell us that this particular chlorophyll has absorbed this light of this wavelength, that much light. In action, they will tell us when the rate of photosynthesis is maximum in which strength, right? The rate of photosynthesis is maximum. When is it maximum? So it is like that. Okay, it is like that. So you people can see here. You people can see here from the NCERT only. Okay, I'll show you other graphs also. So pigments, they have the ability, right? They can absorb the light, and that too at a specific wavelength. They are particular about it, right? So now they are saying when we study the graph, so ability of chlorophyll A pigment to absorb the light of different wavelength, right? So we have the graph, we can see there. I'll pick up the definitions from this part only. So now they are showing that in the blue and the red region, they have higher rates of photosynthesis. So if I have to write the definition, so basically when we are saying absorption spectrum, when we are saying what? When we are saying absorption spectrum, so in the case of that, it is a graphical representation of absorption of different wavelength of light by the different pigments. Okay? So graphical representation of different of absorption of different wavelengths of light different wavelength of light by a particular pigment. By a particular pigment. Is that clear? That is the absorption spectrum. That is the absorption spectrum. So when it is the action spectrum, then what are you talking about? Right, this is the graphical representation which is telling us about the rate of photosynthesis of a particular pigment, right, in a particular wavelength of light. So I will just write rate of photosynthesis. That action spectrum is telling me about what? It is telling me about what? It is telling me about the, yes bache, right, it is telling me about the rate. Okay, so when we are talking about the amount of light, different lights that are absorbed, then it is action. And if there is the rate of photosynthesis at different wavelengths of light, then it is the action spectrum, right? How can we check this? This is also checked with the help of spectrophotometer. This is also checked with the help of spectrophotometer. What's that, people? It is spectrophotometer. What's that, people? It is the spectrophotometer. Is that clear? Is that clear? Yes, people, is that clear? So it is what? It is the spectrophotometer. Spectrophotometer. Okay, so this is what we have. Now let's discuss it from the graph itself. So when you look at this part, right, you have the chlorophyll A, you have the chlorophyll B, right, and even if I have to stick to the NCERT part only, so please observe it carefully. When you talk about your chlorophyll A, right, when you talk about the chlorophyll A, of course this is the blue light, this is the red light, so, and this is the chlorophyll B. So chlorophyll B is going to absorb more blue light in comparison to chlorophyll A, or we can say it in this way, that chlorophyll A is absorbing more red light than the chlorophyll B. One. And you can say both chlorophyll A and chlorophyll B, they absorb more blue light in comparison to red light. In comparison to red light, number one. And here you have the carotenoids. So you can simply see that chlorophyll A is having more blue light. It has absorbed more blue light in comparison to the red light. But if I compare it with chlorophyll B, chlorophyll A has absorbed less. So this is what you have to say. And now it is showing the rate. So if you will see the rate, it is also maximum in blue light, and then it is also maximum in red light. Now I told you red light is better than blue light. So see, red light's absorption is less, but comparison mein the rate of photosynthesis is more. Okay, the rate of photosynthesis is more. Are you getting my point? The rate of photosynthesis is more. So this is what we have studied, or we need to study here. Okay, then ye dekho, different wavelengths of light. Take different graphs we have here. Different wavelengths of light. Different graphs that we have here. So chlorophyll A, chlorophyll B, this. Okay, so these graphs, they are important. Xanthophyll, they all are shield pigment. This is what I told you. Now let's move to the next part, that is photosystems. Right, let's discuss the photosystems. After that, I'll tell you that photosynthesis is having two reactions. But before that, I would like to mention what quantum yield is and what quantum requirement is. Do you have any idea? Do you have any idea what quantum yield is and what quantum requirement is? What quantum yield is and what quantum requirement is? Yes. So what quantum yield is, what quantum requirement is? So quantum yield, number of oxygen molecules evolved, because this is what we have to get now, per quanta of light. So number of oxygen molecules that will evolve, right, per quanta of light absorbed, right, per quanta of light absorbed. So it is 1/8. Then there is the quantum requirement, right? Then there is one more thing, that is quantum requirement. These days we are not getting this type of question, but they used to come. So quantum requirement, my students, you are talking about the number of photons, yeah, quanta required to evolve one O2 molecule. So that is around 8. So that's why quantum requirement is 8. So number of light quantas which are required, right? Number of light quantas which are required to evolve one oxygen molecule, they are 8. ठीक. Now बच्चे we have to discuss what? We have to discuss the photosystem. Photosystems. When we talk about the photosystems now, right? When we talk about what? When we talk about the photosystems, they are basically the pigment molecules which take part in light reaction, है ना? That take part in light reaction, right? That take part in what? That take part in light reaction, right? In light reaction. So in the photosystem, you have two things. LHC, firstly, light harvesting complex. Firstly, write it down, light harvesting complex, which is also known as antenna complex. Then comes the reaction center. Then comes what? Then comes the reaction center. Then what do we have here? We have the reaction centers. Are you getting my point? So basically, what do you have? You have light harvesting complex, also known as antenna complex, reaction center. Always remember, your chlorophyll A will be there in the reaction center. Students, chlorophyll A is also known as the universal pigment because it is present in all the photosynthetic organisms. You'll see it in all the photosynthetic organisms. So we also call it as what? We call it as universal pigment. Is that clear? Hanji. So what do we call it? We call it as chlorophyll A is there, and here you have all the other pigments, all the accessory pigments which are also known as shield pigment. They all are present here. Now basically when we are using the word photosystem, right, जैसे LHC में you can have 200 to 400 pigments या proteins are present, right? Proteins are present. So here you have accessory pigments. Here you have what? Accessory pigments. So try to understand that. Overall, when you are talking about the photosystems, where are they present? They are present in the thylakoid membrane. Where do we see them? We will see them in the thylakoid membrane. Where are we going to see them? We are going to see them in the thylakoid membrane. Now here, these photosystems are nothing. They are just comprising these pigments only, right? They are just comprising these pigments only, right? They have these pigments only. That's what I'm saying. So all I'm saying is that when it is the light harvesting complex, also known as antenna complex, also known as accessory complex, right? Yeah, accessory pigments it is having accessory pigments, right? So in the reaction center, you just have chlorophyll A. So basically it's just like that. Here you have reaction center where you have so many chlorophyll A, but plus they are also surrounded with the light harvesting complex, and in this light harvesting complex also, what exactly do you have? You have other pigments which are also known as shield pigment or the accessory pigments. That is what you have. Now the point here is, my dear students, it's not like that ki chlorophyll A cannot absorb the light. It can, but it needs. Ultimately, why are we studying it? Because now we have to enter in a topic known as Z scheme, right? In the detailed lectures we have studied it, so I am picking up the main points. So it's not like that ki chlorophyll A cannot absorb the light, right? It can, or it cannot exude its electron. It can, but the point is the electron which is required, right? We need to remove the electron from chlorophyll A. We need to exude the electron from chlorophyll A. So for that we need more energy. So there are other light harvesting complex also, right? There are other, so here in the light harvesting complex, there are other pigments also. They will also absorb the sunlight and ultimately they will pass it to the chlorophyll A. And then chlorophyll A, because it needs more energy for the excitation, right, for the exuding of the electron, so then it will pass it. So that is how the things are, right? That is how the things are. So the pigments which are there in the light harvesting complex, they will also harvest the light at different wavelengths, and ultimately they will pass it to the reaction center so that electron can come out. So that is the role here. Okay, so reaction center can also absorb the light energy, but the frequency of excitation is very high, so it needs the light energy of different wavelengths. So you can see this here, photosystems. Now when we talk about these photosystems, we have two things. We have photosystem 1 and we have photosystem 2, right? This naming, जैसे we have photosystem 1 and we have photosystem 2. Beta, this naming is as per, it is not as per function. It is not as per their function, but it is as per their order of discovery. But it is as per their order of discovery. Take PS1 and PS2. Now, when you talk about the PS1, we also write it like P, it is P700. For PS2, we also write it like this, P680. P700 and P680. That is how we write, bache. That is how we write, bache. Yes, that is how we write. So basically it is showing what is the meaning of that. Anybody here in the class? So it means that PS1 contains a chlorophyll A, right? But it shows maximum absorption at 700 nanometer. Is that clear? Right? It indicates that it shows maximum absorption at 700 nanometers of light. So that's why we denote it like that. And it shows maximum absorption at 680 nanometer of light. At what? At 680 nanometer of light. Okay? At 680 nanometer of light. Am I clear? Are you sure? Are you sure? Are you sure? Right? So now, whenever we talk about the photosynthesis, please remember this thing, then we'll relate it. We have two reactions. One will be your light reaction. One will be your dark reaction. I didn't start with the definition of the photosynthesis, but I told you the carbon dioxide will be reduced, which will give you carbohydrates, and the H2O is it's basically the oxidizable compound, right? Water is there, the oxidizable compound, right? So we divide the photosynthesis into two reactions, two parts rather. That is how we study that, right? That is how we study the photosynthesis, that there is a light reaction and there is the dark reaction. There is the light reaction and there is the dark reaction. These are the two things that we have. So light reaction is also known as photochemical phase. It is also known as photochemical phase, and dark reaction is also known as biosynthetic phase. It is also known as biosynthetic phase, right? It is also known as biosynthetic phase. So students, it is taking place in stroma and it is taking place in thylakoid membrane. Now here, the dark word is a misnomer term. This dark reaction is a misnomer term. It doesn't mean that it will occur in the dark. No, actually, when you talk about the light reaction, right? So light reaction is there in the thylakoid membrane. Okay, right? Dark reaction, basically indirectly it is dependent on the light reaction. It is the light dependent reaction. It is light independent, but indirectly it is dependent on the light reaction. So light reaction will give us ATP. Light reaction will give us NADPH2, and light reaction will give us oxygen. So basically this ATP and NADPH2 that will come out from light reaction, they are required for the dark reaction. So if there will be no light reaction, the dark reaction will not take place, right? The dark reaction will not take place. Is that clear? Is that clear? So light reaction is there, dark reaction is there. So light reaction में photochemical phase है, ATP, NADPH2 and oxygen will release. This oxygen will go out of course, and for the dark reaction, what is going to happen in the dark reaction? Here the carbon dioxide is reduced. So it will take place in the stroma, right? Here you study about C3, C4 CAM pathway and all. Right, here you study about C3, C4 cycle and CAM pathway and all, and here you study about the Z scheme. Okay, phosphorylation, photophosphorylation, cyclic photophosphorylation, non-cyclic photophosphorylation. So under light reaction, that is what we study. Okay beta, that is what we study under the light reaction. So yes, we should know that what is present and where is it present, because here the photosystems are going to play a major role. Is that clear? Photosystems are going to play a major role. So definitely we need to know that, we should understand that. ठीक? So light reaction is also known as light dependent reaction or Hill reaction. Done? Right, so light dependent or light independent reaction is there. Okay, light dependent or light independent reaction is there, but again as I said, it is the misnomer term, right? Dark reaction also need, right, your this product of light reaction. Okay, so it is the light independent reaction. Dark reaction, it is also known as Blackman reaction. It is also known as Blackman reaction. Take, okay, right? So now let's understand the PS1 and PS2, which is required for the light reaction, then we'll go. So photolysis of water will not be there in PS1. Photolysis of water will be there in PS2. For reaction center is P700, P680. It is rich in chlorophyll A, then chlorophyll B. It is rich in chlorophyll B. Now molecular oxygen is not involved. So these are the points of comparison. But when will you understand it? Once I'll finish this topic. Okay? Once I'll make you understand that what exactly is going to happen. So first of all, you need to understand that this reaction will take place in the thylakoid membrane. What are we talking? Light reaction. I am saying this is the lumen, and of course in the surrounding we will have stroma. So students, here there are certain steps which are going to take place, but before that, again I need to explain you a few things. They say here you will discuss about phosphorylation. These are the terms. Firstly, let me make the terms clear to you. Photo means light. Phosphorylation means addition of phosphate. Phosphorylation means addition of phosphate. What is the meaning of phosphorylation? Addition of phosphate. So in the presence of light, addition of phosphate is there. Matlab in the presence of light, this is what you are going to get. You are going to get ATP. You are going to get energy currency. You are going to get ATP. You are going to get energy currency. Is that clear? Is that clear? Sure, bache? Yes, please. Is that clear? So this is the photophosphorylation. Okay? Now we have to understand the light reaction in detail. Right? Now we have to understand the light reaction in detail. So in the light reaction, my dear people, right? In the light reaction, what exactly do we have? So basically I told you about their position, and I told you that photosystems are placed. Okay? So basically, first of all, understand few things and then let's discuss it one by one. Alright, I'll tell you what phosphorylation is, photophosphorylation is. Firstly, I'll start it with the diagram. Just ये देखो, this is the diagram that we have. This is the diagram. Just see, some protein complexes are present. You have photosystem 2, you have quinine, plastoquinine, like these are the things which are present there. Look at the position here also. ये देखो. PS2, PQ cytochrome. So this is something that we have here first, right? This is something that we have here first. Okay, so basically, ये देखो, again, this is a thylakoid membrane, this is the lumen, this is the stroma, right? So you will see that here what we have to discuss, we have to basically form the ATP, we have to basically see that how that NADPH2 will form, and we have to see that how the oxygen will release. So this is what we need to understand. This is what we need to understand. ठीक है बेटा? And when we need to understand that, so we need to remember something known as Z scheme. You remember Z scheme? Do you remember Z scheme? Oh, I didn't add the picture. Yes, I have added it. Take, so this is what we need to remember here. Okay, this is what we need to remember here. This is the thylakoid lumen, right? This is the thylakoid lumen. Of course, in the surrounding we have the stroma, and here we have certain protein complexes. So whenever we talk about the light reaction, right, ultimately when you talk about that Z scheme, you are talking about non-cyclic photophosphorylation, that one by one we will understand. But before that, keep it in your mind, when you are talking about grana, grana is having PS1, PS2 both. Take PS2 will be there in the appressed region, PS1 will be there in the non-appressed region, but both are there. Now when you are talking about the fret lamella, that is stroma lamella, it will just have PS2. It will just have PS2. What will it contain? It will just contain PS2. Take, this is the point. Now, overall in the light reaction, first of all, we need excitation of reaction center. When you will understand it, na, excitation of reaction center and all, then I'll show you the comparison. Then we need photolysis of water. Photolysis of water. After that, what else do we need? The electron transport. And after that, there will be cyclic and non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. Now the point here is, see, first of all, you have to remember something. Basically, the way things are present. I told you PS1 and PS2, these are the two words, and it is as per the order of their discovery. PS1 was discovered first, PS2 was discovered later. So it was as per the order of discovery, but not as per their function. So whenever we start this now, we have to remember PS2. So now you do this with me, right? You do this with me, right? You have to remember it with me. Jaise PS2 hai. You know that P680 it is. Then, right, P680 it is. Then after that, what will be there? Yes, everybody, I need your support here because we have to start the photophosphorylation. I'll tell you the difference in PS1 and PS2 as well. But here, I'll be needing your support. I'll be needing your support to remember this. So, just मान लो, this is your P680. Just remember it for a minute, right? But whenever I discuss it, I discuss it in a different way. This is, suppose we have to start it with PS2. Your PS2 will get excited. Here you discuss about pheophyton, right? After that pheophyton, you will talk about plastocunin. That will form hydroquinone. Then there will be cytochrome B6F complex. Then there will be plastocyanin, PS1, ferredoxin, FNR. Then finally NADP, which is oxidized, that will form NADPH2. ठीक, मतलब it is something like that. It is something like that. Now, what is it? Beta, you know that when we talk about this chapter, we need to discuss about something known as Z scheme. Right? This is basically showing the zigzag. And this zigzag thing is as per the redox potential. It is as per what? It is as per the redox potential. So here, this Z scheme is also known as non-cyclic photophosphorylation. It is also known as non-cyclic photophosphorylation. What is it? It is also known as non-cyclic photophosphorylation. And now this is something that I have mentioned. I told you, first of all, you have to remember the sequence. Photosystem 2, then phaeophytin, then plastoquinone is there, plastohydroquinone is there, cytochrome B6F complex, plastocyanin, PS1, unknown electron acceptor A, then ferredoxin, FNR, NADP positive, NADPH. Firstly, you have to remember it. Again, PS2, phaeophytin, then you have the plastoquinone PQ, right? That will form PQH2, right? Firstly, remember it with me, right? Then I'll explain cytochrome B6F complex, plastocyanin. Then you have the PS1, ferredoxin, NADP reductase, and finally NADP. Matlab, this is how the things are. This is how the things are, right? This is basically the thing. This is how the things are. Now, let me explain this part to you. When you look at the diagram, right, when you look at the diagram part, जैसे ये देखो, this is the diagram part here. So PS2 is there, then plastoquinone. Look at the position here. Look at the position here. Then plastoquinone is there in between. Then after that, what do you have? You have cytochrome B6F complex. Then you have plastocyanin. So look at the position of plastocyanin. It is towards lumen. It is towards lumen. Then you have photosystem 1, then FES complex, finally ferredoxin, and FNR is involved here, and this is the oxidized NADP. This is what we have. That is how the things are. Now when you look at this PS2, near to this, right, towards the lumen side, it is having OEC. What is this OEC? It is the oxygen evolving complex. What is this OEC? It is the oxygen evolving complex. What is it, beta? It is the oxygen evolving complex. One more diagram I would like to show you here, that it is the oxygen evolving complex near to the lumen side. Then it is the photosystem 2. Phaeophytin is there. Plastoquinones are there. Right. Then finally cytochrome B6F complex. Then plastocyanin is there. Then cytochrome B6F complex na, so it is having a FESF, जैसे cytochrome B6F. Then plastocyanin. Then P700. Unknown electron acceptor A, then finally again ferredoxin, and from ferredoxin FAD, NADP reductase, and this. This is the sequence that you have to remember. So if you are discussing it for the first time, then yes, you might find it difficult, but actually it is not. Actually it is not, right? So basically, beta, point is that, see, firstly, one by one I'll break the terms and then I'll discuss it. Okay, I'll break the terms and I'll discuss it. So firstly, it is the non-cyclic. It is the non-cyclic photophosphorylation. What is it? It is the non-cyclic photophosphorylation. Now you know the meaning of photophosphorylation, but it is non-cyclic. Non-cyclic means like something has started from A, it will go to B, to C, to D, to E, like this. It will not come back. It will not come back. Now when it is the cyclic photophosphorylation, so it will be A to B, B to C, back to A. It is going to be like this. Then it is going to be like this, right? Then it is going to be like this. Now let's discuss the next point here. Let's discuss the next point here. So here, basically, bache, your photosystem 2, right? We have here redox reaction is going on. What is going on? Redox reaction is going on. Understand that. Redox, red, reductional division, ox, oxidation division. Oxidation, you know that loss of electron, reduction is gain of electron. So basically in this particular process, the oxidation reduction is going on. One will gain the electron, another one will lose the electron. This is how the things are. So basically, first of all, what we are doing here, right? What we are doing here, just say your photosystem 2. When you talk about your photosystem 2, which is there in the thylakoid membrane, right? Near to it, adjacent to it, OEC, oxygen evolving complex, is also there. So very first thing, what is going to happen, that your PS2 will absorb the sunlight, right? It will absorb the sunlight. It's not like that ki first PS2 will absorb, then PS1 will absorb. No, it's not like that, right? Literally it is not like that. It is simply like we have to show you this process, so that is why I'm starting it like this. So when your PS2 will absorb the energy, when your PS2 will absorb the energy, it will become excited. It has absorbed the energy, it will come to the excited state. So when it will come to the excited state, and you know that the other, in the light harvesting complex, other pigment molecules are also there. They have also absorbed the energy. They will ultimately pass it to the reaction center of PS2, and due to which it is getting excited and it will exude its electron. it will remove its electron, right? Now if it will exude its electron, matlab it has lost the electron, it will be oxidized then, because it has lost the electron, it has exuded the electron, then it will give this electron to the primary electron acceptor, that is pheophytin. It is also known as the primary electron acceptor. It is also known as the primary electron acceptor. Now what's going to happen, my dear students? Now, because it is in the excited state, right, it has lost its electron. So you know that we cannot, first of all, we cannot always be in our excited state always. We have to come back to the ground state. That is the reality of our life. That is the reality, right? We have to come to what? We have to come to the ground state. So now it will pass its electron. Now it will pass its electron to the pheophytin. So now when it will pass its electron to the pheophytin, right, this pheophytin will be reduced. And we have to bring this. Who's going to bring this photosystem to the ground level? Your oxygen evolving complex. So when you talk about your oxygen evolving complex, here what is going to happen? In OEC, there will be the photolysis of water. What will be there, bache? Photolysis of water. So breakdown of water in the presence of light. So here we have a reaction 2H2O, and it is Man Ka college, right? It is Man Ka college. What's that? It is Man Ka college. It is, what's that? It is Mn, manganese, calcium, chloride. Man Ka college. So this 2H2O will give 4H positive, 4 electron and 1 oxygen molecule. So let's suppose if instead of 2H2O, if it is single H2O, that is how it is going to be 2H positive, 2 electrons and half oxygen. So if we need to evolve one O2 molecule, right, so 2H2O is required. And bacha, one more thing, right? Initially, I just forgot to mention, there is immersion effect and red drop effect. There is immersion's enhancement effect and there is a red drop effect. There is immersion's enhancement effect and there is a red drop effect. So basically, when there is the red drop effect, what happened there? Matlab photosynthetic yield was less when single beam of lights were provided. Right now, after reading the Z scheme, you can understand that experiment in a better way. So when monochromatic, mono means single colored light beam was provided. Just let's suppose it was above 680. So the photosynthetic yield was less. It was above 680, matlab 700, matlab PS1 will absorb it. Now when it is the enhancement effect, when both the lights of both the wavelength, just 680 or 700, when both are there, so then the yield was more. Why? Because when you are talking about this complete process, you will consider PS2 as well as PS1. So we are starting the story with PS2 and reduction is going on overall here, right? And one more thing, just here everything is going as per redox potential. Do you understand what redox potential is? Anybody here in the class? You people read it, you can define it better than me, right? You can define it better than me. Redox potential, right? Redox potential, matlab ability of an organism to gain the electron. What is it? Right, ability of an organism, again, we are the biology teachers now, everywhere we are using the word organism. So basically it is the tendency of a molecule, of a moiety, right, of a chemical moiety, so that it can, acquire the electron or lose the electron. So it is overall the redox potential, isn't it? It is overall the redox potential. It is basically the tendency of a chemical species to gain electron and get reduced. Can get reduced. Redox potential की ability है of a chemical कि it can gain the electron and it can get reduced. Now when you talk about the redox potential, right? Now when you talk about the redox potential, here we discuss the redox potential as well. I'm not entering into the depth here. So redox potential, let's suppose this is the chemical compound, your chemical moiety A. This is the chemical moiety B. Now if this chemical moiety B is having high redox potential than A, then electron will go to B. Now let's say next is C. This C is having high redox potential, right? Then the B, the electron will go to this. So this is as per the gradient. Matlab, जैसे we in the osmosis we talk about the gradient ना, we talk about the gradient na. So this is something like that. Just one will have low redox potential, the another compound will have high redox potential, so the electrons will go there. So the electrons will go there. It is like that. So here we also consider the redox potential, and as per that, right, it is in this zigzag scheme. So I'll come to that point also. So I am saying that when PS2 has exuded its electron, right, it is in the excited state. We have to bring it back to the ground state, and it has given its electron to the pheophyton. So now it is oxidized, pheophyton is reduced. So overall this oxidation reduction is going on. So towards the lumen side, in this particular case, towards the lumen side, you will see oxygen evolving complex is there, and this oxygen evolving complex is also having an enzyme water oxidase. So what will it do? It will split the water, right, in the presence of light. So for that you need Manka college. For that, what do you need? You need Manka college, right? You need manganese, you need calcium, and other than that you need the chloride. You should know about it. Please write it down. You should know about it. So this is what you are going to get. So basically when you are breaking the water here, the protons will be there only. The protons, they are not going anywhere. They'll be here in the lumen only. They will be here in the lumen only. And what will happen to the electrons? They'll pass to PS2, they'll come back to the ground state. So here, the very first question that can come is that who is going to bring your PS2 to the ground state? Yeah, who will give electron to ground state? It is the photolysis of water, number one. Number two question is in the photolysis of water, which, right, what do we need here? Right, you need to mention these metal ions here. Okay? Now after pheophyton, it will pass it to plastoquinone. And then this plastoquinone will become plastohydroquinone or quinol. So basically here Q cycle is going on, but I'm not entering the depth. I'm not diving deep into this topic basically. So this plastoquinone, it is next to this pheophyton. When it will accept the electron, it will become plastohydroquinone, because it is not just the electron acceptor, it is also the hydrogen acceptor. Right, it is not something which will just transfer the electron, but it is also the hydrogen acceptor. So it will take the hydrogen from the stroma, right? It will take this PQ will take the hydrogen from the stroma. It will become PQH2. Now when it will pass its electron to cytochrome B6F complex, right? So now because this one is oxidized, this one is reduced now. Right, PQ, when it will accept the electron, this one is reduced, it will become PQH2. That is plastohydroquinone. And when I'm saying plastohydroquinone, I can also call it as plastohydroquinol. So it will also accept the H positive from the stroma. Now, when it will pass its electron to cytochrome B6F complex, it will pass it H positive here in the lumen. So ultimately here, what are we trying to do? We are trying to create a gradient. We have to create a proton gradient. We want more protons within the lumen than the outside. Now after cytochrome B6F complex, will pass the electrons to PC, and then it will pass to PS1. Right? Now here the point is, it's not like that. It's not like that your photosystem 1 is not absorbing the light parallelly. It is doing that. It is doing that. But the point is, here we have to show certain facts that electron movement is from PS2 to PS1. I'll come to this. So see, this PS2 will come to the ground state, right? The star is showing the excited state. Then it is the pheophyton. Then it is the plastoquinone, which will become plastohydroquinone. So when it will pass its electron to cytochrome B6F complex, it will pass that proton to the lumen. Then plastocyanin will pass it to the P700. So now, this is the photosystem 1. So photosystem 1 is also simultaneously absorbing the sunlight. It will also get excited. It will also exude its electron. And then it will pass it to the unknown electron acceptor A. And finally, it will go to FD, ferredoxin. So ferredoxin is also associated with FNR, ferredoxin NADP reductase. Ferredoxin NADP reductase. And then it will, right, reduce this NADP to NADPH. So now the question here can come like this. Who's going to pass the electron? How the electron flow is? It is from PS2 to PS1. So when PS2 is excited, who's providing it the electron? Photolysis of water. Okay. So when PS1 is excited, who's going to pass it the electron? Plastocyanin, Priyanka Chopra. It is like that. And finally, ferredoxin will give the electrons to the NADP, right? To the oxidized NADP, that will pick up the H positive from the surrounding, and that is how they have to create a proton gradient here. So this is the most appropriate one. So that is how you have to remember. So P680, it will get excited, pheophyton, then plastoquinone, which will become plastohydroquinone, then it will pass the electron to cytochrome B6F complex, then it will pass, when it will pass its electron to cytochrome B6F complex, it will pass the proton to lumen, then to plastocyanin, then P700, and finally it will get excited to unknown electron acceptor A, finally ferredoxin, with which FNR is associated, and that is how here you will see that NADPH will form, here you will see oxygen will evolve. So now we have to understand that how ATP will form. So quickly have a revision of that. Do it. Done? Take. So that is how it is. That is how it is. That is how the movement is. That is how the things are going on. Now, let's talk about the redox potential. Take. If you read the NCERT, they say that when PS2 passes its electron to the primary electron acceptor, that is pheophytin, this electron transport is uphill. Uphill means against the gradient. है ना, uphill means against the gradient. And here what do we have? Here what do we have? Yes, please. Here what do we have? Uphill means this PS2 is having high redox potential, pheophytin is having low redox potential, but still this is passing the electron to this, right? So this transport is only uphill, rest everything is downhill as per redox potential. Rest everything is downhill as per redox potential. So here, this is what we are going to see. This is what Z scheme is. This is what electron flow is. And that is how PS2 and PS1 is involved. Right? That is how PS2 and PS1 is involved. Now, take, so this is how the electron transport is. So these are the points which are given here, which are written here. You'll read it, you'll get to know about it. So this is the complete Z scheme, and that is what I have discussed. I told you about the splitting of water as well. I told you about the meaning of cyclic and non-cyclic photophosphorylation as well. But now I have to tell you the cyclic one. Right, that what exactly is the cyclic one, right? So basically here, let's have a quick revision. When there is the non-cyclic photophosphorylation, let's answer important question. PS2 and PS1 both are involved, right? Both are involved, yes or no? Right, it is the PS2 that will do photolysis of water, PS1 no, right? Now there is something known as cyclic photophosphorylation as well. Now when it is the cyclic photophosphorylation, when it is the cyclic photophosphorylation, it is cyclic. It will start from PS1 only. In the case of cyclic one, only PS1 is there. Only PS1 is involved. So can I say that it is taking place in fret lamella? Because it is the fret lamella where only PS1 is there. It is the fret lamella where only PS1 is there. So PS1, the electron will go, it will get excited, P700. Then of course it will pass it to the unknown electron acceptor, and ultimately it will pass the electron to FRS, ferredoxin reducing substance. After this, it will pass it to ferredoxin. From ferredoxin, the electrons will pass to cytochrome B6 complex, where it is having FeS also, cytochrome F complex. Matlab, that is how it is going to pass the electron. Right, so cytochrome B6 and cytochrome F complex. So FeS is there, you can simply write cytochrome B6 or F complex. After that, but hello, this is not the right way the electrons will go. So firstly, electrons will go to cytochrome B6, right? And then from cytochrome B6, that electrons will go to, right, bache? That electrons will go to your plastoquinone. So your plastoquinone will become plastohydroquinone, because it will pick up the H positive from the surrounding, and you know that then it passes its electron to cytochrome F. From cytochrome F, again that electrons will pass to plastocyanin C, then PS1. So that is how the cycle is, that is how the things are. Okay, right? So in the cyclic photophosphorylation, there will be no ATP formation, there will be no ATP formation, there will be no NADPH formation, NADPH2 formation, no oxygen evolution, only ATP will form, only ATP form, right? And this process occurs occasionally, only ATP, no oxygen evolution, no NADPH formation. So this is also a PY. This is also a previous year question. Take. This is also a previous year question. This is also a previous year question, and this is something you need to keep in your mind. This is the cyclic photophosphorylation. Now, cyclic mein only PS1 is there, so it will be in fret lamella. You know how the electrons will move. Now, on the basis of that, right, I'll come to the chemiosmotic hypothesis as well. We'll complete all the important points, right? So on the basis of that, now you will be able to understand the differences in PS2 and PS1 as well, because it is one of the most important topics. So now here I just said, right, now here I just said, please have a look everybody, that molecular, when it is the photosystem 1, it is not associated with photolysis of water. Isn't it true? Now you have read it, now you know the topic, so now don't you think that it is true? Yes, now don't you think that it is true? It is true, right? It is true. Now, photosystem 2 is related with photolysis. Molecular oxygen is not involved when it is PS1. Here, photosystem 2 mein photolysis of water is there, molecular oxygen will go. So this one will receive electrons from PS2, this one will receive electron from the photolysis of water. Okay? In this reaction, NADPH is formed, PS1 at the end, while in this reaction, NADPH is not formed. It can participate in both cyclic and non-cyclic one, but it will participate. PS2 will be there in non-cyclic one only. Is that clear? So it is there on the outer surface of thylakoid membrane, inner surface of thylakoid membrane. Theek? So that is how it is. That is how you can just focus on their differences. Right? Right? So uphill, downhill, the way they are going, I have discussed everything. Now, even if you will read this part from the NCERT, you will see that cyclic, non-cyclic, both, how will they form. So ultimately, if you will see why we have done that, because we need to create a gradient. We need to create a gradient. Just see, this is your stroma. Stroma is having low H positive. Low H positive means it will have high pH. It will have high pH. And inside there will be what? There will be low pH, means high H positive. So what is happening here, people? So see, this reaction is going on, a plastoquinone will form plastohydroquinone. So when it is forming plastohydroquinone, it is also the hydrogen acceptor. So it will pass the hydrogen inside, right? Then plastocyanin, which is located towards the inner side. Then FD or FNR. So you should know the full form of FNR. So NADP. This NADP, this is the oxidized one and it will be reduced. This is the oxidized one and it will be reduced then. So what is the difference in NADPH and NADPH2? Beta, when it will take 1 electron, 1 proton, NADPH, when it will take 1 electron, 2 electron, 2 proton, NADPH2. Right, 2 electron, 2 proton, then it will be NADPH2. So this is the chemiosmotic hypothesis. So 3 hydrogens, right? You know that this oxysome is there, CF0 is traversing there in the membrane, so CF1 is there in the stroma. So 3 H positive will pass and that will provide the energy to form the ATP. So you have to remember the proton gradient. You should remember that this is a high electrochemical potential gradient. Here it is high, here it is low. Just look at it, observe it. Done. So that is what we have discussed. So let's quickly revise it from this part, right? And this ATP NADPH will be there in the dark reaction. Okay, so C3C4 cycle, there is nothing much there, so you can, because I'll just complete this topic only because even number of students are also less, so don't worry. I'm going to put the short video sessions now for the quick revision so you can cover these parts. Okay. And even after that, beta, after that, we will have complete syllabus marathon. You know that there are cell cycle, we have discussed there in top 50 concepts. Okay? We have discussed that there in top 50 concepts. Okay? And even short videos are also there. Even we will have complete syllabus marathon. So ultimately we will cover these topics, right? It's not like that if something is not discussed now, it will not be discussed further. So for the morphology, for other chapters, short video sessions will be there for sure. We will discuss it as well. And the next marathon of complete, in the next marathon of complete syllabus, now I'll pick the topics from, I'll pick these topics only, the one which I am going to leave today. And yes, there is also the possibility I'll take the part 3 of the class 11th biology, where we will cover all the topics that we were not able to touch. Okay, right? So firstly, let's revise this important topic here. So when you talk about this part, chemiosmotic hypothesis given by Peter Michel, so they are saying is splitting of water molecule. It will be there on the inner side of the membrane, right? Due to which the protons will be there. The protons that are formed by the splitting of water, they will accumulate within the lumen of the thylakoid. They will accumulate within the lumen of the thylakoid. So as electrons will move through photosystems, protons are transported across the membrane. As the electrons, they move through photosystems, protons are transported across the membrane. So they are saying that primary acceptor of the electron, which is located towards outer side of the membrane, transfer its electron not to an electron carrier but to an hydrogen carrier. So basically they are saying that phaeophytin is going to pass it to the plastoquinone, which is not the electron carrier but a hydrogen carrier. So it will pick up the H positive from the stroma, and when it will pass its electron to the next cytochrome B6F complex, it will pass that H positive, right? The proton will released on the inner side of the lumen. So NADP reductase enzyme is located on the stroma side. So yes, they even ask the location of the enzymes, so keep it in your mind. Right, so along with electrons that come from the acceptor of electrons of PS1, protons are necessary for the reduction of NADP. So these protons are also removed from the stroma, my dear students, right? They are also removed from the stroma. Hence, within the chloroplast, proton in the stroma decrease in number. So in the lumen, their number will increase. Okay, so decrease in pH in the lumen will be there. So these are the important PYQs. So this gradient is very important, bache, because when we break this gradient, the energy for ATP synthesis will come out. Okay, so gradient is broken down due to the movement of proton across the membrane. That will be through the CF0. So CF0 will be there in the membrane. It is traversing. Okay, CF1 there is in the stroma. Right, CF0 is base, CF1 is your head and stalk. Take. So it is ATP synthase. It is also known as CF0 CF1 complex. It is also known as ATP synthase, right? So it consists of two parts. One is CF0 It is embedded in the thylakoid membrane, students, right? And it forms a transmembrane channel that carries out the facilitated diffusion of protons across the membrane, okay? So other proton portion is CF1. So this breakdown of gradient will provide the enough energy so that ATP will be synthesized. What will be synthesized, students? It is the ATP, right? It is the ATP that will be synthesized with the help of that. So this is all about this particular topic, okay? So ATP, NADPH2 will be used for this cycles further. So you know that C3 and C4 cycle is there. So even if I talk about the C3 cycle, so in the C3 cycle, beta, see, remember one thing, that you have to make C6H12O6. So if we have to make the glucose, for one glucose molecule, we have to take 6 turns of, right? How many turns of C3 cycle? 6 turns of C3 cycle we have to take, right? 6 turns, 6 times we have to repeat this. So there are 3 steps: carboxylation, addition of carbon dioxide, reduction, and regeneration. Because the C3 cycle is a cyclic process, so of course we have to regenerate the things at the end. So here we talk about the RUBP, that is a 5 carbon containing compound, ribulose bisphosphate. So because the word is ribulose, ol is the word, ketose it is. So it is going to accept the CO2. Right? So CO2 and water, then carboxylation. This is the carboxylation. So which enzyme is going to catalyze it? It is Rubisco. Right? So ribulose bisphosphate carboxylase oxygenase. It is the most abundant enzyme of biosphere. You know that. Okay, so if this enzyme, it is having its affinity is more for the carbon dioxide, but let's suppose if carbon dioxide is less, it can bind with the oxygen and it can result in the photo respiration, which is a not, of course, not required for the cell. Waste process it is. Then after this, right, you will get two molecules of 3-PGA. When we are saying 3-PGA, it is phosphoglycerate, it is acidic. Now here in this step of reduction, bache, in one cycle 2 ATP, 2 NADPH2 is required, right? And then triose phosphate will give starch. Here in regeneration, 1 ATP is required. So when you talk about one turn, when you talk about 1 turn of Calvin's cycle, or that is C3 cycle, 1 turn will use 3 ATP and 2 NADPH2. And you know that we need, for glucose, we need 6 turns. So 6 into 3 and 6 into 2, it will be 18 ATP and 12 NADPH will be used. Right? So that is what we have here. That is what we have here. That is how we discuss it. So you read it. I'll put the short video as well. You'll get to know about it. And I'll share the PDF in the Telegram group as well. You can access it. Okay, you can access it. And do not forget to watch these revision sessions. Bache, these days you are going to get so many videos for all of you. So we are working day and night for all of you. And I have planned the morphology revision session, a short video for morphology for frog and cockroach, right? Those who are targeting 360 out of 360, I know they are demanding these topics. And other than that, my dear students, even I will put the short revision sessions, morphology, frog and cockroach part, the trick sessions as well. Through shorts also you will get the tricks. So all we are doing, whatever we are doing it for your exam, right? We want you people to prepare sincerely. I am there for all of you. When it comes to biology, you don't worry about anything, right? In the top 50 concept, I have picked up the concept. I have picked up the most important concepts. In the marathon, whatever I was not able to cover in top 50 concepts, I have completed that in zoology marathon, and same in the botany marathon as well. And as I said, my dear people, we will, right, in the next marathon, in the complete syllabus marathon, I will definitely pick up the topics which we were not able to discuss today. Okay, so you don't worry about that. You just give your best. This is what we need from you. And if you people if you find this session useful, do let me know in the comment section.